A very good morning to one and all. Welcome back to the second day of Nobel Laureate Science and Technology Seminar Series, 11th India-Japan Science and Technology Seminar, commemorating 70th anniversary of India-Japan diplomatic relations and 75th anniversary of Indian independence, Azadi Ke Amrit Mahotsav, sponsored by Japan Society for the Promotion of Science, Japan, and Department of Science and Technology, Government of India. Supported by Embassy of India, Tokyo, Japan, organized jointly by Indian JSPS Alumni Association and Sree Chitra Tirunal Institute for Medical Sciences and Technology. The first session of today's lectures are chaired by Professor Babani Prasad Mandal, Department of Physics, Banaras Hindu University, Varanasi, Convener and Chairman of Indian JSPS Alumni Association North Chapter, and Dr. P.M. Guru Basavaraj, Department of Chemistry, Rani Channamma University, General Secretary, Indian JSPS Alumni Association West Chapter. We welcome you both to chair the session. We will introduce the speakers and the chair can coordinate the proceedings. Attendees may please quote your queries in the chat box during the course of lecture.
Hello. Okay.
Hello, sir.
Yeah, please go ahead.
The first lecture of today's session is by Professor Ramlakhan Singh, Honorable Vice Chancellor, Nilambar Pitambar University, Jharkhand. Professor Singh held the position of Professor of Biochemistry and Coordinator, Biotechnology Program at Dr. Ram Manohar Lohia Awadh University, Ayodhya. He has been conferred with Shikshak Shri Samman by Government of Uttar Pradesh. His area of research includes environmental biochemistry and biotechnology and nutraceutical biochemistry. We request the chair to invite the speaker.
Hello. Hello. A warm good morning and good afternoon to all of you. Myself, Professor B.P. Mandal from Varanasi and Dr. Guru Basavaraj from Karnataka welcome you to this second day of 11th India-Japan Science and Technology Seminar. And this session, we have three talks. And first talk will be delivered by Professor Ramlakhan Singh, Vice Chancellor, Jharkhand. And I invite Professor Ramlakhan Singh to deliver his talk. Sir, please.
Thank you, Professor Mandal and Professor Raj. May I be allowed to share my slides, please?
Yes, please, sir.
Please allow me to share my slide. Actually, it is not coming up. Yeah, thank you. So, Honorable Chairpersons, Professor Mandal and Professor Guru Basavaraj, distinguished scientists present over here, ladies and gentlemen. I am grateful to the team which is organizing the 70th anniversary of Indo-Japan diplomatic relations and 75th anniversary of Indian independence. This Nobel Laureate Science and Technology Seminar Series. And especially I am thankful to Professor Mohanan for giving me chance to be with you this morning. The title of my talk is Nutraceuticals, One More Step Towards Good Health. In fact, we all know that during the last 2 years in the COVID situation, we have gone through a lot of trauma, a lot of medication. A number of people used to suggest that one should take this medicine, that medicine, and most of the times we have been suggested by the people to use certain plant-based materials. I think almost 95 to 99% of the persons have once or twice at least used some or the other plant-based materials. Now, the plant-based materials, they have got certain biomolecules and they are really known as the nutraceuticals. Now, if we look at the food we eat actually, so the food, there are opportunities for expanded use of food and their components to achieve one's genetic potential, increase productivity and reduce the risk of diseases. The food materials, they can be used to cure or to treat the various diseases. And this is very well explained in the science of Ayurveda. I have shown you a slope from Bhaishajya Ratnavali, from Mangala Charanam itself, the beginning itself, which states that Ayurveda is the ancient system of medicine based on the idea of balance between bodily systems using diet, treatment, and yogic breathings. And as far as Ayurvedic medicines are concerned, actually it is totally dependent upon the balance in tridoshas, Vata, Pitta, and Kapha. This has evolved perhaps in the pre-Vedic epoch, 4000 BC to 1500 BC, and the very popular book on this has been written by Charak himself. The Ayurvedic philosophy of life is to live healthy life to avoid the appearance of imbalance and unnecessary pain. Now, more than 400,000 Ayurvedic practitioners are there in India who are practicing Ayurveda. Now, something about the xenobiotics before we come directly to the nutraceuticals, we can quickly look at certain factors wherein which are actually damaging our system, and that is why we require the nutraceuticals. The xenobiotics, they are basically the compounds which are foreign to the body, and they may be categorized into two, the exogenous and endogenous. We very well know that endogenous xenobiotics are pigments, hormones, etc. And exogenous, they contain drugs, food additives, pollutants, toxins, etc. And these xenobiotics, they are being metabolized or biotransformed in the living system. Now, there are certain sources of the xenobiotics. The petrochemical industries are there, the plastic industries are there, pesticide industries are there, and a number of pollution coming out from these industries, they enter into our system and they try to generate certain free radicals. They have the free radicals and they can cause a number of diseases, including the birth defects, kidney disorders, respiratory disorders, cardiovascular diseases, liver diseases, and it also includes aging, etc., etc. Now, they are being caused by the reactive oxygen and reactive nitrogen species, which are being generated out of the pollutants, and they include the superoxide radicals, hydroxyl radicals, peroxyl radicals, hydrogen peroxide, singlet oxygen, nitric oxide, peroxynitric, hypochlorous acid, etc., etc. And there are various sources of DNA damage, like the UV light, like the air pollutants, like the ionizing radiations, smoke, and there are certain indigenous sources of the DNA damage. That is the metabolites. A number of metabolites are there which can cause some damage to the DNA, and the inflammation also. So these damage to DNA is caused by free radicals which are generated out of all these activities. And very important body organs like gut, the brain and nerve, heart vessels, airways, they can be affected by the by the free radicals actually. And there is balance between oxidant and antioxidant. And once this balance disturbs, we can see in this slide that once the antioxidants they underweigh and the pro-oxidants overweigh, then certain they challenge our defense system, bodily defense system, and then they go on to cause the oxidative damage. Ultimately, they can cause one or the other type of diseases. Now, by way of going back to nature by using nutraceuticals, we can cure ourselves, we can prevent the diseases. Basically, nutraceuticals, it contains two terms, nutrition and pharmaceuticals. So they are actually the medicine sort of things, nutraceuticals, which can come from the nutritious sources, from the food sources, as we have been seeing throughout this period. And now the nutraceuticals have evolved beyond the tablets and capsule, and they have included newer modes of delivery, including the soft chews, chewing gums, gummy bears, patches, lollipops, quick dissolving strips, etc. and etc. Now, the basic difference is that pharmaceuticals, they say that the cure first and the follow diet next. Whereas the nutraceuticals, the principle of nutraceuticals is that follow diet first and cure next. So most of the times, as I have talked about the Ayurveda, they try to balance between the tridosas, Vata, Pitta and Kapha by balancing the diet, by balancing the activity. So that is how the nutraceuticals, they work actually. The nutraceuticals, they are essential food constituents that provide nutritional benefits as well as medicinal effect. And there are carotenoids, polyphenols, collagen hydrolysate, dietary fibers. They all act as nutraceuticals. Throughout the human history, many natural sources were utilized for their healing and strength restoring effects upon consumption, such as cinnamon, saffron, honey, garlic, etc., etc., which are still being taken up by us in our day-to-day life. The nutraceuticals, they are preferred because of the fact that people have faith, the consumers have faith that they are food-like substances and they may be having less toxicity or they may be almost harmful. Moreover, increased healthcare costs will also, they also divert the person to use the nutraceuticals. The new molecules, pharmaceutical molecules, it is very difficult to discover and it actually costs a lot of money, as well as it has got a number of side effects that can be the reason why people started preferring the nutraceuticals over the pharmaceuticals. They can be broadly divided into functional food, functional beverages, and dietary supplements. Dietary supplements, we have been taking vitamins, minerals, enzymes, fatty acids. These are the things which we have been taking in form of the pharmaceuticals also. Especially the functional food, the bakery, confectionery, dairy, snacks, etc., etc. They come under the functional food, and functional beverages contains the energy drink, sports drink, fortified juices, dairy and alternative beverages, etc., etc. So there are broad category of the nutraceuticals, we can divide them into. If we see the global nutraceutical ingredients, mostly the fibers and specialty carbohydrates, they occupy maximum portion, followed by the vitamins, then omega-3 fatty acids. Then comes the phytochemicals, minerals are also there, carotenoids and antioxidants, probiotics and prebiotics are there. So they all constitute, this is the figure of 2018 actually. Now, the functional food, there are certain examples of the nutraceuticals. Like the functional foods, they contain tomatoes, salmon, soya, fermented milk and milk products, and they contain the active ingredients like tomato has got the lycopene, salmon, omega-3 fatty acids, soya, saponin, and they have various antioxidant, anti-cancer activities, etc., etc. Some more functional foods are there, like the carrot, we know they contains the beta carotene, aloe contains aloins, turmeric, curcumin, marine algae, flucoidans, and they have various good activities which can be helpful in preventing the diseases or in curing diseases many times. And we all know the dietary supplements, these supplements we have been taking, folic acid, vitamin A, calcium, iron, vitamin D. So many people are taking these dietary supplements on the day-to-day basis. Then fortified foods are there, the recombinant foods are there, gold kiwis, etc. They contain ascorbic acid, carotenoids, etc. And they can have various advantages, if we take them. As far as the herbal drugs or nutraceuticals are concerned, basically, there is more than 70% of India's 1.1 billion population. They are still using the non-allopathic and herbal drugs. And almost 6,000 plant species, they are farming about 40% of the plant diversity of the country, and they are being used by the various practitioners, various companies to manufacture the nutraceuticals. Now, the current trends and future prospects of nutraceuticals, if we look at over the last 20 years, there has been a rapid increase in the use of nutraceuticals due to the more and more information we have about the nutraceuticals, about their use, about their good things, good activities. Then the marked side effect and ineffectiveness of modern pharmaceuticals compelled people to look for the nutraceuticals as alternative therapy. Then nutraceuticals for medicinal use has been justified on the basis that they treat disease caused by the deficiency of nutrients. So that is for the supplement food which we have seen over here, the dietary supplements. Now, the development of new medicines, they are more expensive and riskier, as I have told you earlier. And that is why, again, people are shifting for the nutraceutical use. The nutrigenomics is a comparatively newer branch where we study the effect of nutraceuticals and dietary components on the health of the particular individual. And in the figure, in this picture, we can see that how the nutraceuticals, how the food materials, how the plant materials, they can modify the genetic makeup of a human being. In fact, the nutrigenomics future is that we can develop the personalized food for the human being, and the DNA diet, like your gene decide what you like to eat. So that diet we can prepare for individual persons. There are n number of nutraceuticals which are available in the market. These are the brand names like betaine. They contain the carotenoid component and they boost the immunity. So immunity boosters have been very, very popular as far as this corona period has been, it is concerned. Then Zygold, Lipoate, Zenerol. So they all are the brand names of the nutraceuticals which are available in the market. Some more nutraceuticals are there: Z-Trim, Limum Life, Fenulife, T-Max. So they all have one or the other component which can have the antioxidant effect. They can protect the health. They can have some impact on the weight loss. They can reduce the cholesterol, etc., etc. Now, the nutraceutical market actually is expanding like anything, and we can see that this is a figure showing percentage worldwide share of nutraceuticals. So USA tops with the 32%, followed by the Western Europe, Japan, China, rest of Asia, etc., etc. So worldwide, not only in this Asian part, but worldwide, world over, the nutraceuticals are gaining momentum to be used by people more and more. The global nutraceutical markets, if we see, the most of the area is covered by the functional food, followed by the dietary supplements, and then followed by the functional beverages. So, and this is about $41 billion by 2020. Now, this global market size in 2020 has been valued at $417 billion, and it's expected to grow at a compound annual growth rate of 8.9% from 2020 to 2028. And there is growing demand for the dietary supplements and functional food, which is really responsible for this expansion of this global market. The Indian nutraceutical market is growing again in the same way. The data is up to 2016 only. And certain companies which are there in India, they are making nutraceuticals as well as some Ayurvedic drugs, the Dabur, Zandu, Patanjali, GlaxoSmithKline, Cadila Health, EID Paris, Himalaya, Amway, Semilabs, etc., etc. One very important thing here is that the latent market in India is 2 to 4 times the current market size. And it is between $2 to $4 billion with almost 148 million potential customers. So there is again the latent market in India, maybe in the world also, but in India, most of the times people are not, I mean, they don't go for the very branded things. There are n number of people, n number of practitioners who pick up the material from the forest or from the land itself, and they can use it. So that is why this is 2 to 4 times higher than the current market size. These are the international players in this nutraceutical markets: PepsiCo, General Mills, Nestle, Kellogg's, Herbalife, Cargill, Archer, DuPont, Damon, Royal DSM, Innophos. And the major things which I would like to point out here is that there are a number of issues which are related to the nutraceuticals, and these issues, until and unless not addressed, we cannot really go for producing good nutraceuticals. One is the quality, safety, and efficacy. See, if the agroclimatic conditions change, if the place change, the quality, the efficacy actually changes. Then standardization procedure is not very well there. They are being developed. There are certain standardization procedures, but they have to be strengthened actually. Then active compound isolation is not also very good at this point of time. Then structure activity relationship, it has to be established. A lot of scientific work has to be carried out. Then use of marker compound, use of sophisticated equipments, stability testing, accelerated and ambient temperature like the shelf life, etc. So these are certain issues on which we must concentrate to develop a good nutraceutical which can stand in the market and which can go to provide quality health supplements to the maximum population. If we conclude it, obviously the nutraceuticals, they embody a novel and accelerating research field, and yes, they have very good health effects, but we have to be very cautious about the safety and efficacy of this. Then globally, the researchers have realized the fact that proper nutritional dietary supplement can prevent and cure chronic diseases. This is all scientifically tested now. People are testing day by day. The testing is increased. Then the nutraceutical have proven efficacy in numerous diseases, including cancer, rheumatism, diabetes, and other chronic diseases. And further extensive research is necessary regarding their safety and efficacy. Furthermore, use of advanced and high throughput technologies can help us understand the underlying mechanism of action and develop this exciting area of research to new horizons for the betterment of humanity, both in terms of economic benefits as well as the health outcome. These are some publications we have published over the years.
Okay, so volume.
Some more publications. These are the books. This book is just coming in the next year. Actually, it is under the process. This has come in this year only. Advances in Animal Genomics, Food Safety and Human Health, published by the Academic Press, Elsevier. Again, one more book we have published, This is again advances in biological treatment of industrial wastewater, etc. This is our group, our collaborators, and thank you very much for patient hearing. Thank you.
Okay. Thank you, sir, for nice illuminating talk and educating us for our good health. So now we have time for around 5 minutes to for the questions. So if you have anybody has any questions, comments,
Please.
please go ahead. Please unmute yourself and go ahead.
It was very useful, sir.
Hello.
Online attendees, please quote your queries in the chat box.
So it seems no queries, questions, but let me ask one thing. So what is the status in Indian market and the nutraceuticals?
What is the
status in the Indian scenario? You saw the USA and other countries 2018 status. So what is the present status in India? The market
status? Actually, the Indian market, as I have submitted, that it has got a lot of hidden this thing actually. So almost $2 to $3 billion is Indian market actually, which I have shown already in this slide. In fact,
I have one question.
please, sir.
Yeah.
Hello?
Go ahead, please.
Yeah, I would like to know what is the difference between nutraceutical and phytopharmaceutical. So is there any hardline differences there?
Not really, actually. They both are sometimes synonyms, in fact. And nutraceuticals, basically, if you take out some biomolecule or there, I mean, nutraceuticals are more purified, rather, I would say, in comparison to the plant materials which you refer. So this is the only difference. Many a times they are taken as same, actually.
And as far your toxicity is concerned, so in many cases, this toxicity profile of the nutraceuticals and phytopharmaceuticals are not well established, right?
Yes, very right. You are very right, sir, actually. It is not well established, and that is what I have mentioned over there in the concerns, in the issues about nutraceuticals, that these should be actually tested, standardized, and then only the things should be released. So there are, because once you change the climatic conditions, the materials over there, the biomolecules over there, they change. Their efficacy may change, their shelf life may change. So we have to be cautious as far as the toxicity of the materials are concerned.
Okay. Okay, thank you.
Sir, there is one more question.
Please.
What is the technology aspects of what is the nanotechnology aspects of nutraceuticals?
Actually, this people are thinking of it. They started doing something, but still it has not come out. In fact, we have to have very sound background unless we go for the further research. So the backgrounds are being prepared, and then we can go for the nanotechnology portion. So some people are thinking of it, some people are trying it, but there is no, perhaps not very prominent research which has come out of this nanotechnology aspect of nutraceuticals.
Okay. Any other questions?
So there is again a question from Manoj, from Mr. Manoj. Is there any clinical trials which are required to establish the efficacy and safety?
Certainly, yes. Certainly, yes. They are being done. Clinical trials are being done, and without clinical trials, see, once upon a time during the period of Charak, it has to be they used to test on the persons. They started giving the medicines. I mean, giving the plant materials to the people, and then they used to try, see the effect. But nowadays, yes, there is established process, and they are all scientifically tested, and they are all being scientifically done on the animal system or the bacterial system, whatever system of toxicity testing is there. So it is the acute toxicity, it is the short-term toxicity, it is the chronic toxicity, it is the special toxicities like mutagenicity studies, like teratogenicity studies. They all are being performed before the release of a nutraceutical. But most of the times we consider it as safe because of the fact that they are the plant materials and the materials which we have been using for years, for generations we have been using those materials. So that is one reason. But yes, whatever nutraceutical materials are being floated, are being produced by standard firms, they go through all the tests of the toxicity clinical trials.
Okay, maybe we should end this question answer for this speaker. So let us join with me to thank Professor Ram Lakhan Singh for his nice talk.
Thank you, sir. Thank you, sir.
Okay. So over to, yes.
Thank you, Professor Singh, for your informative lecture on nutraceuticals. The next speaker for this session is Professor Shiva Umapati, Director of the Indian Institute of Science, Education and Research, Bhopal. He is an Indian laser spectroscopist and known for his studies of molecular dynamics using Raman spectroscopy. He is a fellow of Royal Society of Chemistry and an elected fellow of Indian Academy of Sciences. He was awarded the Shanti Swarup Bhatnagar Prize for Science and Technology, Government of India, for his contributions to chemical sciences. We request the chair to invite the speaker.
Okay, thank you. So, yeah, I invite this Professor Shiva Umapati for his talks on laser spectroscopy from physics to chemistry, biology, and medicine. Sir, please go ahead.
Thank you, Professor Mandal. Thank you for the nice introduction. It's my pleasure and an honor to be on this Indo-Japan seminar, Nobel Laureate Science and Technology seminar. And of course, celebrating 70 years of India-Japan collaborations. I've been traveling to Japan since 1983. So, it's been a very long association with Japan. And they're very close friends in Japan. And so it's an honor and pleasure to speak in this meeting. We'll start our slides. So I work on laser spectroscopy. Put the first page. First page. So my title is Laser Spectroscopy from Physics to Chemistry, Biology, and Medicine. So I intend to show you some examples of applications in basic biology and some development of biology, and then plus examples of medicine. And then finally, if we have enough time, we can probably look at a new technique that we have developed in India using laser spectroscopy. Now, when we say laser spectroscopy, what we mean is that when you use a laser and impinge upon any chemical material, let's say here I'm showing methanol, you have light absorbing and light emitting, and you also have scattering. So we actually, in this particular talk, I'm going to talk about only Raman spectroscopy, which is related to scattering of light. So if you measure the frequency of scattered light, you can actually identify the chemical bonds that are present in the chemical that you're studying. So in this case of methanol, you can actually see CC bonds, CH bond, OH bond, and CH bending. So the various vibrational frequencies can be seen. For example, if you take these three atoms, they all can move frequently different ways. Each movement is associated with what is attached to these three atoms, and therefore the rest of the molecule will determine the frequency of these movements. So by understanding these frequencies, you can actually learn about the dynamics of a structure or any small changes in the chemical structure using spectroscopy. And that is our basic principle with which we do. So I'm going to talk about application of lasers to biology in the first part. We have a lot of work we do in many, many different areas, starting from tissues, drugs, liver injury, sepsis, neurons, nerve injury, muscles, and also bacteria. Now I'm going to show you one or two examples of tissue studies. If you, for example, look at brain cancer has different grades of brain cancers. When the grade goes to 3, sometimes it's difficult for doctors to decide whether it is the grade 3 or grade 4 or grade 2. So it's a very complex business. So when pathologists discussed with us as part of the team, they said that is there any way that we can make it very objective to look at whether it is grade 3 cancer or not? Why it's so complex? For example, what you see here, if you take a cancer tissue, brain cancer tissue, what is called glioblastoma, you can see that although all the three are graded as grade 3 by a group of pathologists, but they, sometimes the pathologist might think that it is probably grade 4. The difference between grade 2, 3, and 4 depends on the dosage of chemotherapy and radiotherapy. So if somebody is grade 2, if the doctor identifies it as grade 3, then they'll give excess dosage of medicine and radiotherapy. But if somebody is grade 4, if you identify as grade 3, then there will be less doses than necessary in terms of radiotherapy and chemotherapy. So it's a serious problem in the pathological area. So my friends always tell me that when cases like that come, a sample is sent to 3 or 4 pathologists, and then they meet and discuss and make a common decision. So they wanted me to find out, is there a way in which we can objectively identify using spectroscopy? Now, if you look at biology, they are nothing but chemical compositions. I mean, if you look at a cell, you have membranes outside, you have nucleus inside, you have DNA, RNA, cholesterol, and cytoplasm contains a large number of proteins of different kinds of RNAs. So we can study all of them very accurately using spectroscopy. So what we do is we record a spectrum using this. For example, what you see on the left side top is a crosswire. At every point of the crosswire, I can record a spectrum, which means in this screen that you're seeing, something like 750 micron by 750 micron, I can record 40,000 spectra at every point, which gives each point reflects different composition of chemicals present in the tissue. And that is what we study. So we do an analysis of all of these. What you see here, for example, different types of tissues, what is called fibrillary, giant lipidized tissues, small cells, pleomorphic cells in the tissue, and of course the normal tissue. So all these look very different, but then we can do a computation, and what you see here is an algorithm that we have developed. You can see that each algorithm is very unique to the particular property. So by looking at one particular pleomorphic, you can see the ratio of the frequencies at 1240 to 1312. The amplitude must be fixed for a pleomorphic system. And you can also look at the 1338 frequency must go up. So by looking at these kind of unique markers, what we call spectral markers, the computer can identify the material what you're seeing is pleomorphic or fibrillary or lipidized. So once we finish this, we ask the medical doctor to provide us samples. So they gave us some 15 samples of unknown and asked us to predict it. So we actually predicted, then we had a meeting. They brought the samples on based on their prediction. It was 100% match with respect to computer prediction versus what they predicted. So the next step we had to go was to see, can we actually do this to look at a structure of whole of images of cancer tissue. So if we have cancer tissues of 4 micron thickness, if you cut them into a cryotomid to make into thin pieces of slices, we can record all the slices as an image and then computerize it, put it through as a 3D material. And then you can actually talk about propagation of cancer, how long, what is the thickness of the cancer, diameter, how deep it is, and therefore, what is the potential of this being a 3rd grade or a 4th grade in one section of the tissue versus another section of tissue. And this is, for example, one particular example of the kind of projects that we have done. I'll give you one more example of, this is an applied, which is now in the process of being in the clinical trials, trying to see, can we actually do this commercially? We are not there commercially yet, but we still need hundreds of thousands of data before we can go commercially on the tissues. Now I'm going to talk about another work that we have done. What I'm talking about today, all of them are published in the literature, so you can always get a reference for these papers. Now, when you talk about cancers, people always use drugs, chemotherapeutics, right? Chemotherapy treatment, which are drugs. And one type of drug is called HDAC inhibitors. HDAC means histone deacetylase inhibitor. Don't worry about the chemical name. The idea is that it's a chemical that actually controls the growth of cancer. Now, histones are kind of proteins. It's a nuclear protein, and surround which you have DNA coiling around. And so these histones actually play a very important role in trying to maintain our body in terms of killing cancers and any other cancerous material that comes in. And there are a large number of different types of histones. These histones are classified by the biologist, and there are inhibitors which are being given to cancer patients to identify the cancer growth and reduce the cancer growth. What is the principle of this? Is that, for example, when you actually induce in a typical cancer system, what you see here, the equation going towards the left is actually becomes too much. So we have to inhibit them. Normally in a normal person's life, acetylation, which is going towards the right, and deacetylation coming towards the left, is a balance maintained by histones. Now, when the cancer growth is too high, then deacetylation becomes too high, and therefore cancer continues to grow. So these drugs that we are talking about are called the deacetylation inhibitors. So when you put those inhibitors, it reduces deacetylation. Then you maintain a good balance of acetylation and deacetylation, so you can kill the cancer growth. So that's a principle as much as biologists have learned. Now, what is interesting, we want to see there are different kinds of medicines. What you see here on the screen are different kinds of medicines used. One is valproic acid, one is apicitine, citrinol, all different kinds of drugs. Now, all drugs don't work for everybody because different drugs work for different people because people have different kinds of cells in their system. So one has to see which drug is good for whom or which drug is efficacy for a particular cancer. So this, for example, is an experiment we carried out. What you see here is a control experiment along with the solvent, plus also with various types of drugs. And if we actually look at very closely, we can actually learn about whether the acetylation, deacetylation function is working or not. So if you see on the right, I've given 5 different cell lines. Those 5 different cell lines are equivalent to dealing with 5 different people who may have different kinds of treatment, different kinds of response for different cancers. So what we did, we carried out a series of experiments to show that we can monitor very closely whether a cancer drug is effectively functions or not. But what is more interesting in this particular case, the biologists always believed that what they are seeing is only acetylation. During our experiment, we found in addition to deacetylation, we also monitored depropionization. So then that was a revealing for the biologist. So when I told them that we are also seeing propionization, they went and did an experiment. That's what you see here on the left. It's an assay experiment to prove what we are seeing is correct or not. And it turns out that they learned something new, that propionization also is happening in the histones, and therefore the depropionization is actually also an active property in biology in addition to deacylation. So what have we learned in the process? We learned that propionization also need to be monitored, and there is a strong correlation between propionization and cell death. So which means that we can control, depending on the cell type, depending on the drug, we can control the cancer. So this also demonstrates that this could be a good drug screening methodology before somebody can be treated. These facilities are available in Europe and US, but the problem is that once we identify, it takes 3 or 4 days before they can decide whether a drug is good for somebody or not, by taking a small piece of a tissue, and then they remove the cell from the tissue, and then those cells are cultured, and then drugs are screened. And we can do the same now using spectroscopy, which will only take about 1.5 days rather than 4 or 5 days. So this is the advantage of this technique that we have developed in this case. So now I'm going to talk to you about a real life application where the technology is being implemented. We have developed this and patented it. Sepsis is an infection disease. It's a life-threatening disease. Now what happens is it leads to organ dysfunction, and because the host doesn't respond to the infection. Why does it happen? Because it normally happens in ICUs when somebody gets into operation theater, and then after the operation, the patient is brought to the intensive care unit in a hospital. During the stay at the ICUs, suddenly sometimes somebody picks up this infection and then they die. And this is almost 50% of death in hospitals are because of these ICU deaths. Now, why does it happen? Well, mainly because you can't predict sepsis so easily. Normally, most of the doctors identify sepsis only by their symptoms. So when the patient suddenly develops some sort of an issue, then they actually search for whether it is infected or not. So our doctors were saying, doctor colleagues, if we can predict infection before the manifestation of the symptoms, then it'll be much easier for us. So I said, okay, let's work together. So this is the different grades of infection. Local infection is what normally you get by taking tablets and stuff. So it goes away, you get inflammation, and then you can clear them. But in the case of sepsis, the infection leads to organ failure. That means that the sepsis starts functioning, the infection starts affecting various organs. And when it goes to septic shock, if you don't monitor it sufficiently, if we go to it's hypotension and then hyperlactemia, and 99% of the time they die after septic shock. So they have to predict this much earlier. Now, what is the existing method? The existing method involves culturing the sample, taking the sample, trying to figure out what kind of infection it is. So it takes about 2 to 3 days. And that's a long time before somebody can be treated. And so doctors start giving them random antibiotics, assuming something. Other method is to occasionally look at, sometimes serum lactate levels can be seen, sometimes C-reactive proteins also can be observed. But it's very, very slow and time-consuming process. So can we do it much quicker? So this is what we did. So what we did was we took the blood plasma from the patients. But I must tell you that what you're seeing is the final result. This is the work of 4 or 5 years. Initially, we started with animal models, induced infection, and then we looked at different parts of the animal, from tissues to spine to various aspects, in addition to blood plasma. And we found the blood plasma gives you reasonably good concrete signals of sepsis. Then we went to the human model. And what you're seeing is the data from the human model. So what we did, we took samples from healthy individuals and from ICU sepsis individuals and also septic shock individuals. And when you take blood, you can, within 5 minutes, we can tell you whether the person is infected or not. This is normally, we can even see this before the symptoms appear, 12 hours before the symptoms appear, because the symptoms take time to appear. And if we can predict beforehand in the ICU that this person has got infection, but in another 10 hours, you start seeing problems, then the doctors can start treating them before the symptoms appear. And that's what we became successful. And it's not straightforward to do this because it's very complex. we are talking about frequency and amplitude, which is the spectrum, what you see on the left side. So what we do is we use artificial intelligence using convolution neural networks by multi-layer learning process. And then we demonstrated that you can clearly see when a sample is given, whether it is a non-sepsis or sepsis or septic shock. And you see that how it can be classified very accurately without any overlap from each other. And this is in one angle. So now this is now instrument is being made to be deployed in hospitals. And we're going to be now trying to use this technique in the hospital to save lives in the next year or two. We are going through clinical trials right now. So this is, for example, one direct application which goes to the immediately. So next I'm going to show you in the next 5 minutes something about what we have done in terms of trying to find something inside. Now scattering, you always have learned that it's always from the surfaces. And if it is from inside a bulk system, it may not be so easy to find out. So when we were looking for this, the question is, how do we make sure that can we look at particle scattering, which is already inside another set of particles? So we have to learn about diffusion of photons. And there is a lot of studies on Rayleigh scattering applications to do imaging. For example, your X-ray and other tissue examples of tissue densities that you measure using various other ultrasonic methods. So there are already lots of principles known about frequencies being measured at deep parts of the tissue. But nobody has studied, for example, near absorption across tissue is also known. People do brain scans using near infrared to understand the distribution of blood. But there is no measure of any molecular structural information that you can learn. So all these techniques like MRI, CT scan, all of them give you density of tissue or density changes inside the part of the body. But none of them give you chemical composition. So we wanted to see, can we actually use Raman spectroscopy to go deeper inside the body and look at what is there? So that's our principle we started. Our idea is simple. So if you actually put a beam at the entrance of the outside of the substrate, you will have Rayleigh scattering and Raman scattering, and some of them will go to hit the next particle and go much deeper and lead to scattering. So we can measure the scattering from various particles by measuring at different locations around the sample. So this was the principle we started with. So instead we wanted to show, let us say if I have 3 layers or 2 layers, if I put the light in the front part of the layer, can I see what is behind the front layer? And similarly, if I have 3 layers, can I see what is behind the 3rd layer? So that's what we did. You see here now experiments where we could see what is in the front and what is in the back very clearly. So that's what we did in the first set of experiment. Then we became much more bolder after doing large number of experiments in different configuration. We learned that we can actually start looking at what is inside a bottle, even if it is sealed. So, that is why, for example, you can actually carry explosives in water bottles, and therefore they don't allow you to carry a water bottle to the aircraft. Now, in our technique, now we can penetrate these bottles, irrespective of the color of the bottle. I can tell you what is the chemical composition inside. And for example, here I'm measuring hydrogen peroxide and all sealed bottles. We can see that we can go down up to 5% of hydrogen peroxide mixed with water can be easily identified using this technique. So we can penetrate these bottles inside and tell you what is the composition. Now, once if we say that, can we actually look at then envelopes or anything that is hidden behind a cardboard? And this is, for example, a set of experiments we did. We packed ammonium nitrate packed in a cardboard. So, for example, most of the terrorist activities involves packing ammonium nitrate with other explosives, and either in a cardboard box or plastic boxes. So our idea is, can we look through the plastic boxes and stuff? And this is a demonstration that can be irrespective of the color of the box and the material hidden. We can actually tell you what is inside without opening it. So this is one example of a data that I have. So now that we have done this, now we get into bulk material. So now we decided, let's say I have a bulk of 3 different chemicals, one on top of each other. And do I have to scan the whole place, or can I look at it from one place? So if we put the light, for example, in the middle, you can see at the center, the green arrow. If I put a small beam, laser beam there, the light actually propagates in all directions and keep moving, diffuses through to various parts of the system. So if I go and measure the signal that's coming out on the top end, which is called DCB, although the light is at least 2 centimeters away, I can still measure what is the signal that's coming out on the other side of DCB. And I can measure and tell you what is the chemical composition that is present there. Here I know it is DCB, which is a chemical. Therefore, this is a calibration experiment. But you can also put unknown, and then I can tell you what it is if I have this database to identify what they are. So this is very successful. Then we realized that we can penetrate light through much deeper systems. So we decided to go into looking at shapes which are hidden. So what you see on the left side is two chemicals. One is light blue, which is a pellet that we have made. Another one is outside chemical, which is covered with it. So what we are seeing is the ammonium nitrate is the light blue, and the trans-stilbene is outside. So what we are trying to do is trying to measure the signal of what is on the top and what is at the bottom and what is in the middle, and then we can decipher the size and shape of the location. Now that we tried this shape, which works well, then while I was discussing with one of my colleagues in IISc, he said, oh, this is because it's big bulk. That's why you can see, if I separated the bulk, can you see it? I said, okay, let's do that. So we actually went and did this. For example, you can see in both top and bottom, different kinds of shapes that we identified. And we can actually tell you the length and width and the shape of the hidden material. Although you cannot see it by taking a picture from outside, but by measuring scattering, because the outside signal will mask in your photograph, but we can tell you the chemical composition which are hidden inside the system. Now you can see very accurately by tens of microns, we can actually tell you what is the thickness of the material as well. And that is the advantage of using spectroscopy now. Having done that, we decided to go into do one more interesting thing. So we wanted to hide a material inside a tissue. So what we did was we take a chicken breast tissue, which was about 5 to 6 centimeter diameter. We drilled a hole through the middle and then went and hid trans-stilbene pellets. And then we tried to scan and get the signal. See, what you see here is identification of trans-stilbene material in a chicken tissue, which is a 3D image reconstruction of what we have seen. What I'm showing you is the shape of the material that we measured. What does it tell you? It tells you that it is possible in the future. We can go into 3D Raman tomography, like actually we do MRI right now. As of today in my lab, we can go up to 12 centimeter depth with accurate prediction of within hundreds of microns to see what are the chemical composition. So many of our technologies, these are all patented. We have 5 US patents on all these techniques, everything. So we can actually apply them to various applications, from pharmaceuticals to forensics to biomedicine. And that is our dream, which hopefully in the future we will work on it. And the applications are here. You can identify breast cancer is something we are developing now. And surface structural studies, for example, bones and blood, without non-invasively, without cutting. It's like taking a picture, but you can look at chemical composition changes. If a bone is bone marrow, or a bone is destroyed or reduced in terms of weaknesses of phosphates, we should be able to study without having to open the wound. And this is what is being developed at the moment. So I should thank my collaborators and industry partners. Boeing, ITC, Unilever, Procter & Gamble have funded many of my projects. And you can see on my right are my collaborators from various departments of the Indian Institute of Science. Also in hospitals nearby, Mental Health Hospital, Kidwai Cancer Hospital, and Ramaya Hospital in Bangalore. Of course, nothing is possible without the students, because they are the ones who do the hard work. And I must thank all of them. And then thank you for your patience listening to me for the last half an hour. Thank you.
Okay, thank you, sir, for your nice illuminating talk. Now we have couple of minutes for the question. So anybody having any comments, question, please go ahead.
Excellent presentation, sir. Sir, I have a query. You mentioned about this detection of sepsis. So can be directly detected from the blood, or is it necessary to separate the plasma for the detection?
Right now, we have to separate. We are working on direct detection from various parts of veins and other parts of the body. We are working on it because to separate the signal, basically what we are seeing is protein degradation and others. And so we need to understand the signal intensity and noise levels. So we are still working on it. And hopefully in the future, in the next year or two, maybe we'll be in a position to comment on possibility.
Yeah, thank you, sir. Because if we can directly detect, there are a lot of other applications also. That's what.
Correct.
Yeah. Thank you, sir.
Thank you.
Any questions in the chat box? Is there any questions in the chat box?
Sir, I have a question.
Yeah.
So your presentation was very good, and I just was curious whether the UMARS method is available as a device as of now, or
Yes, we do. So I've already given two demo systems, one to Defense DRDO lab in Bangalore, which works on bacteria. So we have a portable system which can give you 100 times more signal than the commercial system, which is only about 8 kgs. So we have already deployed in DRDO Bangalore. And then there is one more DRDO lab in Pune, which is taken for explosive detection in packages at distances, 2-meter distance.
at one point of time, the anthrax was the material which was going on in envelopes. So is it possible to detect that also?
Yes. Actually, the Prime Minister's office got in touch with me, and they want a way to scan all the envelopes that are coming into their offices. And we are in discussion because this requires a supply chain kind of system, because they don't have time to sit and see everything. So we automatically scan the envelopes before it gets opened up. So we are in discussion. Yes, that's right. Anthrax and any other pores, the bacterial pores which are dangerous to humans, can be sent by post. Yeah.
Okay. So I don't think any other questions. So let us thank Professor Shiva Umapati for his nice talk. And I hand over to my colleague, Dr. Guru Basav Raj, for conducting the third talk.
Video. Yes.
Thank you, sir, for your valuable insights on laser spectroscopy and its applications in various fields of science. The final speaker of this session is Sree Rajamanickam IAS. Sree Rajamanickam IAS is the Managing Director, Kerala State Industrial Development Corporation, Trivandrum. Sree Rajamanickam, the recipient of the Best District Collector Award in 2015, is also credited with publishing lots of articles on public policy and administration. His involvement in community participation is well appreciated and duly recognized by United Nations Development Programme State Youth Volunteering in India Action Report. We request the chair to invite the speaker.
Guru Basav Raj, please unmute.
Excuse me, Doctor.
Yeah. Okay. Guru Basav Raj, please unmute.
Unmute.
No, we cannot. Okay, so let me invite Okay, let me invite Sree M.G. Rajamanikam, Managing Director, Kerala State Industrial Development Corporation, for his talk on medical devices. Sir, please go ahead with your talk.
Yeah, good morning, Professor. Can I get the permission to share my content because it's not enabled?
I think they will. Yeah. Now it's okay.
Yes. Is it visible, sir?
Yes. Yes.
Okay. So, very good morning, one and all. It's like really, it's a very great, opportunity and pride for me to talk on this Nobel Laureate Science and Technology Seminar Series, which is organized as the 11th Indo-Japan Science and Technology Forum, commemorating 70th anniversary of our diplomatic relations with Japan and 75th anniversary of our Indian independence. It's really very, great opportunity for me. And thank you so much for inviting me too. See, as far as my topic is concerned, like I'm just going to present, medical devices, a new sunrise sector for India. I'm really thankful to my previous two speakers, especially Professor Sivo Mahapati. It's like very inspiring speech, inspiring presentation. Both of them had done it. And because whenever we are talking about healthcare sector, like we say that healthcare is itself is a major sunrise sector. But the belief is strengthened by professors. My previous speaker's speech is like definitely it's a kind of a scope. A lot of scope is there for healthcare sector, especially for India. And see, as far as healthcare sector's industry is concerned, which is very tremendously growing in India, as everyone knows it. Actually, it is because of growing incidence of lifestyle diseases and rising demand for affordable healthcare delivery, which has been like whatever the professor previously mentioned about is technology and development and technological advancements. And in addition to that, a lot of insurance opportunity, government initiatives like Atmanirbhar package or e-health or whatever. These are all the things packed together has created a kind of healthcare itself as a sunrise industry, among which, like, if you take it as a medical devices, I don't need to tell. Today is like, anything is, considered as a technology as far as medical devices are concerned. It is again one more sunrise, sector as far as world is concerned. India is also concerned. If you take medical devices itself is like, anything is a medical devices today. Is like technologically simple items such as even ice bags or tongue depressor to the sophisticated cardiac pacemakers and proton therapy devices. Anything can be one end to the other end of the spectrum can be devised as a medical devices. This sector, especially medical, devices sector, present and wonderful, opportunity, especially landscape for both domestic as well as international, manufacturers. And it is growing very steady phase in India. That is a very important thing, which has been very, well explained here. If you take, early 90s, the medical device sector, as far as India is concerned, is completely concentrated by the domestic players actually. But post new economic policy sector is open, and today is 75% of our Indian medical device sector is highly import dependent. It's like 75% and plus are import dependent actually. And if you see this one, the current market size of the Indian medical devices industry is close to 11 billion actually. And in fact, this is growing at CAGR of 37% from 2020 to 2025. It is going to increase to reach at least 50 billion US dollar. The medical devices market is going to achieve 50 billion US dollar in 2025. That is a phase in which that medical devices market is growing. If you take the foreign investment year on year in 2020, it has happened 98% increases there. Means like almost close to US dollar 301 million US dollar is the investment which has happened in this year 2020. Year on year, if you calculate, it was only 151 million despite the pandemic actually. So that is the promising opportunity in which the sector is growing. In fact, India is one of the very major destinations for global devices market, as I have mentioned earlier. Why? What is the reason and all? And in fact, in Asia, we are the fourth, biggest leader actually. And if you take up the EXIM scenario, is like, our import is close to $1.77 billion against export of 0.99 billion. It means still, as I mentioned earlier, sector is, highly import, dependent. So if you take a medical devices industry as a segment, if you see, like, say, for example, as I mentioned earlier, very simple technological items like ice bags towards, very sophisticated, like proton therapy items, everything is, under the medical devices industry. If as industry, if I segmentize, if I segregate, you can see, like, this can be classified as a preventive, care device, assistive care device, or diagnostic or therapeutic gas device. In terms of a consumables and disposable, diagnostic imaging, for example, if you take MRI or X-ray or ultrasound, patients aid hearing, like hearing aids, pacemakers, dental, products like, dentures and braces, orthopedics and prosthetics, all, together is, giving a very promising, opportunity for the medical devices industry as far as India is concerned. And in tune with the, in fact, if we can see, the Medical Devices Rule 2017, which is, published by, Health and Family Welfare Department of Government of India, is one of the major, milestone for India is concerned. Because in tune with the global practice, the Medical Devices Rule 2017 introduced a risk-based classification system for medical devices, including the low risk, low moderate, moderate high, and high risk, related items. Based on that only is like, all the manufacturing devices, especially if you take, Indian entrepreneurship is concerned, our MSME close to 450 to 500 million US dollar opportunity is there for class A and class B, like low risk and, low moderate risk type of industry. If you take, a C and D category, that is a high risk industries, 90% of our high risk, value chain of the manufacturing, it depends on the, import only. And a lot of multinational companies have extensive networks in India. But the only problem, if you take, like, most of the things are, they manufacture outside India and then they assemble or they do the service kind of thing. That is where, like, we need to concentrate more on this industry. See, as mentioned here, there are a number of key growth drivers for the medical devices industry. As I mentioned earlier, in 2025 itself, it has a potential of 35% CAGR actually. It means, like, it reaches up to 50 billion US dollar. That is a, strength of this industry actually. As I mentioned earlier, it is a number of medical facility has been increased, tremendously actually. Especially if you take the post, pandemic, COVID pandemic, the healthcare technology has, rapid expansion. It, in fact, witness a dramatic shift in the dynamics of medical equipment sector because there is a rising gap between the supply and demand actually. So that leads to a, kind of, if you take it, that COVID time, we can see a lot of miniature diagnostic market, which has been improved, which has been, what is that? Miniature diagnostic market is one of the very important, like RT-PCR itself is a kind of medical device, miniature diagnostic market. Nowadays, people earlier, it's a clinical, need was there. Now the patient, care is the, key of the hour actually. So the booming miniaturized diagnostic market is one of the another reason. And in fact, if you take it up with digital health ecosystem, the technological disruption actually in terms of technology, if you say artificial intelligence, like previous speaker also mentioned about it, augmented reality, VR, or IoT, any kind of a technological disruption. Because today healthcare sector or medical devices sector, it is not only a kind of a devices assets, because most of the modern medical devices industry is nothing but the technological driven medical care industries actually. So as far as the technological driven is concerned, the technological disruption, especially the startup level and the higher technological level, is tremendous as far as India is concerned. So that is another one of the important key growth drivers for the medical devices industry. And post it is like a COVID becomes an eye opener rather kind of a benchmark for the diagnostic or the medical devices we can say, or healthcare itself we can say, because COVID has brought a lot of attention towards the healthcare to common public actually. So what are all the impact and assessment, if you can see, the COVID, post-COVID and pre-COVID era would be the kind of a benchmark for the any healthcare sector industry, especially for the medical devices. And that rising demand for wearable devices Today, if you take it like, say, for example, a diabetic, India can be named as a diabetic capital of the world. So if you take it any kind of a glucometer which is attached to the body, which is a kind of a wearable device. So tremendous opportunity for such kind of devices have been increased, especially in India. So that is another one of the important factor where that, this medical devices industry can, place in. If you see that according to the World Health Organization, WHO, the chronic disease prevalence was estimated to be, by 57% by, year 2021. So emerging market like India will be the hardest hit, leading to the rising prevalence of chronic diseases and related increase in disability adjusted life years. So we are all going to have, some kind of lifestyle diseases or disability, but nothing but that we have to adjust with that. That kind of adjusted life year is going to be increased. So that increased, disability adjusted life year is going to create an increased demand for the healthcare system, especially medical, device industries. See, as far as government of India is concerned, as I mentioned earlier, that post 91 economic reforms policy, a lot of things have happened. But 2017 medical devices rule is a benchmark. This one, and 2019 actually is another benchmark event has happened, which I explained. So as far as this government of initiative is concerned, the government of India has taken up several proactive steps over, to ensure the growth of this vibrant sector actually. So Government of India published the medical devices rule in 2017, and the Ministry of Health and Family Welfare, in line with the global best practice, adopted a risk-based classification system, which I mentioned earlier. So we also followed that global harmonization task force, though is no longer is valid, this GSDF, which has been rechristened as the International Medical Devices Regulatory Forum. So in line with that GSDF, that Government of India in the medical devices rule given the classification of the risk-based system. So in order to strengthen the R&D and other things, 100% foreign direct investment in medical devices sector has been allowed. And FDA inflow, if you take between 2000 and 2021, $2.23 billion FDA is inflowed into the system actually, and which is ably supported by the recent Atmanirbhan packages. One thing, as far as this Atmanirbhan package is concerned, it is not antithetical to the any kind of foreign investment, though it, given the boost for the Indian industries to develop their system, it also equally welcomes the foreign investments, so as to Indian industry will grow. So as far as Atmanirbhar package is concerned, it is mostly several short-term and long-term measures for the healthcare system, including the production-linked incentive schemes for boosting domestic manufacturing of pharmaceuticals and medical devices, and the scheme for development of medtech zones with the common scientific and manufacturing facilities, which is also part of this Atmanirbhar package. And as I mentioned, earlier, the National Essential Diagnostic List has been finalized to improve the availability of the quality diagnostic across the health facilities in India, and which is notified in the clinical. That's what I said. 2019, another landmark thing happened as a clinical establishment rules 2019, which gives a standardization and better quality for any kind of devices, which has been notified. The salient feature of this MDR, 2070, if you, say, it is a risk-based classification system. A provision of notified bodies is mentioned. A quality management system in line with ISO 13485 is being mentioned here. Provisions related to the essential principle of safety and performance for manufacturer have been especially specified in these rules, and separate provision for clinical investigation of investigational medical devices, that is, the new devices, have been made at par with international practice. And provision is also made to designate or establish central government medical devices testing laboratories to verify conformance with the quality standards. So these are all the parts of the salient features of this MDR 2017, which is placed India at par with any of the global practices as far as the medical devices industry is concerned, in terms of quality, in terms of stringent norms, so that we can also compete equally with the global medical devices industry. So at this point of time, I would like to share the importance of Kerala for the medical devices. If you take Kerala as a state, like everyone knows, like Kerala model of development. In terms of healthcare, Kerala is one of the leading state in India. And if you can see medical devices industry, that I mean, I'll come to later regarding the medical devices industry. If you take, because in terms of a sustainable development goal, Kerala is one of the leading state, I mean, as far as India is concerned. Availability of the skilled, I mean, workforce, strong research base in the healthcare is one of the key aspects and competitive advantage of the Kerala. We have a very vibrant startup ecosystem. I mean, one of the world leaders, we can, I mean, very proudly say. Such kind of startup ecosystem is already, especially medtech, I mean, area has been established here. And in also the government of Kerala has established Bio 360 Life Sciences Park at Trivandrum, where we have a number of, I mean, industries are getting operated. And we have rich natural, I mean, biodiversity hotspots are there as far as Kerala is concerned. And we have a legislative and digital reforms which has been taken, I mean, so as to ease of doing the business, the state is established. So when I mean, mention about Kerala's Life Science Park, as I mentioned earlier, as far as the medtech industry or medical devices industry is concerned, it is the holistic and comprehensive industry. It is not about only the industrial aspect of putting up some kind of industry. The joint, I mean, collaborator of this, I mean, August forum, Sree Tirunal Institute of Medical Sciences and Technology. They are one of the leading researchers as far as, I mean, medical devices are concerned, and one of the leading, I mean, R&D. There is one important aspect as far as this medical devices industry we have to think. As I said earlier, 75% we are I mean, import dependent. If you take, I mean, SCTIMST is concerned, it's R&D institutions. So there is, I mean, no sense in making, we have a strong R&D at the same time, very less manufacturing base. It is our duty, especially Government of India and Government of, I mean, any government's duty to collaborate or to bring, I mean, together R&D as well as a manufacturing base. That is where we are going to get the benefit out of it actually. So that is how Government of Kerala decided to seek the help of Government of India through SCTIMST, so that this R&D institutions, I mean, research and cutting edge technology can be utilized for the development of the medical devices industry. So in Kerala's Life Science Park, wherein that we have a very holistic, I mean, medical devices technology is a part of it. Biotechnology and biopharmaceuticals is a part of it. I mean, the first speaker of this session mentioned about the, I mean, nutraceuticals. He's one of the very strong, I mean, academician. So nutraceuticals is a part of our Life Sciences Park. So it is like through Life Science Park, I mean, Kerala is, I mean, thinking of bringing industry, academia collaborations, which is where the future, I mean, R&D and cutting edge technology can be exhibited through the manufacturing. So what is happening presently is that industry is working, I mean, parallelly, academia is working parallelly. So it's a lot of, I mean, synergies required to bring industry, academia collaborations. That is where, I mean, we are trying to, I mean, establish that kind of a collaborations through Sree Tirunal. We have, I mean, Institute of Advanced Virology, which is placed in our park actually, and Kerala Veterinary, I mean, science universities are there. Number of healthcare industry and healthcare system is also established, or I mean, functioning around this place. That gives a kind of a collaborative system to technologically partnership with all those people to provide the ample opportunity for development of the life science as a sector in India, especially in Kerala. As I mentioned earlier, it is a very important thing as far as the government is concerned to bring industry academia collaborations. So as I said earlier also, Sree Tirunal is, I mean, kind of one of the leader in terms of their R&D and cutting edge technology. So government decided, okay, we can have a joint initiative and collaborative with the SCTIMST so that the separate medical devices park can be created in Kerala itself. In fact, as far as this med device park is concerned, we are going to create a, I mean, world-class ecosystem wherein that it will provide support system for a medical device manufacturing, as well as the research and, I mean, development would be there. Testing would be there, pilot manufacturing would be there, even testing of animal sciences has also been established, technological innovations and knowledge dissemination in the medical devices domain. Because it is parallelly R&D, manufacturing, as well as the testing. This three come together is going to be part of this medical devices park. In addition to that, for the industrial linkages, we are going to provide the startup ecosystem for all medtech industry can come and use this service at one place, so that, I mean, as a medical devices, as an industry can grow, as well as R&D also can go along with that. So that is the whole idea of this park actually. So as I mentioned earlier, the medical devices park, I mean, contains the key facilities like modular manufacturing units and to setting up of the large manufacturing, I mean, industries, medical devices testing center along with the international accreditation is going to be, set up, and effective R&D facility. And this service would be shared by the entities within, MedSpark. And we are going to create the knowledge center for skill upgradation in R&D manufacturing and clinical trials, etc. All is going to be placed under this. And technology business incubation center for promoting the startups and early stage companies, which we are going to seed funded or incubated, going to be coming under this medical devices park. So totally close to 200 acres of land has been allotted for exclusive medical devices park, jointly initiated by SCTIMST as well as, Government of Kerala. And if I briefly mentions about the opportunities for this sector, especially, medical devices sector in India is concerned. So as I mentioned earlier, number of speakers yesterday, today I'm going to speak about the R&D. So it is R&D is, as far as India is concerned now, very strong base of R&D and a lot of cutting edge technology has been developed by our own researchers. So how we are going to use this R&D development, which has been taken place in India, for the development of a manufacturing base, especially in the medical devices sector. That is where I mentioned about industry academia collaborative linkage. So India is going to be India is a strong, R&D base in which it provides a large, ample scope for any medical devices, foreign collaboratives or any industries to come take part and take the cutting edge technology to develop the medical devices, which is one of the key factors as far as India is concerned. And we have a diagnostic testing facilities. So Government of India itself has created a lot of medical devices park under, product linked, incentive schemes. Kerala is one of the early beneficiary through Department of Pharma. As early as in 2017 itself, that it has been allocated to us. Government of India has then granted 25 crore of rupees for developing this one. So this cutting edge diagnostic testing facilities, which is one of the another key factor as far as India is concerned. And a lot of technological innovations are happening. Like as I mentioned earlier, this artificial intelligence, machine learning. Not only this, IoTs are there, and a lot of other technological innovations are happening in India. As I mentioned earlier, medical devices industry is nothing but that. It is not only a devices as such. Technology disruption is also a very important part of it, which is also part of the key sunrise growth sector as far as this is concerned. And a number of miniaturized diagnostics is being developed. It is getting developed. India is going to be the very important base for this, and a lot of robotic surgeries and emerging technological driven assistance for healthcare provision. These are all the very important things as far as Government of India government is concerned when it considered to the medical device industry. So if you take industry as a whole, medical device industry as a whole, as I mentioned earlier, that currently, though it is $11 billion, US this industry is running around. In 2025 itself, we are expecting $50 billion shift would be there. So that opportunity is available for everyone, not only for manufacturing base alone, the R&D, testing, all the facilities. This is applicable. So this sector is definitely going to be one of the key sector. You can see that medical devices sector in India is poised to be the next sunrise sector with the support of proactive government policies and technically and technologically competent resources available in India. So Indian medical devices sector offers a plethora of investment avenues in healthcare medical devices sector for prospective investors. And if you say Kerala itself, Thermopenpal is a Japan company initially. Is placed in Kerala. Nitra Gelatin is a Japanese company, which is government of in Kerala and Japan collaborating company, which is again functioning in Kerala. So if you see all those things together, the medical device sector in India is going to be the sunrise sector, and it is a lot of opportunities are there. This is what I would like to make a presentation. Thank you.
Okay, thank you very much for your nice talk on this medical devices. Yeah, now we have time for questions, comment.
Thank you, sir, for your very informative talk. So I have just one or two questions. What is the approving agency or authority in India to approve this kind of medical devices?
Sorry, can I get the question once again?
Approving authority. Suppose I prepare this type of device. Who will approve me that yes, you can use it? You can go to the market with this device.
It depends. Like that's what I said. This sector now offers because it's all separate places. Medical devices park offered by the government of India is a collaborative place wherein that because what is required for the device is that testing facilities required. And then certifications are required. If you bring all together in one particular place, it will be expedited. So it is like say for example in government of Kerala, not in government initiative. Thermopenpal is a private, initiatives of government. So now they are the world leader in, making a blood, bags actually. So it is all depends on the how much, testing facility offers. That testing facility is very important part of this manufacturing base, which is available right now. So as early as possible. So it is not that, any delay as far as government is concerned actually. As early as possible, it can go to the market.
There are already existing boards that this can accept that medical device. Okay.
Yeah, of course.
Okay, thank you.
Any other questions, comments? So it seems there are no other comments or questions. So please join with me to thank all the speakers of this session for their nice illuminating talk. Now I hand over the.
I had a question for Rajamanikim sir.
Yeah,
Is he around?
please go ahead. Yeah, please go ahead.
Yeah, please.
Sir, you spoke about the Med Park at Kerala, and I was curious about two aspects. Your first or second slide, you said 1.77 billion of imports, US dollar, and 0.99 billion of exports. So just curious to know what are the exports which India, what are the devices that India is exporting to the extent of, it may be less compared to the imports, but still it's nearly half, 0.99 billion.
See, as far as India's export market is concerned, if you, as I mentioned, Thermopenpal is doing blood bags. They are the world leader actually. I think number 3 or something like that. And most of the exports happening in India through the diagnostic markets actually. We have very vibrative diagnostic markets are available in India. They are creating number of kits, easy diagnostic kits, miniature kits. So I would say, like, say for example, Agape is one such industry which is doing a lot of exports. So our export is mostly concentrated on not sophisticated high-end technological machineries, which is still we are import dependent. The export is mostly on diagnostic part, which is India is doing. So that is where our medical devices industry is a sunrise sector. I said whatever the import which is happening at 75% level, that can be developed in India itself, and we can concentrate. Not only export point of view, even for our own benefit. It is very important to have such a vibrant medical devices industry. Presently, our export is limited to diagnostics and mostly bags and less risk items actually.
Yeah, and the other thing is which I'm actually a scientist of Sree Chitra from the very early days, and we have contributed a lot in the device in the R&D, but I think India as a whole lacks a lot of manufacturing capability in every sector, not just healthcare. And when we consider the Med Park, we're still going into internet or artificial intelligence and all the high tech of the present situations. But should we not be focusing on developing our manufacturing capabilities of high risk also? Because what's your opinion on that?
Yes, doctor. Actually, as you mentioned, this, as I mentioned earlier, what is happening presently in India is the R&D, which is like Sree Chitra Tirunal. You are doing R&D. There is no manufacturing base, or else like the manufacturing base is very, very low as far as investments is concerned. So that is why, in fact, you must be remembering about we are collaborating. Sree Chitra Tirunal, Government of Kerala is collaborating with the Medical Devices Park, in which that your R&D, especially the extracorporeal devices, high-end devices on like stents kind of thing, we are going to manufacture it, not only for the use of India, anything extra, it can be exported also. So that is where we are going to concentrate. And in fact, by 2023, the MedSpark will be operational in Kerala. And your technology with the manufacturing based support of, Government of Kerala, definitely going to be one of the world leader in terms of export.
So this is Mohanan from Sree Chitra. How long will it take the MedPark to realize?
The construction part is going on. By 2022 mid that we would be able to, start the MEDS park. As far as the technological, partnership with, SCTIMST concerned, is like already that, we have, devised a DPR in which that what are all the devices, because our concentration is more going to be that affordable as well as the highly qualitative healthcare system to, people actually. So that your R&D with our manufacturing base is going to start at least by December 2023, it will be operational.
Thank you, sir.
Okay, so let us thank all the speakers in this session for their nice talks. Now I hand over this control to the organizers, please.
Thank you, Sri Rajamanikam, sir, for your very informative talk on promising prospects of medical devices sector in near future. We thank all the speakers for their valuable inputs. We thank Professor Babani Prasad Mandal and Dr. P.M. Guru Basavaraj for chairing this session. Let's wind up this session and reconvene after a short break. The next session will commence by 11:45 a.m. IST sharp. Thank you.
Welcome back to Nobel Laureate Science and Technology Seminar Series. Moving to our next session, we welcome Dr. N.R. Kamini, Senior Principal Scientist, Head of Biochemistry and Biotechnology, Associate Professor, CSIR, Central Leather Research Institute, RDR, Chennai, Chairman, IJAA South Chapter, and Dr. Utpal Bora, Associate Professor, Department of Chemical Sciences, Tezpur University, Tezpur, Assam, Joint Secretary IJAA to jointly chair this section. We welcome you both to the session. We'll introduce the speakers and the chair can coordinate the proceedings. Attendees, please note to quote your queries on the chat box during the course of lecture. The first speaker of this session is Professor Chandrabas Narayana, Director, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. He was the Dean, Research and Development at the Jawaharlal Nehru Center for Advanced Scientific Research, JNCSR, Bangalore. He is an optical spectroscopist, mainly Raman spectroscopy and Bryan spectroscopy. He works in many interdisciplinary areas using Raman spectroscopy in biology for drug discovery and diagnostic applications. We request the chairs to invite the speaker. Madam, please unmute.
Good afternoon all. I welcome Dr. Chandrabas Narayana, Director, DBT Rajiv Gandhi Center for Biotechnology, Trivandrum, to deliver his lecture on applications of Raman spectroscopy in life sciences and medicine. Sir, you can make your presentation. Hello.
Sir, please unmute.
There is no sound, sir. Some problem. No, sir, some mic problem.
Is it possible? Unmute, yeah. You can unmute and you can share your PowerPoint presentations.
Sound.
Now, can you hear me?
Yes, sir.
Yes,
yeah,
sir.
Yes, sir. Yes, sir.
Okay, good. Are you able to see my screen?
Yes, sir. Yes, sir.
Yes, sir.
Great.
Slide.
Sorry, there was some issue with my mic. Okay, now it's working. So first of all, let me thank the chairperson, Dr. Kamini Ramadu, for inviting me. I would also like to thank Sree Chitra Institute of Medical Science and Technology, as well as very special thanks to Dr. P.V. Mohan, who has been coordinating this with me, and for thinking of inviting me and allowing me to come into this wonderful forum, Nobel Laureate Science and Technology Seminar Series, and the 11th Indian-Japan Science and Technology Seminar. And good afternoon to all my Japanese colleagues, and good morning to most of you in India. It has been wonderful because I have been associated with Japanese for a long time as part of the KEK Tsukuba, which I have been coordinating for more than 10 years. And now, after taking over as a director, I've actually just relinquished that job of coordinating the beamline at KEK Tsukuba. I am going to continue from where Professor Umapati had left. He was actually looking at diagnostics. And my interest is also in diagnostic, but I am not going to use this time in my lecture diagnostics, but I would actually try to induce many of the biologists to use it for answering very, very important structural, functional relationships of proteins, which is very important for therapeutic applications. My initial introduction will be just to keep people interested in how Raman is such a simple technique. Basically, you take any monochromatic light source and put it onto the sample and collect it and disperse it and take a photograph, and you will get this elastic scattering, Rayleigh scattering, and the shifted frequencies in the Stokes region, which is the red shifted light, and that can be digitized. And each of these peaks are related to the molecular vibrations, and these vibrations are very unique and tells you about the molecular signatures as well as information about their environment in which they are there. And it's very useful for many applications in material science as well as in biology. I have been building Raman spectrometers. This is one of the miniaturized Raman spectrometer, which I built it for IISER Trivandrum as part of the imprint project, where they were actually interested in pesticide detection in fruits and vegetables. And this was actually very well received by DST in the imprint project, and they got the top 4 imprint projects based on these innovations, which we have actually delivered. I also have modified this into a 96 well plate system where we can actually do diagnostic applications. So these are very interesting times where we can actually make Raman spectrometers very, very simple and useful. And you can see the signal is as good as any commercial system, which is the latest, highest high-level commercial system and our system. And you can see it is absolutely exactly the same. Now I come to my research talk, where I'm actually going to show you 3 aspects in my research. These are some of the things where we are answering specific questions which people have. I was actually interested in the question which Dr. Minumathan, who is actually a eye surgeon, a cataract eye surgeon in Chaitanya Eye Hospital in Trivandrum, came to me, was asking about the lens capsule structure. Can we actually find out what happens when this, epithelial staining dye, type-in blue, is put into it? Does it actually cause any change to the characteristic of the lens capsule? And there is no technique which can in situ tell what is happening into the system. So we were interested in it, and my student, who is currently a faculty at in IIT Delhi, Dr. Soumik Siddhanta, actually started working on this. So everybody knows that we go to the eye surgeon when your cataract is mature enough. So it's very difficult to see the lens capsule for capsulorhexis technique, which is used for removing the lens from the lens capsule. So when you are doing this, you actually stain the lens capsule with blue dye, which is the type-in blue. And people have actually seen that there is a possibility of some sort of a problem with the lens capsule when it is in a very matured state when you use these dyes. So the question was, can we find out what exactly is happening? So we started looking at it, and you know that eye capsule is made up of predominantly collagen type 4 superstructure, which is basically a triple helix, which has a node which is called the NC1 node, and the triple helical domain, which is glycine rich in this region. And these triple helices actually form an hexamer at NC1, and then this network actually gets linked up, cross-linked at the disulfide bond formations, which are called the 7s domain. And this is the network of the collagen, which is actually the major part of the eye capsule. And then you have a protein network at the bottom, which is very, very, very thin, which is called a laminin network. And this is basically what it is. So when you actually look at the collagen 4 and pure collagen 4, which you get in the market, and you can take the human lens capsule, you can see one-to-one correspondence between the Raman signature. So it again strengthens the concept that it is a very good fingerprint itself, because it gives you exactly this the constituents of the system. Now, when you look at this, I am actually showing that there is something called the amide 1, amide 3, and many aromatic, and also the 7s domain, which is the disulfide bond region, which are present in the Raman spectrum. Now, why is that amide 1 is very important? Because it is actually looking at the secondary structures. And you know that beta sheets, alpha helix, random coils are the major protein secondary structures which are present. And this is actually the amide 1 vibrations are related to the peptide bond, which is the C double bond O NH bond. And it is the vibration of the C double bond O and NH coupling together, giving you 3 different types of vibrations. And they are very much related to the kind of hydrogen bonding which is present, and it actually gives you an idea of it. And the amide region can be deconvoluted into multiple regions, and you can actually, with a lot of studies, we know the regions are actually dominated in these places. So we can actually find out this from our Raman signature. Now, when we look at the collagen 4 amide 1 region, you see there are 2 peaks, and most of it is coming from the alpha helix. So this is actually the lower peak is related to the polar region of the alpha helix, polar region of the this triple helix. And you also have the non-polar region, which gives you the higher frequency region. Now, if you look at the lens capsule, it also has 2 peaks, and these are actually correlatable directly to these regions. Now, what we have done is that we were actually looking at this secondary structure itself, and then you see that the usually when you have a dye, it gives you the maximum Raman signal. And the protein and the dye, if they are present, you would actually have the convolution of the dye and the protein peaks. So here you can see that it is dominated by this, but luckily for this, amide 1 region is not actually interfered by the type N blue stain. And you also see that 7s domain is not interrupted by the type N blue, so we can actually analyze it very easily. Now, when we look at the lens capsule without the dye and with the dye, you can immediately see that there is a new peak which is appearing at higher frequencies. And this is because there is this is random coil formation, and the intensity of the peak of the alpha helix is going down, which suggests that partially some of these alpha helices are getting unwound. So basically, that is the reason for type N blue actually causing the structural dysfunction of the protein itself. And that means your network is getting loose. And that is the first signature to tell you that if the type N blue is present, in presence of the light, actually it happens. Now, to prove that it is light, which is actually very important to be controlled, we did the experiment with trying to find out the ordered structures, the fragment. That means the area under the curve, we can actually look at it, and the concentration of the type N blue as it reduces. So we are diluting it. So this is as is type N blue, and then this is dilution at higher dilutions. You can see that the order structure remains as is when you are highly diluted type N blue. But when the type N blue is actually in the region in which people use it, 5-time dilution is what most of the times you use it, that you find that the order structures get disrupted. Now, what we did is we also changed the light exposure. When there is no light, you can see that there is no change in the order structure. That means the type N blue doesn't do anything by itself. But the moment you put in light, you can see that the light actually causes order structure. But it also depends on the concentration of the type N blue itself. So if it is 5-time diluted, then you see that even up to 10 seconds, there is no change in the order structure. But as you increase the time, it starts actually getting affected. So that was very important knowledge, because in general, the surgeon needs only a 10-second to create that lens capsular rhexis. And that means you can, in fact, not destroy anything, because the light, you can wash off all the type N blue after this process, and the type N blue actually is not present, and the light is not going to affect the lens capsule. So the success will be higher. That is one thing. Now, another thing we wanted to know is whether the elasticity of the lens capsule gets affected. So people have done these experiments. So they have seen that when you actually look at it with the type N blue, with the light exposure and all those things, you are seeing that with the type N blue and the light exposure, it becomes stiffer and stiffer. And you can correlate the stiffness by also putting glutaraldehyde. Glutaraldehyde is a cross-linking agent, so it actually causes random cross-linking, and you can see the glutaraldehyde gives you highly stiff lens capsule material. So what we did is we added the glutaraldehyde to study whether it is actually causing a strain, cross-linking. And as soon as you put the glutaraldehyde, you can see emergence of two peaks, 708 and the 801, which is related to the cross-linking of this lens, collagen. Now, if you look at the stained type N blue capsule, and for large light exposure, you do see the similar bond frequencies present, and that shows that cross linkages are happening in the system. So type N blue indeed causes elasticity change in the lens capsule, as well as it also causes the opening up of the triple helices and with light presence. So we actually came up with the solution that you can actually use a 10-second exposure time in the type N blue of 5% type N blue, and it still can remain structurally intact. And this was correlated with the experiments which they were doing, and they found that indeed within 10 seconds you can finish this capsular excess, and that's all is necessary for the staining. And so easily you can remove off the type N blue, and this became a very important thing for the cataract surgeons based on this, and it was appreciated both in American cataracts annual meeting, as well as in the European cataract meeting and Indian cataract meeting, and this was recognized. Now, another aspect is that when we are working with the proteins, when the proteins are in very low concentration, you need some sort of an enhancement, because Raman is a weak phenomena. And there was another technique which was discovered in late 80s when people found out that nanoparticles of gold or silver, when molecule is put there, it actually enhances the Raman signal. And this is what is called surface enhanced Raman signal. And this is, though the phenomena is not well understood even today, but it is a very useful technique for studying proteins and many other biological systems, and people have been using this extensively. So one of the examples for this in our study is recent study is we were looking at the restriction enzyme KPN1, and we were interested in understanding the allosteric transition happening due to the divalent metal ion. This was in collaboration with Professor V. Nagaraja's group and at MCBL in IISc, and my student Shantanu, who finished his PhD, and he's working for Renisha company. Now, what we all know that restriction enzymes cut DNA at specific site, and this KPN1 is the coming from the bacteria Calibacilla pneumoniae, and it has a defense mechanism to cut the viral DNA at GGTAC region, and it is highly dependent on the metal ion which is present, the divalent metal ion. So Nagaraja's group had found that calcium, when it is present, it actually gives you high fidelity. That means DNA cleavage is very, very accurate. And it actually binds to two regions, HNH region and EDD region. While magnesium in low concentration gives you high fidelity, but when you increase the concentration of magnesium, you actually find promiscuous behavior. That means there is a problem with the structure of the protein itself. So these are HNH binding, cation binds to the HNH domain. And our question was, because there is no crystal structure present, this was a prediction they did. And we wanted to verify whether Raman could actually give you an insight into the structure of this protein itself. So we were looking at it because if you understand that, we can exploit the similar defense mechanism to do studies. So we were looking at SIRS as in place of crystal structure, can we actually use it extensively to understand what is happening? So we did the homology modeling of the KPN1 MD simulations, and we find that in the wild type, when there is no cation present, the EDD region is a beta sheet domain here. You can see the zinc is present. It's a dimer. So there is a counter zinc present here, and the beta sheet EDD region is here, and HNH region is in this region. Okay. So HNH region is present here. So you can see the calcium indeed binds to both the EDD and HNH region, while magnesium is more present in the HNH region. Now, we also did an electrostatic potential of the protein dimer, as well as looked at what is the hydrophobic and hydrophilic interactions. Because if you know the charges, you can know how it will be oriented on top of the physisorbed on top of the silver nanoparticles which we use. And you can see that these are the positive regions because this is negatively charged. So lysine 105 present in this is the anchoring point for it. And if it is the point, then this is the total distance from the, so the alpha helices are 3 nanometers away, while beta sheet is about less than 1 nanometers away. And the SIRS phenomena dies down as it increases in the distance. So we would be dominated by the secondary structure region amide 1 by your beta sheets rather than the alpha helix themselves. Now, when we look at the Raman spectra, this is the typical Raman spectra of your KPN1. And when you look at the amide region, this is dominated by the aromatic region as well as the amide region. And amide region, when you expand it, you find that there are 2 regions specifically. One is for the beta sheets and another one is for the alpha helix. And you can see that this is far away from the nanoparticles, and that's why the intensity is lower in this case, whereas the beta sheets are much stronger. This is the EDD region, and this is where the HNH region is present. Now, what we did is that we took the wild type without the magnesium, and we took with the magnesium, and we find that the amide one region is exactly same. That means there is no change in frequency. So magnesium, when we do the simulation also, we find the EDD region is not affected. It remains exactly the same. And this is the reason why these two are extremely similar. Now, when we actually look at this spectrum, and then we find that this is so you can see that in calcium case, we find that the beta sheets and random coil, there is an increase in the random coil present in the system. And that is seen from the fact that MD simulation shows EDD is actually, EDD region is beta sheets are getting unwound and it's becoming random coils. And so this is what is the reason why calcium is actually working in the case of recognizing the sequence, and it is able to do that. So we could actually use SIRS to exactly say what is happening in the system, in this system. In the last part of it, so this is what we have actually suggested. So you can use SIRS as a tool for subtle changes in the subtle changes in the protein structure can be actually identified. And here we have shown it in this chain allosteric signatures in the system. In the signatures. Okay, so now the last part, I am actually going to show you something where we use resonant Raman effect to actually do another interesting effect. So we were interested in looking at the red fluorescence coming from a blue Sandersine, and this was in collaboration with S. Ramaswamy, who was actually the director of INSTEM and currently in Purdue. And Sayan, my postdoc, and Shantanu were working with this protein. So sandersinin is a blue-colored protein isolated from the skin mucus of Valley. Sander vitreus is a fish found in North America, and it peaks in the late summer. This coloration for the fish, and this is actually caused because of the UV radiation, which breaks down heme, which is shown here, into biliverdin. Biliverdin is actually breaking this bond and forming the double oxygenation here, and there is no iron trapped in this system, and that's what gives you the blue color of the system. And this is actually combining with the sandersinin protein, and it binds strongly with it, and it is actually linked with the UV radiations. So when the UV radiations increase, this sandersinin actually absorbs this, and it provides the sunscreen for the fish itself. So it is a protective agent. So we were interested in this. This is sandersinin, And this is biliverdin in free form. And this is the biliverdin bound to the androcynin. And you can see the absorption spectra. And you can see in absorption, when the binding happens, there is an additional absorption around 630. When you do the excitation spectra and the emission spectra, you can see a red emission coming around 675, and you can see the red emission seen in the confocal imaging here. So basically, we were interested in androcynin because if you can actually use this as a replacement for GFP as a red fluorescence dye, and since it can be made recombinantly expressed in E. coli, we can also study the photophysics of the bilirubin and the androcynin, and that in fact gives you the interaction, and we can actually use visible resonant Raman and quantum mechanics calculations to study the molecular origin of this fluorescence itself. So, so what is the problem we have is that it is a tetramer. So there are 4 protein monomers come together, and that is not a good idea. So we would like to have a monomer, and you could actually see, study how it binds and still retains it. So we were interested in the crystal structure, and we have actually done the crystal structure done by Rams. That is, Ramaswamy had done in this crystal structure, and we know how this this molecule is binding to the protein itself. It's very interesting to see that there is a D-ring flipping which was seen in this system, though not very clearly in the X-ray, but we could actually say that there is a flipping of the D-ring in this system. And what we found is that if you are looking at it, there are 4 amino acids which are linked with hydrogen pi-pi stacking, hydrogen bonding, and hydrophobic contacts, which is phenylalanine, tyrosine, and histidine. These are the ones which are actually linked up with this. Now, here is a molecular dynamic simulation of both the native and the recombinant androcynin from the structure of the proteins itself. And you can see more or less they are similar in binding. And we can confirm this from the Raman itself, because the Raman taken at 405 nanometers resonant Raman, we can see the Raman spectra is more or less exactly the same for the native and the recombinant. So that means we are successful in actually getting the protein done. Now, we also can do the simulation and find that more or less the molecule binds to the same site, and it is more or less in the same direction. So it is very similar in both. The biliverdin is bound in this in the same form in both native and recombinant androcynin. Now, what we wanted to understand is the photophysics of it. So we actually did a free biliverdin, and we did a computation, and we found out the Raman spectra of it. And we can see that Raman spectra is little broad in these systems. And this is what the biliverdin, which is bound in the androcynin, is supposed to be. So this is the D-ring flipping, which we are talking about. The moment you have free biliverdin, it is broad peaks. And you know that when you have protein and the biliverdin, it will be a combination of both of the spectra. So the protein spectra is there. You also have the biliverdin spectra. But the interesting aspect is that all the biliverdin peaks have become very, very sharp. And this is a typical spectra. So we have done it with both 532 and 405. Both are actually in the different regime of the resonance. And we can find that the sharpness shows that indeed there is a strong correlation between the pi conjugation, which is actually extended when it is flipped, compared to when the conjugation is broken by this flipping. And that's the reason for the broadness of the peak, and that is what you see. So this is what we have actually observed. Now, what we did, mutagenesis, and when we do the mutagenesis, you can actually see that when you actually change the tyrosine 142 with alanine, and then you actually have a red shift in the system. So that means if I was in the resonance here, the resonance should be broken. And so that can be seen from our Raman. you can immediately see that the resonance is broken because the intensity drops down and it becomes broader. And that is purely showing that there is a redshift happening. And this can be verified with quantum mechanical calculations also, that there is a redshift expected when you have a mutagenesis done. So the binding region is very clearly understood. The way the binding is happening is understood. And we can actually use both resonant Raman and your molecular dynamic simulations to actually understand how these small molecules are binding to the protein itself. So this is what we have actually come out with in this one. And I would like to end this talk by thanking all my students who have been responsible for this kind of interesting work. And they are actually very, very important to answer many interesting problems in life sciences. And you can actually use this as a very, very powerful tool in absence of structure of proteins and various other aspects of it. Thank you. Thank you for your attention. Thank you very much.
Excellent presentation, Doctor.
Thank you. Thank you, sir. Thank you, ma'am.
Described about the importance of Raman spectroscopy in medicine, SIRS in protein analysis, and then Sanderson in protein for fluorescence imaging. It was very excellent, and we have got so many ideas from your presentation.
Thank you, ma'am. Thank you so much.
Thank you. Thank you, sir. Now you can ask few questions from the attendees. Attendees can ask few questions. Or you can put your questions in the chat box.
Attendees may please quote your queries in the chat box.
Sir, excellent presentation. Sir, I have a doubt. Can we use this Raman spectroscopy for detection of the different variants of corona, or is there any studies going on in these aspects? Because the protein is changing each time. Nowadays, the sequencing is the only method for detecting different variants. So can we use this as a simple technique?
Actually, there are two aspects to this question. Very excellent question. One is that we have already developed for HIV. We had developed non-PCR based diagnostic for HIV testing, and that is the reason why I actually built that particular Raman spectrometer. We were able to actually see this change in the without PCR. We were able to actually detect the changes, that is even single nucleotide polymorphism we were able to detect. So that is one way to look at it. So you don't need to actually do a RT-PCR. You can use Raman to do this. And this is right now not yet commercialized. We are in the process of actually coming up with the full protocols for doing it. That is one method of doing it. Second, what we are actually, you are asking is the protein signatures on the receptor binding region. So yes, we need to do this, but unfortunately the viruses needs a BSL-3 facility, and for which you need to take a Raman spectrometer into the BSL-3 facilities and try out this whole thing. So for which a trained person in Raman as well as BSL-3 is needed. Currently, we do not have such expertise available, but it is possible because the signatures of looking at viruses is there. But now another issue is that there will be a little bit of complications which will be there, which needs to be ironed out with a collaboration with a virologist, and we need to work on it. So there is a possibility, but it is right now not yet attempted. So it is a good question. It is a way to look forward to.
Thank you, sir, for your informative. Thank you, sir, for your informative and illuminating lecture on applications of Raman spectroscopy in life sciences and medicine. The next speaker of this session is Dr. Ayako Oyane, Group Leader at National Institute of Advanced Industrial Science and Technology, AIST, Tsukuba, Japan. Dr. Oyane received her doctoral degree in engineering from Kyoto University, Japan. She was honored with the Ceramic Society of Japan Award for Advancements in Ceramic Science and Technology, the Award for Young Investigator of Japanese Society for Biomaterials. She has several publications in national and international peer-reviewed journals. We request the chairs to invite the speaker.
I welcome Dr. Oyane, Group Leader from National Institute of Advanced Industrial Science and Technology, Tsukuba, to deliver her lecture on laser-assisted biomineralization for biofunctional surface engineering. Over to Dr. Oyane.
Thank you very much. Can you hear me?
Yes.
Okay.
Yes, ma'am.
I will share my slide. Okay. Can you see the slide?
Sorry, no, ma'am.
No? Okay.
But the desktop window is visible. Ma'am, kindly open the PPT.
PPT, I will try again now. Sorry.
I will stop and you can start again.
Ma'am, can you please do it once again?
I will try once again. Sorry. About? No? Okay, maybe I will share PDF file. Maybe the PowerPoint is too heavy to share. Can you see my, oh. Okay. Maybe I will share my desktop.
You can press Control Shift plus symbol. It will rotate.
Yeah.
Yes, ma'am. Now it's visible.
It's visible? So,
Yes, ma'am.
okay, good. I'm sorry.
Ma'am, can you please make it full screen?
Okay, I will.
Please close that window, madam. And bottom you can see one button, slideshow.
How about?
Yes, ma'am.
Oh, sorry. I'm sorry for the trouble. So, let me start my presentation. First of all, I'd like to express my appreciation to Professor Mohanan and all the organizing committee members of this special seminar for giving me the opportunity to present our study. Before the lecture, let me briefly introduce myself and my institute, National Institute of Advanced Industrial Science and Technology, AIST. AIST is a leading public research institute in Japan. There are more than 2,000 researchers working at AIST. And AIST has 8 regional research bases throughout Japan, and its main base is located in Tsukuba. My laboratory is also located in AIST Tsukuba Center in Ibaraki Prefecture, Japan. And as you may know, Tsukuba is known as a science city because there are many research institutes and 16,000 researchers are working in Tsukuba. In Tsukuba, there are many research institutes, including AIST and JAXA, GSI, and KEK, and so on. And this is Mount Tsukuba. I hope all of you will have a chance to visit Tsukuba in the future. My research career as an AIST researcher started in 2002 after receiving PhD degree from Kyoto University. During the last 2 decades, I have worked with excellent 6 JSPS fellows. 3 of them are from India. As you know, JSPS fellowship term is only 2 years at the longest. However, our friendship and academic network continue even after the fellowship term. For example, Dr. Bodak, he's now working as a principal investigator in CSIR. He introduced me excellent postdoctoral researcher, Dr. Pal, recently, and he has been awarded JSPS fellowship, and he will join our laboratory from March next year. Dr. Shyama Santkumar was also introduced kindly by Professor Mohanan, and she worked with me for almost 2 years. In the last part of my presentation today, I will describe the research work done by the previous JSPS fellow, Dr. Nathaniel. He is now working in VIT India. So this is the outline of my presentation. I've divided my talk into 3 parts. First, I would like to introduce calcium phosphate, CAP, and CAP coating techniques. Second, I will describe our laser-assisted biomimetic process for calcium phosphate coating. Lastly, our recent application studies toward tooth surface modification will be described. Now, let me start the introduction part. First, I will describe briefly about calcium phosphate. Calcium phosphate is a major mineral component found in human bones and teeth. For instance, human cortical bone is composed of organic collagen fibers and apatite nanocrystals deposited on their surfaces. Apatite is a kind of calcium phosphate crystalline phases and constitute 97% of enamel, 70% of dentin, and 60% of cortical bone. Some calcium phosphate compounds, including apatite and beta-calcium phosphate and octocalcium phosphate, OCP, show good biocompatibility and osteoconnect bone bonding ability, different from other artificial materials. Generally, artificial materials are recognized as foreign substances from the living body and encapsulated by the fibrous tissue like as shown in this image. That is, general artificial materials cannot bond to the living bone tissue. In contrast, a hydroxyapatite ceramic implanted in a bone defect bonds directly to the living bone tissue without inducing severe foreign body reactions. Therefore, apatite and some other calcium phosphate ceramics with good biocompatibility and bone bonding ability have long been used as orthopedic and dental implants. However, these calcium phosphate ceramics are extremely hard and brittle, and hence they can be used only under limited conditions. Calcium phosphate coating is a useful tool for biofunctional surface engineering because it can provide biocompatible and osteoconductive surfaces on other materials, including tough and strong metals and soft and flexible polymers. It has been proven that calcium phosphate-coated materials show better integration with soft and hard tissues than uncoated materials, and this leads to the accelerated healing and improved clinical outcomes. As a calcium phosphate coating technique, there are many processes developed hitherto. Among these, a biomimetic process that is based on pseudo-biomineralization in a supersaturated calcium phosphate solution has recently attracted increased attention. In this process, a supersaturated calcium phosphate solution, similar to the body fluid, is used as both the calcium phosphate source and coating medium. Owing to its mild coating conditions mimicking the body environment, the biomimetic process is applicable to low melting point substrate, including polymers. In addition, it is possible to immobilize some cell-stimulating substances, such as biologically active proteins, DNA, and trace elements within a calcium phosphate coating through co-precipitation. A conventional biomimetic process is generally carried out in two steps. In the first step, a substrate is surface modified with functional groups or calcium phosphate pre-coating layer. In the second step, the surface modified substrate is immersed in a supersaturated calcium phosphate solution for 1 day or longer to grow a calcium phosphate layer on the surface. Such a multi-step and time-consuming process is not so practical. Recently, we developed a laser-assisted biomimetic process as a faster one-step calcium phosphate coating technique. In this process, a weak pulsed laser is used without focusing to irradiate a substrate that is immersed in a supersaturated calcium phosphate solution. In the case of a substrate of ethylene vinyl alcohol copolymer, EVOH, calcium phosphate precipitated on the laser irradiated surface within only 30 minutes. This coating process is carried out rapidly in a single step without prior surface modification step or post-immersion step in the supersaturated solution. Furthermore, by adjusting laser irradiation area and position, area-specific calcium phosphate coating on a target region is possible. These are advantageous features of the laser-assisted process over the conventional biomimetic process. This slide shows lower and higher magnification images of scanning electron microscopy, SEM, on the EVOH surfaces after irradiation for 5 minutes, 10 minutes, 20 minutes, and 30 minutes. As shown in these images, calcium phosphate precipitated on the laser irradiated surface within only 10 minutes and covered the entire surface after irradiation for 30 minutes. The next question is the mechanism behind calcium phosphate precipitation in this process. We consider that calcium phosphate precipitates on the EVOH surface as a result of laser absorption and the subsequent surface heating and surface modification. To verify this hypothesis, we carried out two control experiments. First, we performed the irradiation process without a temperature-controlled water bath to amplify the laser heating effect. Second, we performed laser irradiation not in the calcium phosphate solution, but in ultra-pure water to evaluate the base EVOH surface. In the first control experiment, to amplify the laser heating effect, laser irradiation was performed without a temperature-controlled water bath. As shown in this profile, the calcium phosphate solution temperature increased with increasing irradiation time. In the absence of the substrate, the temperature rise of the same solution was negligibly small. Therefore, the increase in the solution temperature should be due to laser absorption by the substrate and the light-to-heat energy conversion at the surface. Even under the temperature control with a water bath, the calcium phosphate solution should be heated locally near the surface during irradiation. The temperature increase in the calcium phosphate solution should have an accelerating effect on calcium phosphate nucleation and growth by increasing the mass transfer rate, as well as degree of supersaturation of the solution with respect to calcium phosphate. In the second control experiment, laser irradiation was performed in water to examine the surface structural change of the base EVOH substrate. As shown in these SEM images, submicron-sized pores formed on the EVOH surface within 10 minutes and increased in number density under laser irradiation in water. With increasing irradiation period in water, the water contact angle of the EVOH surface decreased. That is, laser irradiation increased water wettability of the EVOH surface. FTIR analysis suggested that the EVOH surface was modified with polar functional groups by laser irradiation. A similar surface modification should occur in the calcium phosphate solution as well. The resulting submicroporous structure with a hydrophilic surface provides a favorable environment for calcium phosphate precipitation. In the meantime, as verified from the first control experiment, the surrounding solution should be heated locally near the surface, and this would have an accelerating effect on calcium phosphate precipitation. As a result of these two effects of the laser irradiation on EVOH surface and the surrounding solution, calcium phosphate precipitate at the laser irradiated region only. Our laser-assisted biomimetic process is applicable to various materials. Actually, this process was applied to titanium metal, some polymers, and ceramic, which absorb laser light energy. However, this process was not applicable to optically transparent material due to insufficient laser absorption. Here, I would like to emphasize that titanium metal, an important biomaterial used as a dental implant and orthopedic implant, was also coated with calcium phosphate. For the titanium substrate, UV, visible, and near IR lasers are effective in calcium phosphate coating because titanium has relatively high absorption at wide wavelengths, light wavelengths from UV to IR. However, the optimum fluence depends differs depending on the light wavelength. Therefore, it is important to tune the optimum fluence at each wavelength. The calcium phosphate coating prepared by our laser-assisted biomimetic process has good cell biocompatibility. We prepared apatite coating on a substrate of polyethyl ethyl ketone, PEEK, a super engineering plastic used in orthopedic implants. As revealed by this cell growth curve, pre-osteoblastic MC3T3E1 grew faster on the apatite-coated surface than on the uncoated surface. The enhanced cell proliferation was reconfirmed by these stained cell images on here. To improve further the coating effect, we attempted to immobilize cell-stimulating substances, zinc and fibronectin. Fibronectin is a kind of cell adhesion proteins. Both zinc and fibronectin, FN, have an accelerating effect on bone formation. To immobilize these cell-stimulating substances, we added zinc chloride and fibronectin to the supersaturated calcium phosphate solution. After laser irradiation for 30 minutes, various contents of zinc were successfully immobilized on the titanium surface. By tuning zinc concentration in the solution, the zinc content on the surface was controllable to the level comparable to the reported optimum content for bone cell proliferation and osteogenic differentiation. Similar trend was also found for fibronectin. With increasing fibronectin concentration in the solution, the nitrogen peak that is derived from fibronectin increased. Using this fibronectin containing substrate and fibronectin-free substrate, we performed cell adhesion assay. As shown in these images, cell adhesion was enhanced on the fibronectin mobilized substrate than fibronectin-free substrate. These results indicate that cell-stimulating substances can be immobilized on the substrate by the present laser-assisted process without with retaining the biological activity. This is a summary table of the laser-assisted biomimetic process in comparison with the conventional process. Our proposed new process is a rapid single-step process within a few tens of minutes and effective in area-specific calcium phosphate coating on various laser-absorbing materials. Considering these advantages, this process has potential in dental applications in which a target region is confined to a millimeter scale. In the last part of my presentation, I would like to describe our recent application studies for tooth surface functionalization. We applied our coating process to a model material of teeth and then to a human dentin. The preliminary study using a model material was done by previous JSPS fellow Dr. Nazanier. We aimed to functionalize a tooth surface by fluoride incorporated apatite coating using the laser-assisted biomimetic process. Such a technique has great potential in treatment and prevention of dental disorders because apatite shows good biocompatibility with soft and hard tissues, and fluorine, that is a trace element in the human body, has antibacterial property and remineralization effect and tooth strengthening effect. As a substrate, first we used a model material, sintered hydroxyapatite, SHA, and second we used human dentin. The model material, SHA substrate, was immersed in a supersaturated calcium phosphate solution supplemented with various concentrations of fluoride. The laser irradiation was performed for 30 minutes, and the resulting sample was denoted as follows, depending on the fluoride concentration in the solution. The addition of fluoride to the supersaturated solution changed the coating morphology from the plate-like structure into densely packed pillar-like structure. On all the sample surfaces, calcium and phosphorus were detected by EDX. In addition to calcium and phosphorus, fluoride was clearly detected on the surface of F1 and F3. These two samples and fluoride-free F0 sample were analyzed by transmission electron microscopy TEM. According to the selected area diffraction analysis, SAED, the flake-like precipitate formed on F0 was composed of octacalcium phosphate, OCP. On the other hand, the pillar-like precipitates formed on F1 and F3 were composed of apatite. In the wide-range XRD analysis, it revealed that F1 and F3 gave higher peak intensities than the untreated substrate for 002 and 004 apatite planes. This result suggests C-axis orientation of hydroxyapatite along the surface normal of F1 and F3. The fluoride phosphorus ratio, F/P ratio, was calculated from the EDX peak analysis. And F/P ratio was higher for F3 than for F1. And this is because of the higher fluoride substitution ratio for F3. It is considered that fluoride substituted a part of hydroxide ion side of hydroxyapatite. As shown in these fluoride mapping images, fluoride distributed homogeneously over the apatite matrix on F1 and F3. From these results, it was found that after 30 minutes of irradiation, F1 and F3 formed fluoride-incorporated apatite layers, whereas F0 formed a fluoride-free plate-like OCP layer on their surfaces. That is, we achieved facile fluoride-incorporated apatite coating on the model substrate by the present process. Next, the coating's functions were evaluated by acid resistance, fluoride release, and antibacterial assays. In the acid resistance assay, the sample was immersed in a citric acid buffer for 3 days. After the assay, the plate-like OCP crystals disappeared from the surface, suggesting their complete dissolution. In contrast, pillar-like fluorine-incorporated apatite crystals on F1 and F3 resisted in the acid buffer. The amount of calcium and phosphorus dissolved from F1 and F3 was significantly lower than those dissolved from the untreated substrate. These results suggest that the fluoride-incorporated apatite layers suppressed acid dissolution of the sintered hydroxyapatite substrate by acting as a protective cover. In the fluoride release assay, the sample was immersed in a physiological salt solution for up to 78 hours. As shown in these release profiles, both F1 and F3 allowed the sustained release of fluoride ions over 24 hours. The fluoride ion concentration was higher for F3 than for F1. This might be owing to the higher fluoride content in the layer on F3. To demonstrate the antibacterial activity of the coating, a carrier-causing bacterium S. mutans was cultured in the presence of F0, F1, and F3. As revealed by the decreased turbidity of the bacterial suspension, bacterial proliferation was inhibited by F1 and F3. This result demonstrates the antibacterial activity of the fluoride-incorporated apparatus coatings on F1 and F3. Fluoride ions released from the coatings should be responsible for their antibacterial properties. Lastly, we applied this process to human dentin substrate. Dentin substrate were prepared from donated teeth after obtaining informed consent at Hokkaido University Hospital, according to the conditions approved by the ESCAR committee. Tabular dentin substrate were prepared from the donated teeth and immersed in a supersaturated calcium phosphate solution supplemented with fluoride. After irradiation for 30 minutes, the radiated region was successfully coated with fluoride-incorporated apatite layer. According to the TEM analysis, the fluoride-incorporated apatite layer was about 2 micrometers in thickness and integrated smoothly with the underlying dentin substrate without any apparent gaps. The fluoride distributed homogeneously over the apatite layer, as shown in this image. Antibacterial assay demonstrated that the fluoride-incorporated apatite coating has antibacterial activity against S. mutans. In summary, the laser-assisted biomimetic process is a potential new tool for surface functionalization of various substrates, including human tooth, because it is a rapid one-step coating process under normal pressure and temperature using physiological reaction medium without any toxic reagent. This coating process allows area-specific calcium phosphate coating on a target region. In addition, the coating's functionality is controllable via the immobilization of cell-stimulating substances, such as fluoride. Therefore, this coating process might be useful as a tooth surface functionalization. That's all. Thank you very much for your kind attention. Lastly, I'd like to thank Dr. Nathaniel, my previous JSPS fellow, for his hard work, and all other collaborators and postdocs, students, and also for JSPS Kakenshi Grant for supporting our research. And thank you very much. Hello.
Dr. Vayani?
Yes.
Excellent. It was systematic and you presented more data. It was very good. And you very well described about the biomineralization of calcium phosphate with different laser absorbent substances for dental application. Thank you very much. So at least you can ask few questions.
Attendees may please quote your queries in the chat box. Since there are no queries, we are moving on. First of all, thank you, ma'am, for your valuable lecture on laser-assisted biomineralization for biofunctional surface engineering. And we thank Dr. N.R. Kamini and Dr. Utpal Bora for chairing the session. We'll wind up the session for now and we'll convene at 1:20 p.m. IST sharp.
Yeah, thank you all. Thank you.
Thank you very much.
The lunch will be served in the specific hall
Hello. Can you hear me?
Hello, yes, we can hear you.
Oh, okay, great.
Yeah,
Great.
it is 5 more minutes.
Okay, sure. And can I share the my file? Can I make check my file? Is that okay?
Yeah, please do. Please do.
But yeah, share content doesn't work in my on my
Yeah,
computer.
just try it out. If you can share, just try it out.
Oh yeah.
How are you?
Yeah, very good. How are you doing, Bora?
Yes, yes, fine.
Oh, good, good. How about your family? Yeah, all good?
All good, yeah.
Oh, okay.
Okay, please go ahead.
Yes, yes. Thank you. And still, sorry, my share content, button is. Oh, okay, great, great. Thank you. I will try to share my slide.
Yeah, it's visible.
Yeah, it's now visible.
That's okay.
Okay.
And let's see. Is this full screen right now? It's no problems.
No problem.
Oh, okay.
Very good.
Very good.
Okay, good. And should I take off right now?
There is 5 more minutes because
Okay.
yeah, the time for reconvening has been shifted by 5 more minutes.
Okay, sure. Thank you. And I will release my yeah. Let's see. Oh, stop sharing. Now?
Yes.
Great.
Yes.
Yeah, actually, WebEx is not so familiar for us, especially in Japan. Sorry about that.
No, it's okay.
So how
So
are
nice
how's
to
everything in Japan?
Yeah. Do you know the Gifu?
Yeah.
Gifu prefecture. Oh, nearly center of Japan.
Tokyo. I was in Tokyo with Professor Okano.
Oh, I see. That's great. And used to be, Professor Bora, stay.
Madam, I was working with him only during the SPS.
Right.
So he's your co-scientist.
Yeah, he
Yes.
stays give for a very
Okay,
long time. Yeah,
Yeah, great.
that's a great. And yeah, and let's see. How about the Indian corona situation right now? Is that
We are okay.
okay? Oh, that's great.
We are okay.
I see.
Not much problem, although some 30 people have been identified with the Okinawa strain.
I see. Okay. Wow.
Sensei.
Hi.
Regarding Corona, one very interesting thing. So it starts with Madam's place, Kerala, Madam's place, and it ends in Assam, my place.
Yes, that's good.
If Kerala has some case, after 3 months, we will have that peak.
Okay, sure. And
yeah, that is really very true. And, because I think Malayalis go all over the world.
Yes.
That's interesting.
It was my Kerala students in Wuhan, China, who brought the first incidence of corona to India and Kerala.
Oh, I see. Okay. Yeah. Currently, my younger son is in Cincinnati, Ohio, in United States.
Yes.
Then, yeah, he said he doesn't wear mask in United States. But how about in India? People wear the mask?
Yeah, we are still supposed to wear mask.
Okay.
In
Yeah.
public spaces.
I see.
Yeah.
Yeah. In Japan also, same things.
Japanese people wore mask long back. It is nothing new.
Yeah, right.
Even when we were doing our postdoctoral fellowship, Japanese people used to wear masks.
Yes, yes.
I see. Okay, okay. And Bora, how many students right now in your laboratory?
7 students, including one postdoc. Yes.
Oh, great, great. Then everybody okay? No problems.
No problem. Okay.
I see. Okay, great, great. I see. And in our laboratory also, it's a great. That means total 26 members
okay.
in our laboratory. But recently, graduate students is gradually decreasing.
Oh.
Okay.
Yeah, it's a problem. Yeah, then
Is it because of the corona and people are not moving out or something like that?
Yeah, right, exactly. And
because even in India, educational institutions are not fully open.
Oh, I see. Okay, okay.
Yeah.
Yes.
Now we have another scare with this Omicron variant. What about Japan? Do you have any incidents of Omicron variant?
Oh, yeah, a little bit, especially in Tokyo and Osaka and some other big cities. Still, Gifu and Nagoya is okay.
Oh.
Yeah. Then my area is no problem. But
yeah.
Already Tokyo and Osaka already probably a little bit, but still it's start.
So nice to see that Nagara River in your first slide.
Oh yeah, right.
Nagara River is one of my favorites.
Yes. Exactly. It's a Nagara River, and do you know the Gifu Castle on the top of
yes.
Yeah. And this year is a 75th anniversary for Indian independent.
Yes.
I see. Wow, congratulations.
Thank you.
Thank you.
Oh, I see.
More important, 70th anniversary of India-Japan relations.
Sure.
Welcome back to the Nobel Laureate Science and Technology Seminar Series. We hope you all got refreshed for our next session. We invite Gautam Panda, Senior Principal Scientist and Professor, Central Drug Research Institute, Lucknow, General Secretary, Indian JSPS Alumni Association, and Dr. Maya Nankumar, Scientist G, HOD of Applied Biology and Division Head of Microbial Technology, Sree Tirunal Institute for Medical Sciences and Technology, Trivandrum, Treasurer, Indian JSPS Alumni Association. We welcome you both to the session. We will introduce the speakers and the chair can coordinate the proceedings. The first speaker for the session is Professor Tirunal Sajiki, Vice President, Gifu Pharmaceutical University and President, the Japanese Society for Process Chemistry. Sajiki Sensei was head of the Society of Synthetic Organic Chemistry, Japan, Tokyo branch, Board of Director of the Society of Synthetic Organic Chemistry, Japan, and Deputy Chairperson of the Public Pollution Investigation Commission of Gifu Prefecture. We request the chairs to invite the speaker. Attendees, please write your queries in chat box.
Okay, thank you for that introduction. A very good afternoon to all of you. Konnichiwa. This is the first post-lunch session. We have some interesting talks lined up. And the first speaker of the session is Professor Hironao Sajiki. Sajiki Sensei, over to you. Please give your lecture.
Okay, thank you. I'm ready. And thank you, Chairman, for your kind introduction. Thank you very much. And I'm Hironao Sajiki from Gifu Pharmaceutical University and currently president of the JSPC. It's the Japanese Society for Process Chemistries. It's sometimes confusing JSPC and JSPS. It's quite similar. Sorry. And I'm very much honored to obtain a lecture opportunity at this seminar. and thank you very much for everybody's. And before starting scientific talk, first, congratulations on welcoming the 70th anniversary of India-Japan diplomatic relations and the 75th anniversary of Indian independence. I just heard. Yeah, very much congratulations. And the Japanese Society for Process Chemistry, which I'm currently chairing, co-sponsored Indo-Japanese Conference on Process Chemistry R&D in Mumbai in 2014 with Department of Chemical and Petrochemical, Government of India, and the Indian Chemical Council. Then we have the opportunity to deepen and fruitful scientific exchanges and friendships. And we believe that this opportunity was directly linked to the subsequent technical exchanges among all participants. And the Japanese Society for Process Chemistry would like to continue maintaining good cooperation systems and continue to have more and more active exchanges. Thank you for your continued support. Well, let's get started with my scientific talk due to the limitation of the time. And today's my title will be a development of direct deuterium labeling methods, including application to continuous flow synthesis. First of all, deuterium is widely used in a variety of scientific fields and as a representative nuclide of a stable isotope. And the deuterium-labeled compounds are regarded as useful tools for tracing pharmacokinetics, quantitative microanalysis of trace ingredients such as residual pesticide, and optical fibers, functionalized liquid crystals, or organic ELs. And especially recently, heavy drugs, something like these, using kinetic isotopic effect is quite important. And especially deutetrabenazine, it's OSTET, was approved by US FDA in early 2017. Well, and finally, isotopic contrast by neutron scattering and so on. The major preparation methods of deuterium-labeled compounds could be classified three types: 1, 2, 3, 3 types. The first one is a total synthetic method starting from deuterated small raw materials. The second one is deuteration of reducible functionalities using deuterated reducing agent. And the post-synthetic, third one is a post-synthetic direct deuteration. It's so-called HD exchange reactions. Of course, HD exchange reactions are most preferable method due to the time, cost, and the environmental advantages. Then we have already established platinum group metal on carbon catalyzed easy HD exchange reactions. HD exchange reactions proceeding in D2O applicable to the wide variety of substrates, something like this. And these reactions are applicable to the wide variety of substrates, and in the presence of, especially important point is that in the presence of hydrogen gas as an activator of these catalyst metals. It's a quite important point. And deuteration patterns are particularly characteristic depending on the metal of the catalyst. I will introduce these in briefly. Then as part one, I will introduce HD exchange reaction under hydrogen gas atmospheric conditions. The benzylic site selective HD exchange reactions was adapted to a wide variety of substrates, as shown in this slide, including ibuprofen and estrogens as a medicine. And let's see, these reactions proceeded at around room temperature to 50 centigrades in nearly quantitative isolated yield and deuterium efficiencies, see, 98 to 93 or something like that. And further heat conditions facilitated HD exchange reactions even at the inactive non-benzoic sites, the alkyl side chains. The efficiency of the HD exchange reaction on the alkyl side chains temperature dependently enhanced and fully deuterated product were obtained at around 110 to 160 centigrades in a sealed tube. See, the side chain was nearly quantitatively deuterated. Okay, by the way, the HD exchange reaction on the aromatic ring systems was strongly enhanced by the use of platinum on carbon instead of palladium on carbon, especially phenol and aniline as electron-rich aromatic rings were found to be preferable substrates for the effective aromatic HD exchange reactions. For example, phenol was fully and nearly quantitatively deuterated at around room temperature. And aniline was fully deuterated at around 80 centigrades. However, the only drawback right here of the independent use of platinum on carbon and palladium on carbon is poor deuterium efficiency at the also positions of alkyl side chains. Due to the strict hindrance effect. However, the mixed use of platinum on carbon and palladium on carbon synergistically gives fully deuterated product as shown in this slide. And even includes the alkyl side chains and also positions. And these deuteration methods are applicable to a wide variety of substrates, including these building blocks and medicines, ibuprofen and oxytazine, D26 or something like that. And let's see, diamino, this type of this aniline derivatives are raw materials for liquid crystals and polymers. Then these HD exchange methodologies have already been launched in practical use by Fujifilm work or pure chemical corporations based on our cooperative study results. Okay, the next topic is the HD exchange reaction of alcohols, secondary, primary, and polyalcohols, could radioselectively proceed at the alpha position of hydroxyl group by the use of ruthenium on carbon as a catalyst. And the efficient incorporation at the alpha position of secondary alcohols proceeded at around room temperature to 50 centigrades for 3 hours. And the primary alcohols requires at around 80 centigrades for 24 hours. Under hydrogen atmospheric conditions, it's activator for the ruthenium metal itself. And in these reactions, flexible and liner alcohol can be deuterated with loss of the stereochemistry. However, the deuteration of stereochemically rigid substrate, such as cyclic sugar derivatives, preferentially proceeded by a stereoselective deuteration path. The wide variety of sugar derivatives, such as glycosides, like this, glycosides and disaccharides, amino sugars and ribose derivatives, and so on, were possessing multi-hydroxyl group within the molecule were also radio and stereoselectively deuterated, totally deuterated. Although the deuteration speed is obviously different depending on the steric crowding circumstance on the carbons. Please compare the position 3 right here is very much fast. And let's see, position 4, like this line is relatively slow. It's a difference. Okay. The ruthenium carbon right here was found to effectively catalyze the deuterium incorporation on the carbons adjacent to the only free hydroxyl groups, non-protected hydroxyl groups. And the HDXN reaction never proceeded at the alpha position of the protected hydroxyl groups. Even methoxide also no reaction. And therefore, free OH group is clearly essential for this HDXN reaction. Therefore, the HDXN reaction site selectively proceeding by the use of appropriately protected sugar derivatives, something like this. Then finally, quite inactive alkanes could also be efficiently deuterated under rhodium on carbon catalyzed conditions at around 160 centigrade under rhodium on carbon catalyzed conditions under hydrogen atmospheric condition. It's quite important. Not only Reiner alkanes, but also cyclic and branched alkanes underwent deuteration to afford a fully deuterated product with high deuterium efficiency. Well, let's go moved on part 2. This part describes the deuterium labeling method that does not add hydrogen from the outside without external hydrogen conditions. Okay, recently we have reported quite simple dehydrogenation methods of secondary alcohols under basic aqueous reaction conditions using rhodium on carbon or platinum on carbon as a catalyst indeed in H2O. Based on these results, deuterium labeling without external hydrogen gas conditions were newly developed right here and applied to the wide variety of substrates. For example, the present platinum on carbon catalyzed isopropanol D2O combined deuteration also smoothly proceeded at only 80 centigrades by the addition of cyclohexane as a solubilization agent, even without the addition of external hydrogen gas, in the same way as in the presence of hydrogen gas. The aromatic HDXN reactions, as shown in this slide, was applicable to a wide variety of substrates in excellent yield and excellent deuterium efficiency. And let's see, since only a tiny but sufficient amount of hydrogen gas for the activation of platinum metal is generated during the deuteration, these coexisting reducible functionalities such as aromatic carbonyl group and olefins could be totally survived under the deuterium labeling reaction condition. And let's see, I next investigated the deuteration of saturated fatty acids. Although platinum carbon, these schemes, platinum carbon has been traditionally used in D2O for the preparation of deuterium-labeled saturated fatty acids. The harsh hydrothermal reaction conditions and repetitive deuteration processes were required in conventional methods. due to the very much strong coordinable properties of platinum metal and carboxylic acids. However, our method, platinum carbon catalyzed method, various fatty acids, including bioactive valproic acids, its medicines, are efficiently deuterated using platinum carbon catalyzed isopropanol or isopropanol D8 and D2O combined solvent at around 120 centigrade. And a substrate possessing an easily transformable ketone within the molecules after undergoes deuteration without reduction of the ketones. And also dicarboxylic acid is also transformed to the corresponding deuterated, fully deuterated product. Well, next topics is deuterium labeling of acrylic and metacrilic acid derivatives, which are further susceptible to hydrogenation. Okay, after detailed studies for the related to the reaction conditions, platinum carbon catalyzed HDXN reaction of normal dodecyl acrylate in isopropanol D2O mixed solvent in the sealed tube efficiently deuterated product, efficiently to give the deuterated product. The alpha beta both unsaturated carbons. However, isolated yield quite low due to the polymerization of the normal decyl acrylate. However, the addition of a catalytic amount of hydroquinones as a polymerization inhibitors could improve the yield of the desired product up to the 73% yield and with excellent deuterium efficiencies on unsaturated carbons. And this method was applicable to the wide variety of acrylic and metacrilic derivatives. And furthermore, the obtained deuterium labeled benzylic acrylate and benzylic metacrilate derivatives could be transformed to a wide variety of organic deuterium labeled organic compounds, including polymetacrilic acid maintaining the deuterium efficiencies. And next, I'd like to talk about I'd like to introduce a part of ongoing HDXN project in our laboratory. Since the nitro-aldol reactions, like this, nitro-aldol reactions, by the deuterium labeling of nitroalkanes during the reaction, during the reactions such as nitromethanes has never been developed. We have developed direct deuteration and synthetic method of beta deuterated and beta nitro alcohols in D2O, using a heterogeneous basic resins, so-called WS30, and quinine derivatives. Quinine derivatives as an organic catalyst for the enantioselective synthesis. The deuterium labeled beta deuterated beta nitro alcohol alcohol derivatives could also be transformed to a variety of deuterated syntones such as beta nitrostyrene and beta amino alcohol derivatives with retention of the deuterium efficiency. And various racemic is all racemic catalyzed by WS30 and chiral catalyzed by quinine derivatives. Beta deuterated beta nitro alcohol were effectively produced with high deuterium contents and excellent enantiomer excess. And we have also developed a practical continuous flow synthetic method for deuterium-labeled beta-nitro deuterium-labeled beta-nitro alcohols by the use of catalyst cartridges. Catalyst cartridges containing WA30 beads, WA30 beads as a catalyst. The present continuous flow reaction systems using WA30 was successfully employed to catalyze synthesis of deuterium-labeled beta-nitro alcohols without degradation of the catalyst. That means the WA30 without degradation of the WA30 at least 72 hours with 93% isolated yield. 72 hours means 3 days reaction is possible. 3 days continuous flow reaction was possible. And with 93% deuterium efficiency. That's excellent results. Then since the cost of the D2O is a problem in HD exchange reactions, we are vigorously promoting the development of continuous separation and circularization method of D2O along with the development of efficient catalyst for various HD exchange reactions possessing robust and low flow resistance. That's quite important. Then finally, I'd like to conclude. We have developed a wide variety of efficient deuteration methods in D2O, and these results will contribute to a wide variety of scientific fields as convenient preparation methods of deuterium-labeled building blocks, something like these. Then I'd like to say special thanks to my colleagues. These pictures are my faculty staff in my laboratories. The first 3 pictures were former faculty staff in my laboratories. And the last 4 pictures are current faculty staff in my laboratories. And also many of the undergraduate and undergraduate students were related to the HD exchange reaction project. Then I'd like to say special thanks to all of them. And JPAC, Fujifilm Work of Pure Chemical Corporations, NE ChemCat Corporations, and Canon also supported to the HD exchange reactions. And finally, these official grant also supported to our HD exchange project. And I'd like to say special thanks to all of them. Okay, well, that's it. Thank you very much for your kind attention.
Please unmute. It's not audible.
Thank you, Sensei, for that most illuminating talk on deuterium labeling and its myriad applications. The session is now open for questions.
Okay, sure.
You can place your questions in the chat box also.
Over to you, Chess.
Okay, it looks like there are no questions. So, Sensei, you have been able to make yourself very clear to your audience, and thank you very much. Arigato.
Okay, arigato.
I have one question.
Oh.
Okay, please.
Go ahead.
Yeah, actually, yeah. Professor Sajiki, this is Dr. Gautam Panda from CDRA, Lucknow, India. This is a nice method for incorporation of deuterated compounds.
Yeah.
I am only wondering whether your methodology has been applied to incorporate deuterium into some drug molecule or some bioactive compounds. because these days deuterated compounds and drugs are very highly demanding for innovating of new chemical entities in drug discovery research.
Right. And let's see. If you would like to use these methods, anytime welcome to teach and I'd like to show you more detail for the methodologies and experimental sections. Then please contact to my email address, please. And especially medicinal chemistry fields, next generations new drugs, that means heavy drugs is quite important. Then deuterium labeling methodologies will be able to widely used in the medicinal chemistry field, I think.
Okay. Okay, thank you.
Okay, thank you.
I have one question.
Sure.
Hello.
Hello.
Yes, I have one question for Sensei.
Sure, please.
How do you characterize these compounds? Because you seems to have there are a lot of deuteriums which are obviously NMR inactive.
Right.
So what is the best way to characterize these compounds?
Okay, and first of all, proton NMR, fully deuterated product was disappeared. Yeah. Then we are using DNMR, that means the deuterium NMR systems and high mass systems. The mass can obtain the quite important information, especially for the deuterium efficiencies.
Okay. So the stereochemistry will remain same as the hydrogen molecules, right? The stereochemistry does not change much.
Yeah, actually, but okay. During my talk, I introduced total 40 or 50 deuterated products, especially alkyl derivatives. Okay, at that time, okay, on the regular TLC was moved. Then it's physically different, but okay, 2,3 deuterium atoms are incorporated to the product. Physical and chemical property doesn't change. Then it's a good point, I think.
Yeah.
Arigatou gozaimasu. Thank you very much.
The next speaker is Professor Sree Ninomiya, Emeritus Professor and Project Professor at International Field Phenomics Research Laboratory, Graduate School of Agriculture and Life Sciences, University of Tokyo, Japan. Professor Sree Ninomiya is the incoming president of CIGR and member, Agriculture, Forestry and Fisheries Research Council, Japan. Professor Sree Sensei is an expert in the field of plant phenomics, biometrics, applied statistics, applied genetics, agroinformatics. We request the chairs to invite the speaker.
Professor Sree Sensei,
Yes.
over to you for your talk on smart farming for small scale farmers.
Am I audible?
Yes, you are, sir.
Okay, so am I sharing my window?
Yeah, clear.
Okay, thank you very much for the very kind introduction. My name is Sree Ninomiya from the University of Tokyo, and I'm very glad to have an opportunity to give a talk here at such an important meeting of 75th anniversary of Indian independence, as well as the 70th anniversary of Japan-Indo official diplomatic relationship. Okay, so I think I'm probably the only one speaker from agriculture sciences this afternoon, and also I'm very glad to be given such an opportunity from this sector. Okay, this is so-called planetary boundaries. This is a kind of quantification of 9 risk boundaries for reversibility on Earth system, on our planet, such as novel entities, climatic change, biodiversity, land use, freshwater use, etc. and the green indicate still the class is in the safe status, and yellow indicates the quite increasing risk and the status, and also the red or orange shows indicate the high risk. For example, the biodiversity is facing really high risk. It's almost impossible to get it back to the original position. And also nitrogen cycle is terribly bad. It's almost impossible to get back. Like such as novel entity, for example, we have not quantified yet. So we don't know what's going on with this, but novel entities such as the plastics from the agriculture should be affecting something else. So anyway, when we think of the relationship of the agriculture to those boundaries, the situation is quite serious. For example, agriculture is quite responsible for greenhouse gas emission. Actually, 25% of gas emission is caused by agriculture activities, for example. Just a few people know it, but actually it is. And also biodiversity has been highly impacted by chemical uses by agriculture. And agriculture is the main user of land. And also agriculture uses fresh water. And of course, agriculture uses much fertilizers, chemical fertilizers nowadays, which is affecting nitrogen cycles really badly. Now, in the 21st century, actually in 20th century, we were quite successful in maximizing productivities using chemicals. But in 21st century, we cannot follow the same way. We have to realize both productivity and sustainability at the same time. It's a really big challenge for us. And now I think some of you have heard of smart farming, which tries to utilize the IT and information science and robotics, etc., to provide improves the productivity and also addresses the labor shortage. But we expect that the smart farming also can contribute to the sustainability. Just I'm showing two examples here. One is so-called pinpoint pesticide spraying. So usually people just spray the pesticide over the canopy. But if we can spray, just conduct a pinpoint spray so that we can reduce the amount of the pesticide. Also, it can reduce the cost because pesticide is quite expensive. So it also helps the farmers. And also, of course, we can reduce the impact on the environment. Variable fertilizer application. It's a kind of optimized fertilizer application based on the condition, local condition of the canopy, the field, so that also we can reduce the amount of the application of chemical fertilizer, and so on. So now people are expecting that this kind of smart farming should contribute to realize both a productive and sustainable agriculture productions. And let me quickly summarize about smart farming. In agriculture, agricultural management in the field, there are 3 steps. The first is the farm field monitoring, just to observe the crop status and observe environmental conditions such as weather, soil, water, etc. Then based on the observation, farmers make some decisions about their farm management. Then based on the decisions, they go to the field to conduct farm management. This kind of 3-step cycles is always going on. So in conventional farming, the first step, the observation is done by visually and manually, which is inconvenient and quite subjective. And the decisions are made quite empirically based on the observations. Finally, the farm operation, farm management is conducted by human, which is quite laborious. And because this kind of management is quite strongly depends on tacit knowledge of the farmers, it is quite difficult to transfer to the next generation. While in smart farming, the first step is done by so-called IoT sensors, which is automatic and objective. And the decision can be made by so-called, very simply to say, artificial intelligence based on the observations by IoT sensors. Then the farm management can be conducted by robots or auto navigation systems, etc. And because everything is based on the data, it is quite easy to transfer the technology to the next generation, etc. But there's an issue that most of the current development of the smart farming technologies focus on large-scale farmers in developed countries. You see that I just mentioned that the robotics and things. No, I never think that they can be acceptable by those small-scale farmers. So we do need smart farming technologies for small-scale farmers in Asia and African countries. As I mentioned, expensive hardware-oriented smart farming technologies such as autopilot robots are not acceptable for those farmers. And now, to address these kind of issues, we have been conducting a Japan-India smart farming research project since 2016, and mainly targeting software-based smart farming technologies, because we noticed that now the popularity of the smartphone is increasing very quickly, even among the Indian farmers, and we thought of utilizing such equipment. Okay, so today I would like to just introduce you about the project. This is a Japan-Indo international project being supported by DST and JST, Japan Science and Technology Agency. And we try to solve the problem using a latent information science and information technology, such as data science, artificial intelligence, and IoT sensors. Our site is located in Hyderabad, in the middle of Deccan Plateau, a typical, with some terms of missing, a typical semi-arid area of India. And from Japan, there are several different graduate schools of University of Tokyo are participating, while from Indian side, IITH, Indian Institute of Technology, Hyderabad, IITB, PJ Telangana State Agriculture University, located in Hyderabad, and also IIITH are participating in this project. Not only for developing technologies, but also we are planning to establish Japan Indo Joint Research Hub here in this location to sustainably continue this kind of activities in India. And well, I don't think I need to explain about the background here, but demand for the food is expected to increase even in India, rapidly India, due to population growth and most important things, expansion of middle class people, because due to the economic growth, which requires usually more food, which is taking place everywhere in the world. And 80% of arable land in India is semi-arid. And also, I didn't know the total arable land of India is the second largest after the United States, a huge land in there. And now the production in semi-arid region is threatened by climate change. And in the semi-arid area, they have two seasons, rainy season and dry season. And even during the rainy season, the farmers are suffering from the shortage of water. And of course, the farming in the dry season purely depends on the groundwater stored during the rainy season. So the shortage of the water in the rainy season caused the shortage of water in the dry season as well. So, and also more than 50% of the entire population are farmers in India. And we need to improve the farmers' profit, and is also an urgent task. So we set up the three objectives in this project. And the first one is to develop a system for optimal irrigation in the dry season corn crops to conserve the water use. Because the farmers, we found that the farmers tended to use too much water. Overuse of water causes problem with water shortage. The second, the development of information transmission method to solve the problem of individual farmers. It is quite easy to show the kind of general solutions to the farmers, but the issue that each farmer has is quite different among them. So we have to provide customized knowledge to each of the farmers. So we are developing a kind of system to transfer customized knowledge to each of the farmers. The number three is a bit different from the above two. we just developing an efficient breeding system for rain-fed paddy rice cultivars, which can be grown under the shortage of water while producing sufficiently high yield and sufficiently good quality. Now we have 6 working packages under this project. Number 1 is to develop IoT sensor system in the field, and working package number 2 handles the big data things collected by IoT sensors or drones, because we have to have the very good platform to handle big data. And the number 3 is the AI system to analyze the data collected by the IoT sensors. Based on the 3 basic technologies, we are trying to develop the 3 different systems based on the objectives. The number 1 is additional support for the crop management, such as optimized irrigation. And the number 5 is efficient knowledge transfer. The number 6 handles high throughput breeding system. Now, for the working package number 1 for the efficient data collection in the field, we have been fully utilizing the drones, which price has been decreasing very quickly, and it's not very difficult for us to use the drone there. And we have developed a kind of pipeline to collect the images of different images like RGB images, hyperspectral images, multispectral images from the drones. And also we, particularly in the semi-arid area, soil moisture sensor is particularly important, and we've been trying to develop the very cheap soil moisture sensors. It's cheap, also it can be easily handled by the people. So this one sensor you can see on the left, the bottom left, is a fully autonomous sensor. It doesn't require any solar panel, but a very small battery can run this system for almost 1 year to collect the soil moisture and transmit the data to the mobile network, and etc. Okay, and now finally we are trying to combine the different data from the field sensors and drones, and sometimes from the satellite images, etc. altogether. To do this, we need a so-called interoperable data platform is necessary, and in a conventional system on the left-hand side, we used to develop a system to each of the sensors, because each of the sensor is, those sensors quite different. They can generate the differently formatted data, so that the same application cannot utilize those sensors. So that finally they have to provide a different application for each of the sensors, but it is quite really nonsense. So what we did was to provide a kind of intermediate platform which can absorb the heterogeneity of the different sensors altogether, so that all the sensors look the same from the applications. Those platforms can provide a consistent access to those different applications, so that we can maximally utilize those data collected from the different sensors. And working package number 3, we mainly focuses on utilization of images collected by the drones or ground-based cameras. And I'm showing some of the examples. Those on the top left show the automatic detection of the rice panicle heading, and also we can count the number of the panicles automatically. And on the right-hand side top is showing 3D reconstructed. This is not the real image, but a 3D reconstructed image of this case is a soybean. Of course, we can do it the same for the rice as well, and we can estimate the height of the crops, and we can estimate the leaf area of the crops quite easily. And on the left bottom, it shows the growth curve of the rice canopy, for example. Now it is almost impossible to measure the canopy coverage by human, but by using the drone images. It is quite easy, and just we can have the growth curve, how they grow by. So, and this is an example of the 3D reconstruction. This is case for the orchard, for the pear or something else, and in this project, it's highly requested to I mean, so kind of social implementation is highly requested. So we succeeded in licensing this 3D reconstruction technology to NEC, one of Japan major company for the electricity. And working package number 4, so optimal irrigation and fertilization support system has been developed. And as I have mentioned at the beginning of this presentation today, we found that the conventional irrigation uses too much water because they don't have any support by data-centric approach. They just empirically uses water. So usually they use too much water. And to monitor water stress, so to provide the optimal timing of the irrigation, we have to monitor the water stress of the crops. So in this project, we have been trying to take two approaches. One is to use the soil moisture sensor, and now we have developed a model to predict the future soil moisture by using a very single soil sensor. We don't need many sensors, but single soil sensor is sufficient to predict the water soil moisture in the future, so that we can send a warning to the farmers, asking them to conduct irrigation. And the number 2 is to use the hyperspectral drone images to monitor the water stress conditions. But the study is still going on, so yeah, quite soon we need to verify the most cost-efficient way to monitor water stress. And finally, we are thinking of utilizing smartphones to transfer knowledge to the farmers in this case, to let them know the most optimal timing of the fertilization and irrigation. And the other one, working package number 5, I just mentioned that in this working package, we've been developing a knowledge transfer system, customized knowledge transfer system. And now we have released Android application named CrocDarpan. Yeah, you can access it and you can download it from Google Play Store. And now more than 5,000 copies have been downloaded, given 4.3 rated out of 5. It's quite good, but now still only available in Telugu and English. So the farmers in Telangana state can only access this system at this moment. But because of the very good design of the application, the localization of other languages, other Indian local languages, is not so difficult. And now this application can provide the information of pest and disease identification and management method to handle those diseases on cotton and rice, paddy rice. And we also adding new databases for other crops as well. And something very interesting about this smartphone application, or this algorithm we implemented in this smartphone, is that understanding that the farmers are not able to ask the proper questions to the system. So this system first asks the farmers, list up some of the questions, then the farmers choose one of them, then finally they can reach the knowledge they need to know to identify the pest or diseases, or to know how to manage such pest diseases. And this is a very simple tree structure, the Q&A system. But depending on the answer of the farmers, the tree structure is dynamically reorganized. It's using a kind of AI approach. So the algorithm using this application is very, very new. And working number package 6, we are developing a smart grading system. And before explain about what we are doing, I need to explain about how we try to accelerate the breeding system. In conventional breeding, okay. So when we have the new genotypes, we have to conduct a selection to choose the better genotypes. But usually we have to grow them in the field to know the disease resistance, or to know about the yield, or to know about the growth performance, etc. We have to grow them in the field. But so-called genomic selection, we don't have to grow them, because to grow them, we need 1 season. It takes a long time, which makes the breeding a very, very long business to produce just only one cultivar. For example, in case of rice, usually it takes almost 10 years, or even more than 10 years, to have a new cultivar after the crossing. But based on the idea of the genomic selection, we don't have to grow them, because we try to predict the performance of the crops using genomic information. Like I'm showing a very simple formula here, but we use, because it used to be quite difficult for us to have the full genomic information. It cost a lot, and it took a really long time, but it takes only a few days and doesn't cost much to have the full sequence information of one genotype. So just having, if once making this kind of prediction model F, then we can predict the performance of crops without growing them in the field. This is the basic idea of our approach. And now we have this kind of field for the crop paddy rice, and you see by flying drones every 3 days, we can have this kind of a 3D reconstructed field of different genotypes that we are handling more than 400 different genotypes to breed the new varieties. And by flying them, we can have the flying over the canopy of these plots. We can have this kind of growth curves for each of the plot quite easily by flying drones every 3-4 days. So this kind of growth, in agronomy, this kind of growth curve is very important. This can be a very good information to know the growth performance of the cultivars, right, crops. And also now we have developed the estimate, for example, the rice grass height from the genotypes, etc. But still the study is going on, but we have already released the smart breeding support system like this. And using this system, we can identify the important genes which may control the trait, such as the height or yield, etc. And also we can predict performance of the crops, such as yield or height, etc., based on the full genomic information. And this kind of analysis or calculation is very, very difficult. So usually the breeders need the help of the experts to conduct this kind of analysis. But now this web-based system can let those breeders in the field can conduct this kind of analysis quite easily by themselves. So now we have held several different workshops and research progress meeting, etc. And unfortunately, last couple of years, we are not able to visit each other. But still, by using online meeting system, we quite often have some discussions to continue the studies. So yeah, of course, some part of the project have been quite delayed, but still anyway, somehow we're doing kind of researches quite actively. And yeah, also this project has provided a good opportunity for those young scientists, young students of both end, Japan and India, learn about the different cultures and different way of the cultivation, etc. And this is has been very good training training place for those young scientists and students about how to conduct international collaboration and how to discuss with people from having different cultures, etc. It's really, I was very happy to see those growth of the young scientists and students very much. So anyway, and also we had an opportunity to interact with the farmers in Indian Japan, and some of the Indian students and Indian scientists visit Japan and visit Japanese farmers as well. And of course, the Japan side also interacted with Indian farmers as well. So in this project, we have been fostering startups, and we have been collaborating with several different startups from a university, and one of the top one, Quantumix, was developed by our group during this project. Okay, and well, I think the time is almost gone, and well, if we discuss about the relationship of this project, SDGs, it is quite interesting. Of course, the number 2, zero hunger, and the number 3, good health, was directly related to this project, of course. But we are trying to support the farmers, so indirectly we can solve the issue of the poverty. But also, we are thinking of the water saving, which is related to number 6, water. And also, we are trying to save the water and the chemical uses in the land, which helps the biodiversity, number 15, etc. So, of course, it is directly related to climate change. So anyway, our project is almost related to almost all the targets of the SDGs. Okay, so there are the list of the people, and the Indian side PI is a Professor Uday Desai from the IITH, and he has retired, but still he's been the leader of this project. Okay, thank you very much for your attention. Thank you.
Thank you, Sensei. That was a very interesting talk on sustainable technology for maximizing agricultural production. And it was very interesting to hear about the Indo-Japan collaboration in that area, and how the small scale farmers who form the majority of the farming community in India is going to be helped by smart technology. Thank you, Sensei. The session is now open for questions. There is some questions posted in the chat box. Mr. Shyam Patrail has said, Sensei, are you using citizen science approach to collect data directly from farmers to get inputs about smart farming?
Very good question. Yes, we are thinking of it. Actually, from the working number package number 5, yeah, we released crop Dalpan, and this is a kind of way to collect the data from the farmer side as well, and the new way of getting kind of the crowdsourcing way to the data collection. So I don't know if we can say it directly the citizen science, but I mean the farmers are sensors of our project. So it's a new idea that we just recognize that the farmers can be a good sensors. Yeah, we cannot deploy the good, very expensive sensors in the field for the small scale farmers, but by using, for example, smartphones, they can be good sensors.
Yeah, yes, that's true. Another question from Mr. Nagesh. The question is, what are the differences you find among Indian and Japanese farmers?
Well, it's okay. So the first common thing that we are growing paddy rice.
Okay.
Yeah, that's rice is our major staple food. Maybe, yeah, quite similar Indian side as well. And well, because The Japanese farmers are anyway able to purchase a little bit expensive tractors and things, but the scale of farming is not really large. Okay, the government is pushing the farmer to expand the scale of their farming, because the many of farmers are quitting their farming because of the age. So those farmers, young farmers, try to collect the land from those farmers who have stopped farming. So in some part of Japan, the farm scaling is increasing quite quickly. But anyway, still the farm scale is a kind of common thing between two countries, I guess. So maybe the biggest difference, maybe
Okay, thank you, Sensei.
not very different, I guess.
Okay, maybe the poverty among the Indian farmers, especially the small scale farmers, is a hindrance to them even harnessing technology. But I am very happy to hear about this initiative by the India Science Forum. Thank you, Sensei, for that most illuminating talk.
You're welcome. Thank you very much. Okay.
Thank you,
Any more
Sensei.
questions?
The next speaker of this session is Professor Neelam S. Sangwan, Dean, Central University of Haryana. She is a fellow of National Academy of Sciences, Indian National Science Academy, National Academy of Agriculture Sciences. Her expert areas include the biotechnology, genomics, transcriptomics, plant natural product, secondary metabolism, medicinal and aromatic plants, Ayurveda, anti-cancer therapeutic molecules. We request the chairs to invite the speakers.
We invite Professor Neelam Sangwan, Dean, Central University of Haryana, India, for delivering her talk. Madam, please.
Thank you. Thank you so much. My slides are visible.
Yes,
Can you
it's
see my
visible, ma'am.
slides?
It's visible.
Yes, ma'am.
Okay.
You can put it in presentation mode.
Will you please put it in a full screen mode?
Is
Yeah,
it
good.
now?
Yeah, good.
Thank you very much. First of all, I thank Professor Mohan and organizers for giving me this opportunity. And thank you, Chair, for introducing me. So I'm going to speak about the traditional medicinal plants. This is my title, From Crude Drugs to Molecular Medicine, Emerging Scopes and Opportunities. So the talk is basically about the medicinal plants which are used in various traditional system of medicines. We all know that plants are known to be used in various healthcare systems since prehistoric times. And before the introduction of the chemical medicines, humans have basically relied on the properties of the plants. So some of the plants, in fact, they are the Sree recalls that they are thousands of years old in their use in various systems. And we can see that billions of people, especially belonging to lesser developed countries, they are still employing medicinal plants as their healthcare system. So that there are major couple of traditional systems of medicine, very important and very famous and popular ones are Ayurveda, belonging to India, which is thousands of years old, then Chinese system, then Kempo system, that is a traditional Japanese system, then traditional Korean system, Unani, which is now integrated in Indian system, and many more. So the basic is that all of them are accepted by the government as a tool for as a healthcare system, and also many of them, most of them, in fact, employ medicinal plants for the curing abilities. So let me just give a very small and very popular example of the plant Sanjeevani. Everyone, in fact, in India knows about the plant or might have heard during it comes from, its mention is in epic Ramayana. and it is used for reviving the, the person, the Lakshmana who was about to die. So see how old is this mythological conception about the medicinal plant, and we will see how these can be scientifically proven to be true. The only requirement is the scientific evaluation or scientific validation. And recently there was a mention of this Sanjeevani plant, or Sanjeevani name, I should say, by the Brazilian president when Indian Prime Minister sent him the COVID vaccine. This was last year. So I have attached the postcard where the Sanjeevani carrying Hanuman picture was sent by the Brazilian Prime Minister. So this is just to mention that these medicinal plants, they have a very prehistoric mention even in the epics and even mythology, but they do have a lot of experiences of people which have culminated them into the traditional practices. So now just giving a glimpse of the type of number of molecules that this plant Sanjeevani could have. We can see a large number of structures, chemical moieties of terpenoids. There are lignans, there are flavonoids, there are polyphenolics molecules, alkaloids, and also there are unique molecules which are not found in any other plant. These are known as seleginylis. So this is just to mention that it is not just mythology. Mythology has its roots, and because of many novel molecules and novel phytomolecule, the properties of the medicinal plants are bestowed with. So the medicinal plant in general, we all understand it's a very important aspect, and not only for the traditional medicine, for herbal industry, but side by side, this also provides a livelihood to the large segment of people, to the farmers, especially which are, which are the marginal farmers or low income group farmers. And so far as the herbal industry is concerned, it is going to be increasing, and there is a very positive indication that these industries, which stands at $120 billion, is going to expect it to reach to $7 trillion by 2050, because more and more people are now having interest towards the medicines from the plants. So as per the last count, if we check through the database, there are 3,26,000 compounds which have been isolated from different organisms, and they have various properties, many of them who have been shown to be associated with various properties. Now, coming back to they are called as bioactive compounds. Why they are called bioactive compounds? Because they show the activity in the biological system. And in general, if we call them plant secondary metabolites, because they are isolated from plant. And it was believed that they do not have any apparent function in the growth and development of the plant. However, this concept is now changing. They are very much involved in various processes of the plant. And different plant families, the main important thing is that they are very unique molecules found in unique families of the plant. And even within the family, depending upon the species, they might be present and they might not be present. So meaning thereby their existence is very, very specific. And these molecules have very important medicinal functions. Let me take you to the one successful story of plant medicine from the plant, and that is the Artemisia annua. So the anti-malarial drug, that is the crude drug artemisinin, is now established medicine. And this molecule, which is known as artemisinin, it is having a unique peroxide endoperoxide linkage. That's why this molecule is very unique. And this was isolated by this lady pharmacologist known as Yu Tutu. She was the head of the research of Project 523, and she was to work on the looking for the treatment for the fever. And she worked on various traditional system plants used in traditional systems. She narrowed on Artemisia annua. And finally, this artemisinin was found to be the active against the malaria. So further researches at CSIR, especially at Lucknow lab, CSIR CDRI, as well as CSIR CMAP, they have done a lot of work in this area. And finally, the clinical trials were done and this drug has come into the market. So recently, these two, you can see the field, the plants, artemisinin plant, Artemisia plants, growing in the field. They have different canopy, and as the molecule is required for the preparing the drug, which is now available in the market. So there are two aspects. Either we can have the drug from the recombinant manner, that is in Saccharomyces. That is what the Bill and Melinda Gates Foundation supported this program of Kiesling, and this drug was prepared in Saccharomyces cells. And the another option remains to have the plants with higher concentration of molecule. So this is the plant which has more than 1% of artemisinin in its leaf and roots, and this was achieved by using various molecular, metabolic, and genomic approaches and molecular breeding coupled with the chemistry and clinical biochemistry. So now coming back to some of the salient observations and salient achievements or salient work related to the ashwagandha, another very important plant. The medicinal name is Vidania somnifera. So this plant is belonging to the family Solanaceae, which has given us most of the vegetables like potato, tomato, chilies, tobacco. So these are all belong to the family Solanaceae. And this mention of this plants, that is Vidania somnifera, is in various Ayurvedic preparations. More than 200 preparations are commercially using this plant. And many of the activities related to its properties are established. And most of the properties, it is believed that they are because of the molecules with enolite. They are carbon-28 based secondary metabolites. So this makes it an important plant. So the research direction involving several laboratories, CSR laboratories, various other knowledge resources and previous reports, as well as students and colleagues, we all focused on holistic direction involving many things at a time, so that we understand the plant properly and we try to handle the target, that is to look for the Vidania plant, look for the ashwagandha plant, which has best qualities to be treated as a medicine. So we conducted our R&D on ashwagandha, including biochemical, phytochemical, genomic, transcriptomics, metabolomic studies, and explored its constituents, its pathway, its diversity, because diversity makes one thing work or it may not work at all. Because when these molecules, if their diversity is not in a desirable manner, then even if they are present, they may not give the desirable effects. So that is why this is very important to understand. And then side by side, engineering of ethanolite biosynthetic pathway, as we were looking for the artemisinin biosynthetic pathway expression in the Saccharomyces, so that can be an analogous manner to have these molecules. So that using the biotechnological intervention and a conventional way to develop a superior variety and formulation which has its pharmacological property. So the traditionally acclaimed therapeutic properties are many, which are shown here through our Charak Ayurveda system. And to summarize it in a simple way, the traditionally the Vidania somnifera has been attributed with insomnia, rejuvenative property, nervine property, life prolonging, that is anti-aging, and increased energy, youthful vigor, endurance, strength, health, and blood purification, treating asthma, and so many properties. They are mentioned in the traditional literature. So the recent research findings, these are the properties which have been documented in literature, various recent literature that are showing the ashwagandha plant to have the anti-inflammatory, immunomodulatory, anti-apoptotic, hormone, regulatory, adaptogenic, cardiovascular, bone health, anti-hypertensive, neuroprotection, antioxidant, hypoglycemic, as well as in the hepatoprotection. So virtually, if we say a large number of properties are attributed to the plant. So the root as well as the shoot part, both the part have specific properties, and some of these properties were shown by established by our group in collaboration with CSIR, CDRI. So if we see that the number of the publication coming up in the global interest towards these plants, this has been increasing. And from 2006 onward, there is a sudden increase we can see in the number of publication, and we also feel fortunate in having contribution to this, because we also started having intense research interest in this area. So now the plant has a global interest out of the only Indian laboratory. Now various other laboratories are also working in this plant. That's the flagship herb of Ayurveda, and now so many of the preparations, they are available on the Amazon as well. So the export-import of these molecules, just to give an idea, the interest on this plant is very high. And this is owing to, again, the market potential of this molecule. And the challenge is that the component of, or the presence of this molecule is very, very low compared to the similar story of artemisinin, where artemisinin presence was very, very low. So these molecules, they are present in the plant at a very low concentration, and some of them rather traditionally roots were used more. So even that is more difficult, because obtaining anything from the root, then we have to uproot the entire plant, and the root recovery or the root yield is always lesser than the herb, so, or the aerial part. So that is a limitation. There are many challenges if we look into taking any medicinal molecule to the drug. In the morning lectures as well, it was very well highlighted that how difficult it is, because there are so many factors that we need to establish before we can take any molecule to that level. And if we are taking from the plant, there are many challenges, including climate, including the long life cycle of the plant. If it takes longer time, so it has to be integrated into the farmer's cycle of the crop. And if it doesn't fit it well, then that is also one of the negative points. Then I mentioned the low root yield and high fiber content. So in case of ashwagandha, this plant, the quality of root in terms of fiber, it is one of the important criteria that is still many of the industries they take in. So these are some of the points. So what do we need? What is the first point to work upon? So we feel that the first thing is to understand the plant in a systematic manner and try to have a uniform plant type where the valued products can be prepared. So here it is an example to show what it makes a difference if we don't narrow down on the uniform plant type. We studied a couple of products of based on the ashwagandha, and when we analyze it in terms of a molecule, the active molecule, we found there is about 70-fold variation in the component. So that amounts to a huge variation, and that brings out to our attention that the quality of the herb is one of the most critical issue. And one of the parameter how we can control is to have a uniform, specific, defined, high-yielding variety or engineered metabolite. So as we can see, these are the major types of molecules, phytochemicals, which are present in the ashwagandha root and ashwagandha plant. And these molecules, you can see when they vary, obviously, the therapeutic potential are going to vary. So the occurrence of these molecules, there are a number of molecules. I'm not going to name them. There are too many. And their proportion, their presence in leaf, root, stem, and various other portion, it keeps on varying. And this is the main key point to look upon. So this molecule, that is the vidanolite, the major molecule that was the main focus. They are synthesized through these steps, and as can be seen, it is a very complicated step. Too many number of steps are involved in the biosynthesis of this molecule by the plant. So obviously, this molecule, the chemical synthesis is really very difficult. Now, coming to as most of these molecules, they are structurally very similar. So their resolution is also very difficult. So what happens when we try to take any plant and evaluate its quality? So we have to be equipped with the parameter that can distinguish two very close sister molecules, two very close isomers, or two very close molecules where OH is just varying in one position. So we developed specific methods for the HPLC-based methods where the detection of molecule which are very similar in their structures can be made out. And utilizing this method, we did the analysis of the various chemotypes, various extract and plant type, and that enabled us to understand the qualitative differences as well as the quantitative differences among various kind of extracts that we are preparing or optimizing the extract, or also in the selection of the accessions of the plants. So obviously, for any going for further optimization or standardization, or for isolating this molecule in the large quantity, our target was to have a elite variety or elite plant type of ashwagandha, where a particular marker molecule, lead molecule, is present in a higher amount, and then we could have a enriched root extract where we can chemotype it and we can assign the proportion and the marker molecule in it. So for having the phytoconstituent accumulation well understood, and as well as to have make the attempts for future to improve their presence, we try to understand the biosynthetic pathway. And during that time, absolutely there was no information available about how these pathways are, which is a pathway, how they are synthesized. So the first step was to understand the two pathway, and the MBA pathway, which is involved in the synthesis of IPP, and DOXP pathway, which is involved in the another unit of IPP, that is DMAPP. So these are the two major pathways which combine together to give the terpenoid or steroid pathway. So the question comes that when if we are looking into the biosynthesis of these molecules in the root, so our first question was whether the roots are synthesizing these molecules themselves, or because they do not have the carbon precursor. Carbon precursor mainly comes from the aerial portion. So are these transported from the root as in the case of alkaloid? So we created the orphan root, we created the discrete shoot system, and then we did the using the isotopic methods, we identified and we found that both the tissue systems, they have independent biosynthetic potential. this is not related to their transport. They both have their dedicated system of biosynthesis. So here comes again the pathway. It shows the role of DMAPP, that is the plastid. The another one shows the cytosol. So as the IPP and DMAPP, they join together to form this molecule. Another observation that we wanted to make, that what is the way of their synthesis, and using 13C enrichment experiment, we were able to assign which are the carbons which are coming from the DOXP pathway and which are the carbons which are coming from the IPP pathway. And by superimposing them, we could assign that the biosynthesis of this molecule within a molecule, it happens in the isoprenogenetic way. Now, after having understood the certain aspect about the biosynthesis of this molecule, now the attempt was how can we improve, how we can understand what are the factors which are involved in their qualitative enhancement as well as quantitative accumulation. So for quantitative, that means when the production, overall production is more, that means we need to look for the metabolic flux, and this metabolic flux at the molecular level, it will be governed by the triterpene synthesis and transcription factors. Whereas the another aspect that is very important in case of this plant, that is a metabolic diversity, because if we are having very closely related molecule, but we need only one or we need lesser the another one, then the cytochrome P450s are important because they will control this diversity. So we try to study this triterpene synthase pathway. The gene is central. From this point, there are number of branch pathways takes place. So we identified the gene which is involved in this step. This is 2.2 kb gene, and we expressed in the plant systems. Similarly, for vithanolite metabolome diversity, because this is the it is controlled by the cytochrome P450s. So the another step is required, which is for the diversification of the diversification of the main frame to convert it into various type of molecules. For having understanding these two things, we needed alternate biosystems of ashwagandha. So which can be fastly grown because a plant will take more than 6 to 9 months to grow, and then that cannot be controlled. So we wanted a control system. So we cultured and we prepared in vitro systems, callus system, micropropagation system. We prepared some cell suspension cultures, rhizogenic root, and as well as the entire micropropagation protocol, which was efficient and utilizing less time. This is then we also developed the genetic transformation system, because having understood the gene, then it need to be incorporated, and this genetic transformation system was applicable on the six number of accessions that we tried. So it was a sort of the process which could be employed to a larger number of accessions, because these accessions were varying in their phytochemical composition. We express the triterpene synthase gene, and so the overexpressions line showed the enhancement in the vithanolite content. So this means the gene which we located to be the centrally positioned, this is responsible for the overall flux regulation. The another step that was the cytochrome P450 diversity. So again, here we can see that this diversity is because of the P450s, because they are carrying out oxygenation and hydroxylation reactions. We mined out by this time, we also had the with us the transcriptome. So we mined out about 179 transcripts from the plant, and we finally narrowed down to the 4 they were being the homologue of the homologue pair, and two pairs, they were closely related to each other. We did their computational modeling and studied their active structures. And we also created how this network of cytochrome P450s can be positioned, and how different molecules we can use these candidate transcripts which are with us. We check their expression profiling, and depending upon the tissue they come from. And we also used this gene to clone, and one of the gene in the central, you can see it was expressed in E. coli, and successfully it could hydroxylate one with a furanate molecule to its hydroxylated product. Another example of transcription factor family genes, which increase the overall pool. So we identified large number of transcripts from root as well as shoots, their expression profiling in various tissue systems. And then we compared with, we wanted to know why this vidanolide specifically in Vidania. So do they have similarities with the genome sequences of other solanaceous plants? So we compared with the capsicum, we compared with benthamiana, and we compared with capsicum, so that they can also be the suitable system for their overexpression. We studied two transcription factors, that is WDR transcription factor and Wuschel transcription factor. These two transcription factors, they were also, as you can see here, they were present in both the solanaceous plant, and they were found to be regulating vidanolide biosynthesis. So thus, using the transcriptomic resources, now we had huge number of gene pool with us, many number of gene involved in the pathway, and total transcript. We created their interactome, and we also put this gene located on the entire metabolic pathway. And this was the global assignment, and this is the specialized product assignment. So most of these gene we had with us by this time. And now, so by these parameters, we characterize these species, and side by side, we develop the species which was having most of the desirable criteria. And one of the plant which came up high on this category, it was tested for its property of protection, neuroprotection, along with CDRI. And this molecule showed that the post 6-hour treatment, it was able to afford the neuroprotection. And this was one of the nationally released plant variety, which was highest in one of the vidanolide A presence. Then obviously when the molecule is present, extract is suitable, both are working. But to even further maximize the dose requirement and to minimize the composition of the molecule and minimize the extract, we prepared its liposomal delivery system, again with the help of CSR CDRI. And we can see that further the optimized this liposomal delivery system was efficient in affording the neuroprotection. So the now coming to the conclusion is that we, by doing a holistic study covering many area, now we have the plant. So the another part was that this plant has to go to the farmers, and especially we found the southern part of our country. In Telangana area, there were 5 districts which have adopted the cultivation of this plant, and you can see that most of the as per the climatic condition, the plant grew very well, and this crop gave better yield and better returns than the traditional crops that the farmers, in marginal farmers especially in that area, they were growing. So many of the farmers, they showed interest, and they distributed the seeds, and they started growing the variety which is having neuroprotective properties. So this is the variety at the trial stage, and this is having the major 3 molecule in its composition, and this is culminated because of the studies related to phytochemical, molecular, biochemical, in vitro, genomic, and pharmacological study. And this plant had high yield if we go in terms of the yield per acre or yield per hectare. This was quite good, almost double than to the wild and then to the varieties which were growing prior to that. And this was released from the Vigyan Bhavan. And in this, I acknowledge the help of all the funding agencies, so many scholars, postdoc, and collaborators from other laboratories, funding agencies, everyone who has traveled in this journey to have the assigned a molecule with a specific pharmacological action, and also to have a variety which is pharmacologically established. With these words, I again thank you all for your patience hearing.
Thank you, Professor Neelam. That was extensive work that you had carried out to, validate traditional medicine and the use of herbal plants. I have just one question. When we use Ayurvedic medicine, it is usually a consortium of various herbs. And also, you have either the alcoholic extract or the aqueous extract that is taken as medicine. And maybe some of them are metabolized to an active molecule once you consume them. So in such cases, how will you validate?
So first, there are 2 things you said. Many of the drugs, they are either used in water form or they are used in ethanol. Actually, what you are saying is the indirect way of saying the use of specific molecules. Actually, the way you put, if they are given in the water, if they are given in the boiled water, if their kind of decoction is made, or if they are extracted in ethanol, if they are extracted in other polar substrate, that's what controls their composition. And see, our forefathers, they were knowing about it. They were knowing about it, and that's the way many of the preparations, they are prepared in given with the ghee, many are given with the honey. So that is what is the they didn't have the column chromatography, they didn't have the flash chromatography and HPLC. So that was the precise way of having the particular component in a particular proportion. So initially, in fact, we did the experiment that you are mentioning. We did the same kind of experiments to see how much is this particular molecule is accumulating in a particular, so that is have a very actually large impact on such kind of drugs, because, and that is the reason why many people say that this medicine didn't work while it had this particular plant. So that is why it is very essential to know.
Very interesting how you have gone about, analyzing all these things. Session is open for questions. Any questions? You can post it in the chat box also.
We request the student participants to get ready with their presentation. Student presentation will start after the plenary lectures. The final speaker of this session is Dr. Umesh Shaligram, Executive Director, Serum Institute of India Private Limited. Dr. Umesh Shaligram leads the manufacturing of Covishield and Covovax COVID-19 vaccines. He also leads the research, development, and manufacturing of several products at Serum Institute. These products include recombinant vaccines, proteins, polysaccharides, and newer technologies like polysialization, biosimilars, and biobetters. He is also spearheading Serum Institute's strategies for tuberculosis eradication, and HIV prevention and therapy. We request the chairs to invite the speaker.
Dr. Umesh Shaligram, a talk which we have been eagerly waiting to hear. Over to you, sir.
Unmute झालंय. Hello, this is Umesh.
Yes, sir.
Can you
Please.
see me?
Yeah, your voice, you are audible, but we cannot see your slides. You
Can
are
you see
visible
me
now.
now? Yeah. Just give me a second. I'm just uploading my presentation. Once
Fine,
that's
sir.
done, then I'll be able to, start. Just give a second.
Yeah.
आम्ही तुमच्या ज्यांना
तुझ्या याच्यावरती गेले
लिंक
मी तुला प्रेझेंटेशन पाठवतो आम्ही एका सेकंद मी तू चालू कर मी तुला प्रेझेंटेशन पाठवतो
एक सेकंद
प्रेझेंटेशन पाठवतो
आणि प्रेझेंटेशन अपलोड पण करतो तुमच्या चंद्रवर नाही करू Yes, available. Let's open.
Can you see the share button, sir? Bottom.
Just a second.
Yes.
Any difficulty?
तुम्ही अपलोड करा
एक सेकंड
नाही कर तिकडनं ट्रॅक कर
तू हे कर ईमेल तुझ्या ईमेल आले माझ्या एक सेकंद इकडनं just give us a few seconds.
Sir, you're able to see the share button?
Yeah, and we are able to see there is some issue with the system actually. So we are just logging in from another system. Just give Dr. Shaligram a minute.
Yeah.
स्क्रीन ला ठेव की अपलोड करू
अपलोड करा होतं की नाही
Yes, sir.
Are you able to see the presentation?
Yes, visible.
Yeah. Okay, am I audible? It's you are able to listen. Okay.
Yes, you are.
Thank you.
Please go
Yeah.
ahead.
Yeah. So first, apologies for little challenges. You know, it's typical Murphy's law. What we say when you want things, always electronic things always gives a tough time to you on. So it's okay, no problem. And first of all, my sincerest thank to Dr. P.V. Mohan and his entire team for his kind invitation for such an elite SNT seminar between Indo-Japan collaborative efforts and then inviting me. I really sincerely want to thank him and all the organizers of this very big meeting and really sincerely thanks. And with that thank note, I just would like to continue my presentation. I hope I'll try to be brief. I will not try to bore because I'm not a great speaker. So don't worry, all the students in the class just enjoy you. I hope I'll be able to give a pretty good overview to you about evolving technologies in vaccine development, case studies on COVID-19 vaccine. So the next slide. It's not going for the next slide. Yeah. Yeah. So in Serum, Institute of India has played a key role in COVID-19. So I thought that before I get into the technologies and evolving technologies, basically I would like to give a bit of overview about Serum, what Serum has done. Serum in COVID has actually was developing a lot of products right from the beginning. So Covishield is one of them. Everybody knows. Covax is next product. So Covishield, we actually have a MA emergency authorization in 34 plus country. We have supplied this vaccine more than 100 countries, and our own country, we have supplied almost close to about 1.33 billion doses, and we have manufactured 1.6 billion doses at present. So we are having quite a bit of product in our hand. So it's really great that we could really make sure that our country is self-sufficient in vaccine need actually. So that is Serum's COVID portfolio. Covax is now approved by Indonesia and is in advanced stages of approval by several countries. We'll be supplying that product. It's a nanoparticleized adjuvanted product. Covishield is an adenoviral vector-based product. Cyvac is a unique nanoparticleized receptor binding domain display product. We are also manufacturing Sputnik Light for export purpose. We also develop equine antisera for therapeutic purpose, and we also have a recombinant BCG for immune boost actually. So there are several different products which we developed together. Key is that I would like, before I get into the more technological discussion, certainly, sincerest thank to our Honorable Prime Minister. The kind of a coordination complex is set in. We could really set a world example of vaccination, and entire world is now looking at Serum Institute of India as an example of vaccination. So nice coordination complex which worked out between ICMR, DBT, DCGI, and everybody, RCGM, every other agency which is involved, which I probably may miss taking some names, but obviously under the guidance of Professor K. Vijayaraghavan and Professor Vinod Paul and a lot of team members all across, they all came together and really worked day and night. And we could, as a country, could set in a very nice example of we can take the challenge. We could take the challenge of COVID-19 not only in terms of entire management but also vaccination has been superb example in the world. This would not have been possible without all government's effort. And I would like to specially mention Dr. Somani, our DCGI, day-night work, regulator work, day-night. Trust me, day-night they work to really get into a lot of details, approval process. So all of the system worked very nicely, and especially DCGI team, day-night they really worked very hard. So without which we could not have really done such a wonderful vaccination program. So sincere thanks to all involved, and therefore we could really work on COVID-19 portfolio and develop a lot of vaccine and supply globally a vaccine. Now I go to the next step of my presentation. So in a COVID-19 uniquely has led to a development of a new technology actually. And conventionally, if you look, COVID-19 has not only taught a unique, faster way of licensing the vaccine, but also it has taught historically vaccines takes 10 to 15 years to license because you have to do something called as a clinical efficacy trial. So you have to actually vaccinate people and wait for reduction in a disease burden or reduction or elimination of disease. And that's what people have done classically with vaccines. And it took time to then starting from manufacturing of vaccine, then it takes about 10, 15 years, 8 years, classic people have tried to develop some mumps vaccine in less than 4 years. So there are a lot of time challenges were there. So technological challenges as well as clinical development related challenges. So these challenges COVID-19 really surpassed and came out with a pretty nice kind of a newer technologies for manufacturing and development. And these newer technologies, if you look at it, they can be easily categorized into 4 major categories. Viral vectors, actually, like a human adenovector or chimp vectors, which Covishield is one of the example. mRNA technologies, which was unique actually, and Moderna and Pfizer manufactured the product, and they came out with these mRNA technologies, which could be manufactured way faster in high amount and quickly made available. Novel adjuvants also came into picture, like IMDG, which Bharat Biotech has developed. We are also having Matrix M adjuvant, which is also developed. So these are new and newer manufacturing technologies. They suddenly came up in the entire COVID-19 area actually. And this is what I will just give a quick overview about them. So what has happened is in a viral vectors, the big pluses of the viral vectors is this kind of a well-known and exploited field for gene therapies and vaccines, and it was in works since 1970s in Oxford and several areas, and initially focused mostly on Ad5 vectors and gene therapy. A lot of safety work was done already, and when COVID-19 hit, there were already a lot of background work which a lot of people have done in the field of HIV, malaria, RSV, Zika. In fact, some of the SARS-CoV-1, in that one also advectors were used actually, and people found a lot of advantages of using advectors. So this is certainly the technologies which came up, easy to manufacture and easy to deploy actually. So this was done, and you can see Covishield, Vaxzevria, Serum, AstraZeneca, Oxford relation licensed viral vector vaccines are there. Then J&J has Ad26. Then Sputnik V as well as Sputnik Light are coming up. So a lot of advector based COVID-19 vaccines are coming up actually. So that's one technology which has come up. There are several advantages of advectors. Ease of method of manufacturing is certainly a key advantage. But still it needs a lot of fine level understanding and testing and development. And now several billions of people have got this technology. So it's very well proven technology. So in a future pandemic also viral vectors can be easy, cut paste technology, and you can use it. That's then mRNA technology, which is also was tested long back. People had a lot of idea about mRNA technology, but mRNA also has a lot of degradation, very difficult to handle. So unique method of manufacturing, which evolved out over the period of time using disposable, helped in mRNA manufacturing quickly. And this was developed for several cancer and flu-like diseases and others. But however, in COVID-19, the good part which came out is lipid nanoparticle delivery. So mRNA can be entrapped into lipid nanoparticle and then it will be injected. And that's why the biodistribution is good. Then mRNA goes into a lot of cells. It expresses the product of it on the cellular surface, and then cell-mediated response goes up, and that also activates the antibody responses, and both put together really gives a very high antibody responses. So it's very clear that Pfizer, BioNTech, and Moderna has done a large part of a developed world vaccine. Yet developing world is a lot more to be vaccinated, and we are coming up with our lot of products to supply developing world also. So large part of developed world has got vaccinated, and obviously developing world also they are supplying the vaccine. So it's getting it's now proven a newer way of use of mRNA and technology. There are several advantages, certainly non-infectious, non-integrating platform, considered to be more safe, various modifications in mRNA. It's quick. You can, if you have a variant, you can quickly translate into mRNA and make this product available. Certainly, there are cold chain related challenges, for developing world, these are major challenges. Injection site reactions are there. And there are some limitations, but obviously these limitations gets overcome as technology evolves out and it's get developed. Another field which got developed very uniquely was novel adjuvants actually. So there was need of novel adjuvants, and one of the adjuvant which we are ourselves are working on, matrixamin in the field of malaria, is also used pretty heavily. So that adjuvant really proves to be very unique in our Novavax collaborative product called as the Novavax. That's an adjuvant which is used. It is based on a bark of a tree and this unique way of method of manufacturing. So that is one. We are also working on squalene water emulsion based adjuvant called as SWE, which is used in our product called as SAVAC. And then also there is a CPG, which I believe BE is using this. And IMDG is well proven adjuvant now, as Bharat Biotech has already launched Covaxin with IMDG adjuvant. So the new adjuvants technologies came up in COVID-19 and people started using these new adjuvants pretty uniquely and easily actually. Also what COVID-19 landed up is in a technologies completely changing the technologies of method of manufacturing. Mostly classically people used to have a classical way of manufacturing using stainless steel bioreactors and CIP, SIPs, and everybody started realizing these technologies have a lot of limitations. So use of a lot of disposables and single use, which has an excellent turnover time, was clearly got validated and people are using. It's not like people were not using single use disposable, but they have started using enormously at a huge scale disposable. Like we ourselves have a 16 to 4,000 liter fermenters which are only disposable, and now we have created a 16 to 6,000 liter disposable fermentation suite actually. So you just imagine previously thinking about anything about 500 or 200 liter about disposable was a big challenge, but we have created this. These are unique example in the world. Probably nobody has this, but we have created this, and that has really helped in quickly turning around from one batch to another batch and making the vaccine available quickly. So certainly the overview, if you look, COVID-19 and emerging technologies. So there's a field of adjuvant which got developed. This field of manufacturing technologies and scale up got developed, and also the field of a newer viral vector and mRNA technologies got really developed actually in this entire COVID-19. So certainly as a Serum, we are extremely happy. We are working on all the technologies at present today in Serum. As a country, we have really done very nice in expanding every other technologies, because there's every other manufacturer is working on every other technologies in India, which is really great. And this certainly helps in really consolidating and creating a technical skill, which really helps in our Prime Minister's vision of Atmanirbhar Bharat actually. So with that, probably I would like to stop over here, and I would like to thank once again all the organizers, and especially Dr. P.V. Mohan for inviting me for this delight seminar. And I hope I could give you kind of overview you have expected. I know it is short presentation, but I thought that probably this would kind of ignite young generation to ask some question. I'm available for questions, and we can go through question answer session.
Ma'am, please unmute. Ma'am.
Thank you, Dr. Umesh. We were eagerly awaiting your talk, and you have stopped at a juncture where you are, stimulating everybody to think. That's good. The session is open for questions. Anybody wants to raise a question, please do so, or please post it in the chat box.
Attendees, please, any questions?
I have one question. When you are manufacturing these various types of vaccines, how was the facility tuned for that?
No, truly speaking, a lot of our facilities were not ready. It is all basically in a short time. It's certainly Mr. Radhar Poonawala's visionary ideas and Dr. Poonawala's visionary efforts, and all of us coming together, teaming up, which is what I gave an example. Even our Honorable Prime Minister also did a very nice coordination complex of teaming up. So teaming up happened, and we were having only shells ready. So in record time, like 3 months, 4 months time, we could really establish the facilities of 6 into 4,000 liter scale. So if you would have come last July, you would have seen only shells, and July, August, September, facility was ready. And October, we started manufacturing actually. October, November, we started manufacturing. So that was like speed and coming together continuously. And also having decision maker which are stick to the ground was really important for us. So that helped us a lot very quickly in moving things faster compared to others. In fact, I'm very proud as an Indian, as a team, we all have done wonderful. But what is important is that what we are making around 250 to 300 million doses a month, which is a unique world record. Nobody has been able to do this actually. And that is certainly a good, as an Indian, we all should be proud about it. We could do that actually, no doubt about it. And that was certainly because of team efforts, which was possible.
Dr. Shaligram, I believe you are part of the Gavi program also. So your COVID vaccine has been reaching the countries in the Gavi program.
Yeah. Our Covishield and Covax is already reaching Gavi. We already supplied quite a bit of Covax facilities, called as Covax facility, under Covax facility, and we have a big tie-up with them for giving a Covovax, which is our Novavax collaboration effort. We will be supplying them almost more than 1 billion doses to them.
Yeah, Dr. Umesh, this is Mohanan.
Yes,
I'm really happy while we were talking about this vaccine, and I was in the committee for evaluating the safety and toxicity of all your products. Okay, and the first time we got all the documents, we were surprised to see all these documents. We don't know how to approach and all. But finally, in the second stage, okay, we are happy. And one of the other member, Dr. Neelam, is also there. She was also in the RCGM committee. We are looking all these things. Thank you, and thank you for your support to the humankind. Okay, thank you.
So kind of you, sir. Thank you. So kind of you. Thank you for all your support, and all of us together done wonderful. So great.
And also you supported for me for a generous supply of how many doses to one hospital.
Yeah, one. Yeah.
Yeah.
Thank you.
Thank you.
Yeah,
Thank you very much.
There is a question posted in the chat box. What you think about booster doses? Is the 2 doses sufficient? I would like to know the controversies about efficacy of vaccines from Prajita to all panelists.
Okay. So it's a well-known fact that vaccines after 2 doses, always there's a immunity wanes off. So when immunity wanes off, so the disease actually, when you have an early infection, body takes a lot of time to mount back that level of antibody titer which is needed even for protecting an early entry of a virus. So there is certain level of antibody titer you have to maintain so that even you are not getting infected. Then in case those titer drops down, then there's a chance that you will get infected. But the body's, if there is a virus has generated a very good T cell response, a memory response, then body will mount back a right memory response, and then it will take time. In that situation, lot of time what happens when you are having a comorbidity or old age or other challenges, if antibody titers are not high enough lot of time, then in that case, there can be a breakthrough infection. And if virus has the ability to quickly mutate, change, then it can overcome a barrier which was created by the vaccine. So in such a way, in that case, instead of depending on mounting of a response, which will take time, if you boost yourself, your immunity, then you have a high enough titer of antibodies, and then you can have a protection from even early infection or a mild disease also. That's the reason why booster is generally recommended. But virus is RNA, it is going to mutate, it will have its own ability to change. So one has to take care of all other precautions as one would take, along with taking a booster immunity, so that you are well protected. No doubt about it. That's the view. And there was another question. What was the additional point? The controversy
About Omicron.
Omicron. So, you can literally see, very interesting. In India, in Pune itself, there are about 10 people they found out Omicron. And out of those 10, most of them were Covishield vaccinated. And you can see that they are having a very mild or almost no symptoms actually. Most of them are doing health-wise good actually. It's already coming into the public domain information. So certainly vaccine is protecting. See, I'll tell you how vaccine protects. So if you're vaccinated and if you're boosted, then your immune systems are trained. Though there will be variants which will come a lot of time. And it's better to have variant this one vaccine, but that's impossible to create so in such short duration. But your body has something called as an affinity maturation. So your body learns itself. So immune system has the ability to go through a learning curve called as affinity maturation. So even if your body is trained for seeing an antigen, you are seropositive, then if there is a change in the antigen, then body has quick ability called as through affinity maturation, changing itself and learning how to fight against the right antigen so that it protects itself. So if you're primed rightly, then and if you have a memory, there's a high chance that you will remain protected because body has the ability to learn itself also to fight against variant. That's the whole thing. So there is a chance if you are vaccinated, if you are having a boosted, because if there's a comorbidity, then there's a high chance that you will remain protected. But there's a chance. I'm saying there's a chance because one cannot give any assurance about this because these are all subject to a lot of studies, detailing. There can be some cases where you will have a full breakthrough infection, but these are individual to individual immunity related challenges. That's all.
So, can you take the third booster?
Yeah, I mean, basically, it's recommended. Western world has gone with a booster. Most of them have gone with a booster. Luckily, good part is because largely Indian population is vaccinated with Covishield. T cell response compared to mRNA vaccines are better in the advector-based vaccines. So that's what people are seeing, that there is a better protection in in advector-based vaccine. So let's see what happens. But these are all developing science. I don't want to comment anything firm. Let's see how things evolve.
Yeah, thank you, Dr. Umesh. Thank you.
Thank you, sir, for very informative talk on this hot topic. And also congratulations for doing a great job for India. But I have just a small question. Even though the efficacy of this vaccine is proven, but in rare cases, some side effects are reported. So could you please comment on that?
So each new technology you bring in, you will always have some side effects. So that's what they say, risk to benefit ratio actually. So there is the government agencies has to take a call between risk to benefit ratio. So every technology will have its own challenges, will have its own side effects because it's expected, because it's finally new technologies, newer version. mRNAs have different challenges, advectors will have different challenges. Classical vaccine technologies have a lower level of challenges, but when they were getting developed, that time also there were a lot of severe challenges. Now if you take even pertussis based vaccine in childhood, you take that vaccine, BCG vaccine you take, you land up with site of injection challenges, all those things comes up. But finally it is question of a risk benefit ratio. That's
Thank you, sir.
Thank you. I believe the session is now coming to an end.
Thank you, sir, for a very informative lecture. We thank Dr. Gautam Panda and Dr. Maya Nandakumar for spending your valuable time to chair the session. Thank you, ma'am. Thank you, sir.
Thank you. Thank you. Thank you.
And now we come to the end of the lectures for the Nobel Laureate Science and Technology Seminar Series, and we will move on to the students' presentation, jointly chaired by Dr. Darshak Trivedi, Department of Chemistry, National Institute of Technology, Karnataka, Editor, Indian JSPS Alumni Association, and Dr. Srinivasalu Tadisetti, Kakatiya University, Warangal, Editor, Indian JSPS Alumni Association. We request student participants to make sure that your video is on while your presentation.
Dr. Shakti Kumar, can you take over the session, please? Dr. Shakti Kumar.
Yeah, hello.
Yeah, can
Hi.
you take over the session,
Yes,
please?
yes, of course. Darshak was there. I was checking where was Darshak. Darshak was there. Yeah, Darshak.
I have joined, sir.
Okay, thank you. Okay, thank you. Over
Darshak
to
was
you.
there. That's what I was checking. Yeah, Darshak. Yeah, I will also be along with you.
Yeah, sure. Then you can start, sir.
No, Darshak is your job, your duty. You please do that.
Okay. So, a very good afternoon to one and all present here, and we are at the last technical session of 2 days Nobel Laureates Science and Technology Seminar, which is the 11th in the series of jointly organized India-Japan Science and Technology Seminar. And it is a second session for the students' presentation. And there are total 17 presenters in the second session of posters presentation. I will be calling name of each one of you one by one, and there is 2 minutes allotted to each of you for your presentation. And I request all of you to please adhere to the timings. I hope all of you are ready. Shall we start? So the first presenter is Mahalakshmi Shubhaya.
A very good morning to one and all present here. I'm Mahalakshmi. Under the guidance of Dr. S. Naharajan, my topic is an nanostructured copper oxide-based high-performance supercapacitor. Ever-increasing global energy concern is a major topic today's world. That the supercapacitor is one of the received great attention today. It's a high stability, high cycle stability, and charging, ultra charging and discharging. Whereas the copper oxide is a phytic semiconducting material widely used in the field of photocatalyst, photovoltaics, and energy storage application due to its high chemical stability, notable electrochemical performance. In this context, it has been prepared using a co-precipitation technique. The electrochemical measurement has been measured using a VSP 300 instrument using a 3-electrode setup in a 3 molarity of KOH solution as an electrolyte. Cyclic voltammetry, charge, GCPS and PES and cyclic stability study has been undergone to study the electrode-electrolyte interfacial reactions. From CD and CV, it is obvious that material shows a pseudo-capacitive nature, and then material undergoes a Faraday reaction at the surface with determined electrochemical AA charging. And therefore, the material achieving a max stress capacity of 300 farad per gram at a current density of 1 ampere per gram. And also the material achieved a 65% of cycle stability over the 10,000 cycles with a constant achieved from the active material utilization with internal resistance of the material, which is a dependent on electrolytic ion in an electrolyte. This effectively constructs the AA charging and undergoes a direct reaction at the surface. The gives in the application of hybrid supercapacitor with AA material and solar capacity material for the proficient design of an advanced energy storage system. Thank you.
Yeah, thank you, Mahalakshmi. The next presenter is Munmun Baglari.
Okay, greetings to one and all. Myself, Munmun Baglari. The title of this presentation is Clostridioides difficile infection, interaction with the gut epithelial cells. So to begin with, Clostridioides difficile is an opportunistic pathogen and it causes diarrhea as a repercussion of a prolonged antibiotic therapy. Upon broad spectrum antibiotic treatment, the normal microbiota of the intestine is disrupted, and this allows Clostridioides difficile to colonize and affect the host. So due to its increasing mortality and morbidity rate worldwide, this infection has been regarded as a widespread issue. Some of the quick facts, like the risk factor can be age, underlying diseases, impaired immunity, and prolonged hospitalization. Symptoms can be watery diarrhea, fever, and weight loss. And one of the ways to prevent this infection is by indiscriminate use of antibiotics. So as I have mentioned, upon broad spectrum antibiotic therapy, the colonic microbiota is altered and C. difficile can colonize the gut. And this bacteria, as a main virulence factors, it produces toxin, which are the toxin A and B, and that leads to the diarrhea and colitis. So when these toxins gets internalized into the cytosol of the epithelial cells, as a result of the action of the toxin, the mitochondrial function is disrupted, leading to the ROS production, and that ultimately leads to the breakdown of the tight junction. So here, the role of the immune system as well as the role of the gut microbiota as a probiotics in controlling this disease needs to be investigated and studied well. Here, the immune response are being analyzed at both the genomic and the proteomic level. The cytokines that are being analyzed are interleukin 8, interleukin 17, 22, and 1 beta. RT-PCR and ELISA are the techniques which are involved for the analysis. Coming to the conclusion and future prospect, the role of probiotics in influencing this inflammatory immune response will be a step forward in controlling the disease. Thank you.
Yeah, thank you, Ms. Munmun. The next presenter is Amruta DS.
Good afternoon, everybody. The topic of my presentation is active principle identification of active
Amruta, your voice is not clear.
Am I audible?
Yeah, now it is fine. Please proceed.
Okay, the topic of my presentation is active principle identification of methanolic extracts of Syzygium cumini and Erva lanata. The Syzygium cumini and Erva lanata are two plants that are used traditionally for many years in wound healing, and they have wound healing activity. The major objective of my study is to scientifically validate the effect of Syzygium cumini and Erva lanata through the molecular mechanisms of wound healing. Here, the preliminary studies of these extracts are done. The cold methanolic extracts of Syzygium cumini and Erva lanata were prepared, and principal active compound detection and antioxidant activity was studied by DPPH assay, and also GCMS analysis were done. The cytotoxicity evaluation was done by MTT assay. The results shown that the qualitative phytochemical analysis results shown that alkaloids, flavonoids, saponins, tannins, and glycosides were present in Syzygium cumini, and flavonoids and saponins were present in Erva lanata. The antioxidant activity shows increasing order of antioxidant activity with increasing concentration, and GCMS analysis showed various volatile compounds which have the capability of antioxidant and anti-inflammatory activity. The cytotoxicity assay showed less cytotoxic effect or non-cytotoxic effect of these extract with Rho 264.7 cell lines, and The future study will be
Your time is up. Please
concentrated
continue.
on the molecular mechanism of wound healing by these extracts. Thank you.
Thank you, Amruta. The next presenter is Manoranjana Ponraj. Manoranjana Ponraj, please.
Good afternoon. My topic is on design and development of protected and unprotected hydrazine decorated 2D nanosheets. So here we have taken hydrazine sheets along with GO. In that intercalation distance we have studied. First, GO is treated with BOC and tertiary butyl hydrazine carboxylate. In that time, the interspace distance was increased to 1.21 nanometer. After BOC is used to protect hydrazine in that compound, when BOC is protecting hydrazine, after dilute HCl is added, in GO hydrazine, the distance is again decreased from 1.21 nanometer to 0.94 nanometer. This is confirmed using XRD, FTIR, and NMR studies, and DFT calculations also used to confirm these studies. So in C13 NMR, we have 3 peaks for CH2, CH, and for tertiary butyl. But whereas in GO hydrazine case, we have only 2 peaks, that is for CH2 and CH. So this confirms that hydrazine moieties were successfully intercalated on GO surface. And this GO nanosheets enhance the hydrophilicity of functionalized materials, and this it can be used as a suitable material for energy storage. Thank you.
Thanks, Ranjana. The next presenter is Manisha Mashelkar.
Hello, sir. Am I audible?
Yes, please proceed.
Hello, everyone. I am Manisha Malsekar, PhD student from Demping College, Goa, giving presentation on the topic fissile thermal decomposition of zinc succinate precursor yielding nano-sized zinc oxide material. The method of synthesis profoundly influences the structure and morphology of oxide materials. The main objective of the research work was to prepare nanoparticles of zinc oxide and serum doped zinc oxide with light absorbing capacity. So these oxide materials were prepared. Sir, the poster is visible, sir. Hello, sir. The main objective of the research work. Okay. The main objective of the research work was to prepare nanoparticles of zinc oxide and cerium doped zinc oxide with visible light absorbing capacity. So these oxide materials were prepared by precursor method followed by thermal decomposition to form final oxide. XRD confirmed the phase formation of cerium doped zinc oxide as hexagonal woodsite. SEM and TEM images were captured, which reveals that all oxide materials are in the range 20 to 100 nanometer. And high resolution TM also showed crystal planes in it. UV DRS were also recorded, and the band gap of zinc oxide and cerium doped zinc oxide were calculated. The band gap of zinc oxide, that is 3.263 electron volt, was reduced to 3.131 electron volt by doping cerium. So it was reduced by 6%.
Yeah, your time is up. Please conclude.
Zinc oxide and cerium oxide materials were prepared by simple precursor method. Because of reduction in band gap, the materials have a tendency to absorb in visible region. So zinc oxide is used as a biosensor and photodetector. Thank you.
Thank you, Manisha. The next speaker is Nivedita K.
Good afternoon, everyone. Myself, Nivedita K, PhD student under the guidance of Dr. Annie Abraham, University of Kerala. I am here to present the topic stem cell seeder decellularized tendon tissue to mimic the tendon ECM, a promising scaffold to mend injured tendon. Tendon are the fibrous connective tissue that attach muscle to bone. Achilles tendon is the strongest tendon in our body. Despite its strength, it is more vulnerable to injuries too. WHO has reported 33 million musculoskeletal injuries, out of which 50% involving tendon and ligament injuries. The currently available treatment for tendon injuries using surgical replacement with expensive synthetic polymeric graft. However, there are chances of re-injury of the repaired tendon, and here comes the need of an alternate treatment method using biological scaffold derived from decellularized tissue, which mimic the normal tendon. Coming to the methodology, here we are trying to fabricate a new advanced tissue engineered medical product. Here, decellulation of tendon using chemical combination method, and stem cells were seeded onto CT scaffolds. After in vitro study followed by in vivo study, it will be a promising scaffold for tendon tissue regeneration. Coming to the results, the decellularized tendon were characterized by H&D staining, the nucleus staining, and for its efficiency of decellularization. After that, isolation of stem cells were done, and they are characterized by flow cytometry. And the isolated cells were seeded onto this decellularized tendon scaffold in vitro and characterized by live dead assay, SEM analysis, MTT assay, and these results proved that cells are attached on the surface of decellularized tendon scaffold, which prove its cytocompatibility and which are non-cytotoxic, and we can conclude that we are successfully combined the biomimetic properties of the surrogate extracellular matrix of decellularized tendon, which along with the regenerative potential of stem cell, along with will be a promising solution to treat tendon injuries, which is an unmet clinical need in orthopedic reconstructive surgeries. Thank you.
Thank you, Nivedita. The next speaker is Swati N.
Am I audible, sir?
Yeah, be little louder, Swati.
Okay, sir. Excellencies and distinguished dignitaries. My name is Swati, and I'm a third-year BMS student from Vaidyaratnam PS Warrior Ayurvedic College, Kottakkal. The title for this presentation is a retrospective case series analysis on Ayurveda treatment protocol in children with spastic quadriplegia. Spasticity management in conventional practice is technically skill-oriented, expensive, and requires long-term interventions. Ayurveda claims cost-effective, home-based, and effective management not only in the spasticity, but also in every sphere of development in children with cerebral palsy. This study is a scientific observation regarding one of such claims from an academic institution, which is a tertiary referral center for Ayurveda pediatrics. Coming to the methodology, this study was conducted among 40 children falling in the age group of 4 to 6 years admitted in Vaidyaratnam PS Warrior Ayurveda College Hospital under two categories during the period 2008 to 2012. First group was administered exclusively with Ayurveda treatments, which is denoted here by group AY. Second group was administered with skilled physiotherapy in addition to Ayurveda treatments. Among these 40 children, 20 each are from the above two groups. The treatment methodology in the Ayurveda group is as follows. Children were provided with herbal extracts for internal administration and procedure-based therapies for a duration of 31 days at an interval of 6 months. TBT consisted of both Snehana and Vrukshana. These are two unique management strategies in Ayurveda, which contributes powerful contrast stimulus in multiple ways so as to produce high gravity of neuronal plasticity in a developing brain. Next, I would like to draw your attention to the results of this study. Both these groups show statistically significant results in terms of muscle tone and general motor assessment based on GMF, CSE, and all. The results in the first group were only Ayurvedic treatments were administered invites many innovative in discussions in the research perspective. This brings me to the end of my presentation. Thank you.
Thank you, Swati. The next presenter is MS Vijay Kumara.
We will move on to the next participant.
Yeah, the next participant is presenter is Aarti Ashok.
Hello. Good evening, everyone. Am I audible?
Yes, you are audible.
Okay.
Yeah, but yeah, this is not okay. This is Aarti's presentation. One
No.
back, madam.
Okay.
No.
Next one.
No.
No, this is not Aarti's presentation.
Yes, sir.
This is Aarti's presentation. Yes.
So am I
Aarti,
visible, sir?
yeah, your presentation is visible. You can switch on your camera. Please proceed.
Okay, sir. Good evening, everyone. I am Arati Ashok. The title of my presentation is Cellular Responses of L-cysteine-cap zinc oxide nanoparticles using conventional and organ-on-a-chip approach. The work was carried under the guidance of Dr. P.V. Mohanan. So in these recent years, the organ-on-a-chip-based microfluidic technology has been proposed as novel cell-based systems that can replace the traditional cell culture systems. So in this context, the main objective of the study is the development of a simple microfluidic lung-on-a-chip device to investigate the cellular uptake and toxicity of cysteine-coated zinc oxide nanoparticles. The cysteine-coated zinc oxide nanoparticles were synthesized and characterized using various techniques like DLS, TEM, FTIR, and XRD. The cellular responses of the nanoparticles, including the cell viability, the cytoskeletal integrity, mitochondrial membrane potential, lysosomal integrity, etc., were characterized in A549 cells. The cellular viability of the cysteine-coated zinc oxide nanoparticles exposed A549 cells in the microfluidic cell culture was also analyzed using calcium. Yes. Hello.
Yeah,
There is
please
an issue in
continue. You
your
have a 45 seconds.
Hello.
audio.
Audio. Am I audible now? Hello.
Yes, Arti, yes. Please
Am I
proceed.
audible?
Yes.
Okay.
So the highest concentration of 160 microgram per ml cysteine-coated zinc oxide nanoparticles was used to treat the cells inside the device, and it was inducing the death of the cells when we performed the in vitro assays. The cells cultured in the device without the flow of media showed an increased number of dead cells. Dynamic conditions exhibited a reduced LDH release and NO generation as compared with the static conditions. Similarly, the damage to lysosomes in the cysteine-coated zinc oxide nanoparticles exposed under dynamic conditions were minimal than the static condition. Therefore, it is necessary to consider the residence time of the nanoparticle, the local concentration of the nanoparticle, and flow conditions at the target cell type while predicting the toxicity associated with the nanoparticle exposure. Thus, the present study concludes the importance of considering organ-on-a-chip technology for conducting the toxicity assessment of nanoparticles for obtaining physiologically relevant results and thereby minimizing the need for animal models. Thank you.
Thank you, Arti. The next presenter is Geeta Gautam. Geeta Gautam.
Am I audible, sir?
Yeah, be a little louder. Please
Yes, you're
start.
audible.
Yeah.
Okay. Good afternoon, all the dignitaries and my fellow participants. Today I am here to present work based on the topic toxicity of 4-nonylphenol on behavior and hematological parameters of stinging catfish Atropneustes fossilis. In this study, we exposed catfish Atropneustes fossilis to a toxicant that is 4-nonylphenol belonging to the family of alkyl phenols to a sublethal concentration of 80 micrograms per liter for 21 days and studied its effect on various hematological parameters. What we found was there was significant decrease in all the blood parameters, that is total count of RBC, WBC, as well as the hemoglobin compared to the respective control. Later on, we prepared a blood smear slide of different fishes, experimental as well as control, and observed them under the microscope to assess their morphological features. And what we found that there were varying degree of alterations in the morphology of RBCs, such as sickle cell formation was there, fusion of various RBCs were found to lead to Rowley's appearance, swollen and vacuolated cells. Besides that, there was significant induction of micronucleus in the cytoplasm of RBC. All these observations ultimately indicated the hematoxic role of nonylphenol on this fish. Besides the detection of micronucleus somewhere clearly indicates the DNA damaging role of 4-NP also on the fishes, as which can lead to various physiological disturbances, ultimately leading to the decline in the catfish population. Although these parameters need to be further assessed and should be researched in because of the bioaccumulating role of NP, which is posing various problems, and being an estrogen mimicking compound, it also affects the reproductive activities, and it needs to be further assessed. Thank you very much.
Thank you, Geeta. Next presenter is Vinit Barwal. Vinit Barwal.
Please move on to the next presenter.
Next presenter is Rakesh Kumar.
Good afternoon.
Rakesh, please proceed.
Yes.
Yeah, good afternoon to all. My name is Rakesh. I am pursuing PhD from Bhabha Atomic Center, Mumbai. So my PhD topic is to predicting the CHF critical heat flux value under the oscillating flow conditions. So CHF critical heat flux also known as a dry out. So in boiling water reactor, CHF is known as a dry out. So what is the CHF? So energy generated by the nuclear fusion or fusion, it is removed by the coolant. So the maximum amount of energy which can be removed by the coolant is determined by the phenomena which is known as the CHF value, critical heat flux value. If we go beyond the CHF value, then what happens? The heat generated by the nuclear fusion cannot be removed by the coolant, and the temperature of the nuclear fuel load will be shoot up, and it is not a desirable condition for the of the reactor. So first, so we are like last 15 years, people are evaluating the value of the CHF at the constant flow condition. But in the new generation reactors, like reactors, we are India designing is advanced heavy water reactor. In that condition, the circulation is not happens because of the external pulse. It happens because of the natural circulation by use of the gravity. So in that case, in the natural circulation, so what happens? The flow is not an independent parameter. It's dependent on the flow itself also. So flow oscillations happens. So in the flow, what is the condition? What is the value of the CHF if flow oscillate with time? So in that case, we have the predicted the CHF value for the our new design of the HWR, and we found that in the maximum cases, the CHF value under the oscillating condition can be only 40% of CHF value under the steady state condition. So this is my conclusion. Thank you, sir.
Thank you, Mr. Rakesh. The next presenter is SP Chauhan. SP Chauhan.
Yeah, we will move to the next participant.
Okay, madam. So the next participant presenter is Amala Jose. Amala Jose. Amala Jose is present here.
Yes, sir.
Yeah,
Hello, sir.
please start your presentation. Over to you.
Hello, sir. Can you hear me?
Yes. Please start your presentation.
Okay, sir. A very good evening to all. I'm myself, Amla Jose, represent a startup company, LAUREATE Exploration India Private Limited. My topic is evaluation of phytochemical, antioxidant, anti-inflammatory, and antimicrobial study of Syngem selanicum. In our study, we have used Syngem selanicum plant leaf to evaluate the phytochemical, antioxidant, anti-inflammatory, and antimicrobial studies. In our study, we have find that certain Syngem selanicum leaf have certain phytochemicals, then antioxidant and anti-inflammatory and antimicrobial properties. Due to the antioxidant property, the Syngem selanicum leaf can be used for the can be used in the food industries as well as in the cosmetic industries for the preparation of can be used as an ingredient for the preparation of ointments, creams, etc. Due to the presence of anti-inflammatory property, the Syngem selanicum leaf can be leaf extract can be used as an ingredient for the preparation of ointments. This can be used as skin irritation ointments. Therefore, hence we can conclude that the Syngem selanicum can be used as an ingredient for the for commercial purpose in industries. In it have many applications. And thank you, sir.
Hello, Darshak.
The next we will move to the next presenter.
Yeah, please. Yeah,
A very
Darshak.
good evening to all of you. I am Amrita Ajit Kumar, representing in the behalf of Dr. Rajashree S. from the startup LAUREATE Exploration India Private Limited. My topic of presentation is nano-lipid emulsion as parental nutrition. Lipid emulsions are mostly used as parental nutrition products for critical care patients. The products available on the market are imported, expensive, and made up of polysaturated fatty acids like sunflower oil, soybean oil, with lots of disadvantages. Hence, our proposed product is made up of monounsaturated fatty acid like coconut oil, which is widely available in the state Kerala with a blend of antioxidant vitamin E. The data of our study showed promising results compared to the parent nutrition available in the market. Hence, our proposed product, if developed indigenously, it will be very cost effective and hello.
Yes, you are
You
audible.
are audible. Please proceed.
Can you hear me?
Yes, you are audible.
Okay, okay. Hence, our proposed product, if developed indigenously, it will be very cost effective and very useful for common man with no or less effect. Thank you.
Thank you, Amruta. The next presenter is Joseph Xavier. Joseph Xavier.
Yes, sir.
Yeah,
am I
over
audible,
to
sir?
you. Yes, very much. Please
Yes, sir.
proceed.
Okay, good afternoon all. Today I'm going to present the work that we did for the synthesis of gelatin nanoparticle using a microfluidic platform. So gelatin nanoparticle is a versatile class of nanoparticle which there are a lot of applications and a lot of the functional groups are there, so we can modify the gelatin nanoparticle. But conventional method of producing a nanoparticle of size greater than 100 nanometer. So the main aim of this work is to produce a nanoparticle size less than 100 nanometer. So for that we use a microfluidic flow focusing device for that. So for conjugation we also use carbon content rods, which is prepared by using microwave synthesis method. And for the microfluidic method of synthesis of gelatin nanoparticle, first we did a multi-physics software simulation to find out the flow which is required for the particle formation, and then we finalize the flow. Then we fabricated the device using a soft lithography method. And in the device there is a 2 inlet and 1 outlet. In the inlet one we are adding a gelatin together with a QD, and another inlet we are adding a organic phase which consists of acetone and polaxomer. So when the flow is simulating, when the flow is starting at the junction, the solubility change due to the solubility change, the nanoparticle is forming due to the nanoprecipitation, which is collected and further characterized using the physiochemical characterization we are done. So in that it is observed that we are able to get a nanoparticle size 25 nanometer around in amorphous state. And we also did the characterization such as the UV spectra, PL spectra, which demonstrated the characteristic peak of QD, and the fluorescence which is less, which was less compared to the raw QD. And we also did the cell cytotoxicity assays, which we found that the nanoparticle were quite biocompatible even with MTT and NRU assays, and as well as LDH leakage and the reactive nitrogen species generation. As the concentration is increasing, the site scattered was increased, which demonstrated the particle uptake. And also we performed some organelle toxicity assays such as for the lysosome and for the mitochondria, which demonstrated active mitochondria as well as active lysosome even at the higher concentration. So we conclude that the nanoparticle preparation method using microfluidic is a good alternative method, and it is a continuous process, so we can use for the conjugation purpose also. Thank you, sir.
Thank you, Joseph. The next and last participant for today's presentation is Veerabhadragu Patil.
Yeah. May I audible?
Yes, please proceed. Over to you.
Start. Yes. Good evening to everyone. I'm Veerabhadragu Patil. I'm doing PhD in University of Pardubice, Czech Republic. My topic of presentation is new insight in the energetic conglomerate structure of attractive nitramines. Usually, energetic materials experience the sensitive to external stimuli, they undergo detonation and they detonate, means like you can call explosion. To avoid that, you need to modify the surface of the material such a way that they can't decrease their energy without affecting their energy. We need to modify their sensitivity. To that, we used diammonium trinitrobenzene as a modifier. via co-agglomeration method. After co-agglomeration method, we found the crystals dried and we thoroughly characterized. We found that there is a molecular level by like Raman, IR, and PXRD methods. There is like slight shifting in the stretching vibrations. So concluded that there is alpha beta to delta modification in HMX and sigma to beta in CL20. So this is this method. The crystals are structure modified themselves to adjust the DATB structure such a way that they can register more stimuli, external stimuli. Later we crystal observed in the same image are very smooth surface and there is no hotspot to which can impact on the sensitivity. So we observed that impact sensitivity and the detonation parameters. There is no decrease in the detonation parameter. They are sustained, and also except CL20, remaining RDX and bicyclo and HMX. Especially in HMX, there is a huge increase in the impact sensitivity. It's very good for suitable DATB is shown very good suitability with HMX. Then we in molecular level how it is adjusting itself.
Please conclude.
Yeah. We studied intermolecular studies. So here with we conclude that DATB is going uniformly distribution around this traditional M's, HMX and RDX, CL20 and DATB. So this is also forming the co-crystals in the molecule, making them stable and sustainable to external stimuli. Thank you.
Excuse me, sir. One more presenter is there.
Yeah. So thank you, Mr. Patil. And last presenter is Mr. Satyendra Chauhan.
Yes.
So, sir,
Yes.
one more presenter
Good
is
evening.
there. Yeah.
Hello, good evening. My name is Satindra. I'm from BARC and I'm working. Hello. May I start, sir?
Yes, please.
Yeah, what is your good name?
Sir.
Satyendra.
I'm working on investigation on the
Satyendra Chauhan, right?
thermovibrational.
Yeah,
Yes, sir,
please
right.
start. Over to you. Yeah.
Okay, thank you. In the large pool, the desired seismic and thermal loads. So I start with the introduction. In the light of the recent Fukushima nuclear accident, the focus of industry shifted towards the understanding of thermal performance of the large pools under the seismic excitation. So under the seismic excitation, sloshing can occur in large pools, which may significantly impact the coolability of the passive heat removal system relying on the large pools in the nuclear reactors. So usually thermal stratification is observed in large pools, which reduce the thermal performance of the passive systems. So therefore, the investigation of passive pools under the combined thermal and vibration load need to be examined. There is one example for the gravity-driven water pool used in the ASWR. So for this, we have done multiple experiments with carrying the parameters excitation frequency and amplitude. So because the excitation frequency is important parameter that may affect the heat transfer characteristics in the pool as thermal stratification is established in the pool due to the weak convection current. This causes a reduction in the liquid participation in convection cycle. Thus, the liquid participation is a key part of the present study. So the participation of water reduces, increasing the natural frequency. So we have considered only first 3 reports and 4 amplitude we have taken. So we just mounted our experiment on the SEC table and then we excited. So we have found that there is two another important parameters, that the orientation of heat exchanger and the submersion depth. So but the result part, we have found that the heat accumulate near the free surface is observed when the heat is heater inclination approach towards the vertical, which leads to the thermostratification. But with sloshing, that accumulation of heat is diluted. And secondly, the heat transfer coefficient get affected for the lower amplitude of displacement, which were not observed for the horizontal heater mounted. For the conclusion part, the heat transfer in the pool with submerged heat exchanger get affected mostly during the first mode of excitation. However, it is highly dependent on the amplitude of displacement. And the second one, as the heater position move towards up, liquid below the heater exchanger remains stagnant and it advances on the onset of subcooled boiling in the pool. With excitation, HTC get affected. HT means heat transfer coefficient affected when heater position is in the convective zone, then the impulsive liquid zone. For the same, we have developed correlations to evaluate the heat transfer coefficient for the natural convection region and the nuclear boiling region. Thank you.
Thank you, Mr. Chauhan. So I think we are done with today's poster presentations. Thank you, madam. Over to you.
Thank you, Dr. Darshak Trivedi and Professor Shakti Kumar, for spending your valuable time for chairing this session. We will start the validatory function by 4:25 IST.
Afternoon, sir. Welcome, sir. Prasad Krishna, sir.
नमस्कार जी प्रोफेसर त्रिवेदी
Namaste, sir.
कैसे हो आप?
Namaste, sir. बस ठीक ठाक, sir.
Good to see you. Yeah.
Yeah, we will be starting, sir, in just another 2-3 minutes.
The small state of Kerala has contributed richly to the science and heritage of India. Thiruvananthapuram, the capital city of God's own country, Kerala, is the abode of Lord Sri Padmanabha, apparently the richest god in the world. Referred as the greenest city in India by Mahatma Gandhi, Tirunal is also fortunate to have high-quality research and educational institutions apart from its natural beauty and rich culture. At the heart of the city, Sree Chitra Institute stands tall in the prestigious Medical College campus.
In 1972, our last Maharaja Chitra Tirunal, that was his Shashtapurti, 60th birthday. So the royal family wanted to celebrate, and they decided to build a specialty hospital block in Medical College campus. Achyuta was our chief minister here. And he had already put this hospital building as a Chitra Medical Center, as it was called. So why not make it into specialties hospital for cardiology, neurology, for which we had hardly any facilities in Kerala. And this also helped us in the sense that the concept, medicine and technology, if you bring it together under one roof, you can produce very impressive results.
Sree Chitra is an autonomous institution of national importance, catering to students within India and abroad.
I'm Dr. Shafiq, foreign student from Maurice Institute. I had done my DM cardiology training in the institute 10 years back, and at present I am doing postdoctoral fellowship in cardiac electrophysiology. I have found the teaching to be of high standard, and that's why after 10 years I am again here in the institute undergoing training in EP under a world-class team. My training here has immensely benefited my country in terms of mortality reduction in acute coronary syndrome, setting high standards for healthcare in the country, and especially gaining trust in cardiology services in the country.
Hello everybody, I'm Dr. Gausia Mukhtar. I'm proud to belong to the beautiful Valley of Kashmir, and I have come here to do my course in DM cardiology. And always I wanted to be here because it was a central institute, and I must say after coming here, it has been a great experience overall. And there was great learning. I have learned a lot from the faculty here, from the excellent faculty of Chitra, and I'm actually thankful that they provided me an opportunity to be here. It has been a great experience, especially we get to see many patients of congenital heart disease, and I'm actually learning a lot from here, and I hope that it keeps getting better. Thanks.
Sree Chitra Tirunal Institute for Medical Science and Technology consists of the hospital wing, the biomedical technology wing, and the Public Health Division. The hospital is a tertiary care center for catering diseases of the heart, the vascular system, thoracic, and the neurological system. Along with the patient care, where we have got a 250-bedded hospital, we also conduct super specialty courses in all the above mentioned specialties. We have got the DM courses for the medical science department like cardiology and neurology, and MCH courses for the surgical specialties like cardiac surgery and neurosurgery. Along with this, we have got the DM courses in the Department of Anesthesia and the Department of Imaging Science and Intervention Radiology. Also, we admit PhD students in the Cellular and Molecular Cardiology Department and Pathology Department and Biochemistry Department. We also have an MD course in Transfusion Medicine in the Blood Bank Technology. This year, because of the COVID pandemic, though every year we were having the entrance exam conducted in the hospital, this year it was an all India virtual entrance exam, and we found it much easier to get candidates from all over India. Because of the COVID pandemic, the number of cases that are coming for our primary care have come down drastically, but even then, considering other diseases also, along with COVID, that is prevalent in the community, we basically operate as a non-COVID hospital catering to diseases of the heart and the brain.
The biomedical technology wing of Sree Chitra.
A very good evening and a warm welcome to all of you for the valedictory function. We had the opportunity to listen to eminent speakers for 2 days, and we sincerely hope you all had a fruitful experience. With great respect, we invite Dr. Kailash Manda, Scientist, Institute of Nuclear Medicine and Allied Sciences, New Delhi, Vice Chairman, Indian JSPS Alumni Association, to deliver the welcome address.
Thank you. Namaskar. Am I audible?
Yes, sir.
Good evening to all. I am extremely honored to have given the opportunity to welcome on this occasion of the 11th Indian JSPS Alumni Seminar. I especially express my gratitude to session delegates of this special valedictory session, especially Professor Nair, President, Kerala Academy of Science, Professor Krishna, NIT Calicut, Kiyoshi-san of MEXT, Nishikawa-san from the JST, Dr. Ujjwala, Senior Scientist from the DST, Professor Annie John, Emeritus Scientist, University of Kerala. It is more than a decade when our alumni club had started a journey of the scientific collaboration between India and Japan. During this course, we have developed a strong, creative, and productive scientific network between two countries. A large number of collaborative projects were successful, which includes many TOTs, MOUs, student excellence program, dual degree program, and a large list of the outcomes which resulted out of this scientific collaboration and of this club. And all this achievement is because of the people and organizations who believe in us, especially the JSPS, but also MEXT, JST, Ministry of Foreign Affairs, embassies. So thanks to all for the contribution and trust in the Indian JSPS Alumni Association. Again, I express my gratitude and hearty
Thank you, sir. With immense pleasure, we invite Professor G.M. Nair, President, Kerala Academy of Sciences and the Dean of School of Biological Science, Central University of Kerala, to deliver the presidential address.
Thank you. Thank you. Good evening. It has been my great pleasure to be part of this major 11th India-Japan Science and Technology Seminar, the Nobel Laureate S&T Seminar Series, commemorating the Azadi Ka Amrit Mahotsav, 70th anniversary of Indo-Japan, establishment of diplomatic relations. I am an academician representing Kerala Academy of Sciences, a professional organization where we have people across the disciplines, science, technology, medicine, bureaucrats, all those people assembled together to make a forum where we take science to the masses. When I say that we take science to the masses, the idea is to mainly inculcate scientific temper among the students of the state of Kerala. We have stalwarts as honorary members, and we have 65 members in the academy and about 700 life members. I was extremely happy to be a part of this, especially to host these 10 schools. We have chosen 10 schools across the state of Kerala, wherein the students were exposed to a Nobel Laureate lecture, the lecture by Professor Tasuka Ponjo, as well as a special address by Professor Vijay Raghavan, the scientific advisor to the Prime Minister of India, to the government of India. I got the feedback from the schools. They say that they were immensely happy to listen to the Nobel Laureate. It's a lifetime chance for them to see and listen to a person, a stalwart like a Nobel Laureate. And this lecture was well received, and we have praises from all over the state that we have been instrumental in making them see this great personality and listen to him. I was also happy to listen to the almost 17 plenary lectures after this. Each one, I should say, they're all treasures, especially for academicians and students on diverse areas. And I'm sure that everyone who have listened to the people would be extremely happy to have enriched knowledge on different aspects of science. I'm also happy that my friend, Professor Mohanan, has been instrumental in managing this, in organizing this, and he has done it meticulously well. And I'm sure that everyone is happy about this conference. I have been representing Kerala Academy of Sciences for the past 3 years, and we have taken science to the students, inculcate scientific temper among them, and especially as we understand that the children will have to be taken into science so that we will have great scientists as we had in the past. And unless the science progresses, the country also does not progress. So we realize the Nehruvian vision, and we try to inculcate his temper, his ideas, and take it to the masses. We also have several programs being conducted, like webinars, offline seminars. All these we conduct periodically and take the science to the international forum, especially Kerala science to the international forum, as well as to the masses, fully realizing the scientific social responsibility of a professional organization, for an academics, as well as for the university. I really feel happy for being a part of this program, and I really congratulate all those who have been associated, who have listened to the people with an enriched knowledge to go back. Valedictory session, you're not supposed to speak much, because, and it's only 5 minutes, so I just restrict my talk over here and wish that next seminar will be in India. I was told that it's in Hyderabad. Probably that also would be something fruitful like what we have. Thank you very much, and wish you all the best. Thank you.
Thank you, sir. We are truly delighted to welcome Professor Prasad Krishna, Director, National Institute of Technology, Calicut, to deliver the valedictory address. Professor Krishna has more than 37 years of professional experience in metal casting, automobile manufacturing, precision machine, tool design and development, and space research. He had served prestigious organizations including DRDO Bangalore, VSSC Trivandrum, National University of Singapore, All India Council for Technical Education, and National Board of Accreditation. We welcome you, sir.
Thank you. Namaste and good evening to all. Am I audible?
Yes, sir.
Okay, fine. It is a great honor for me to be part of this valedictory sessions when we are celebrating the 75 years of India's independence, along with 70 years of cordial relations with India and Japan. My association with Japanese products and design goes back to my early days when I was the design engineer in one of the machine tool company owned by the government of India. And I was really impressed. Whenever we think of precision machine tools, in those days, I'm talking about in the early 80s, we could not find any other company other than the Japanese companies like Morisiki, the Toyota Machining Center, and when you talk about the Mitutoyo, in any of those areas, whenever we talk of the high precision machines in products, we have to look for Japanese. And we all got, as designers, got inspired by the Japanese engineers and the way they design and develop these products. I was told in the prototype meeting, when we and some of the Westerners come with lengthy reports and nice presentation, Japanese engineers used to come with the actual prototype. They come with the actual prototype, no just lectures or fancy presentation. So we have to learn a lot from this dedication and the quality consciousness of the Japanese. It is very surprising that the basic lessons of quality management and even the quality circle concepts we all introduced in those days in the early 80s in Indian companies because we got it from the Japanese. And it is also very surprising that Japanese got it from none other than Edwards Deming, who just, he was thrown out of from the American corporates, and then he was welcomed by the Japanese, and all the total quality management, the Deming's 14 points, fortunately, Japanese took seriousness about that, and the whole spectrum of the quality circle, quality function department, entire gamut of total quality management, just in time, everything we owe to the Japanese. So that what the beauty of this country is, they always find the something in every opportunity, in spite of their difficulties during the World War. They find it as an opportunity to come out of all these adversities and become the world leader in precision products. Later on, of course, I had an opportunity when I was in Delhi to visit, when I was in charge of the accreditation board, National Board of Accreditation. I had the opportunity to visit Japan twice, once to attend Japan wanted to constitute a Asian accreditation board. And we attended that meeting. It was organized by JABI, the Japanese Accreditation Board for Engineering Education. So this meeting was in Tokyo, and then this idea was mooted. When there is Washington Accord with the Western nations, primarily the Western countries, it started, with the Commonwealth and the US, the Washington Accord, which is a mutual recognition agreement with the accreditation bodies, primarily from the Western countries, the developed nations. Japan came forward to have an Asian accreditation board. And that was, then we all sit together. Unfortunately, the person who pioneered is no more, Professor Onaka from Japan. And subsequently, of course, nothing happened. But this is a great beginning where Japan came forward to have our own, for all Asian countries to have our own accreditation accord. Subsequently, what happened is we had several collaborations with the elite universities in Japan. And what I have observed, we in India, we fight for a lab space. All our researchers find I don't have enough space. All our universities and elite institutions fight for lab space to do cutting edge research. When I visited some of the universities in Japan, I was really surprised. In a very small, even a mezzanine floor, they have the world-class equipment doing cutting-edge research and publishing very good journals and patents, technology transfer. Because space is a very precious commodity in Japan, and they could do wonders even with the smallest space they have within their universities. So we have to learn something from the Japanese. It's not that it is the dedication and commitment of the researchers that can produce excellent research outcomes, and not the space and other thing. The other thing which I was really taken aback is the kind of quality and the humility. This is a country which I have learned, no matter however technologically advanced one may be, you can still be humble. And this is a great thing. The Japanese still retain the respect for the elderly population and the humility of the Japanese. However, great, we may be a Nobel laureate or all the degrees, but if you don't have a benevolent heart, there is no point in whatever success we have. And that is where Japanese stand for this humility at whatever level they may be. They are so humble and the clean populations. And later on, we took this collaboration to various levels, and I'm really happy. Today, it is not the household products which every child knows, the Japanese product, but now we are in the precision engineering area, where even the term nanotechnology, if I remember correctly, is coined by the Japanese professors, Japanese scientists. Even precision nanotechnology by the Japanese, the Taniguchi and Toyota, all these people have come out with precision technological advancement, which the whole world is, encouraged by all this. So I am extremely happy. But what we should do in India is the quality. India is also within the 75 years of independence, we have made rapid strides in science and technology. We can be proud of our achievement. However, the quality, we have to be very, very careful. Our products, if it has to get global market, we should learn the lessons from the Japanese so that we will have a very good market. So that is where I feel. And second thing, to inculcate the scientific temper among the youth of the country. India has its advantage of the youngest population. Maybe 29 is our average age of Indian population against maybe 48 for the Japanese. So this is a win-win situation. The wisdom of the elderly people and the enthusiasm and energy of the young Indian should, use it as an opportunity so that we can do wonders in addressing the challenges and issues of this world. But what we need in our country to have the power of observation for our children. We should not force our ambition by the parents or the peers into our younger ones. Let the younger one decide what they want in life, whatever the course of studies or career path they want to choose. Let them leave it to the younger ones, and we should use the power of observation. This is what India lost. India had this power of observation, power of contemplation and reflection when we had the great universities like Nalanda and Takshashila. We learned, we questioned, we debated, and we contemplated, and both the teacher and the student learned together. Somewhere we lost that, and I think this is the time to learn the lessons from the Japanese, and let us allow our younger ones to observe nature and learn from the nature so that they will become great scientists and engineers. I am extremely grateful to Sree Chitra Tirunal Institute of Medical Science and Technology for organizing this 7th India-Japan Technology Seminar. This is, of course, when we think about Sree Chitra, we are proud of your achievement, because this is the first time it has shown to the country that medical science and the engineering research can go together. I think this is an excellent example. They are the pioneer in telling us that we can learn with this, the medical fraternity and the engineering fraternity can work together and can solve many of the problems the common man is facing today. So that is the great thing about that. And as far as NIT Calicut is concerned, we are already collaborating with Chitra, Tirunal Institute for Medical Science and Technology. And I'm sure, and we are extremely grateful for the Embassy of Japan and all those who are behind this 7th Indo-Japan Seminar 2021. And I'm sure we will learn the lesson, and then we will look forward to have more healthy partnership between India and Japan in the days to come. Thank you for giving the opportunity. I am grateful to Dr. B.V. Mohanan and all the organizers, the scientists, the engineers, our delegates from Japan and India for giving me this opportunity. Thank you one and all. Jai Hind.
Thank you, sir, for the inspiring words. With great honor and respect, we welcome Mr. Yuji Nishikawa, Advisor for International Relations and Cooperation, Japan Science and Technology Agency, to deliver the special address. We welcome you, sir.
Hello? Can you hear me?
Yes, sir.
Okay. All right. Good afternoon or good evening, ladies and gentlemen. Distinguished guests, speakers, my dear friends, organizers, participants, ladies and gentlemen. Thank you very much for having me here today. I am delighted to participate in this seminar to commemorate the 70th anniversary of India-Japan diplomatic relations and also the 75th anniversary of the independence of India. I really praise the effort of India JSPS Alumni Association, especially Professor Shakti Kumar, for their successful planning and implementation of this great program. In the past, I also participated in IJAA's annual meet in India in the year 2016 and 2017. And I was amazed that it was so well organized. But this time, it is even better. Now, looking back the history of Japan. Japan has a long relation with India. It started with Buddhism, which reached ancient Japan from India, somewhere in the 7th or 8th century. In the pre-war era, after the Meiji Restoration in 1868, Japan imported a large quantity of cotton from India, which contributed greatly to industrialization and modernization of Japan. In the post-war era, Japan imported a lot of iron ore from India, which helped greatly the reconstruction of Japan after the devastation by the World War II. And as a result, Japan became the second largest economy in the world in 1968, which is only 23 years after the end of the World War II. Japan maintained the number 2 position for 42 years until 2010. Now, India has made enormous growth and development in the 21st century, and is expected to be the third largest economy within a decade after US and China. I have been dealing with India for over 45 years, but in business field and also in promotion of science and technology. Based on my long association with India, I strongly believe that India and Japan can be the best partners in many fields, including science and technology. As our Director General, Dr. Kishi, told in the opening session yesterday, JST's collaboration with India, especially with DST, dates back to 2005. And in 2014, our Prime Minister Abe visited India and JST decided to open a liaison office in India. I was appointed and I moved to India and set up our India office in the year 2015. I stayed there for 3 years until 2018. During my time in India, JST started 3 joint research projects with DST in 2016 in the field of ICT, which involved IIT Delhi, IIT Bombay, Hyderabad, and from Japan, University of Tokyo and Kyushu University. In the same year, JST started another project called the SATREPS, jointly with JICA, Japan International Cooperation Agency, in the field of ICT-based agriculture, involving IIT Hyderabad and Nagoya Electric Works. Sorry, this is not agriculture. This is for the mitigation of traffic jam and the reduction of CO2 emission. In addition to those large-scale joint research projects, JST started an invitation program called the Sakura Science Program, that is SSP in short, for India in the year 2015. I still remember that a JST mission, including myself, visited the DST in January 2015 and requested their cooperation for our SSP. At that time, DST instantly agreed to send top 60 high school students out of their most prestigious program called Inspire Manak. Since then, DST every year sends 60 bright and talented students selected from all over India by their Inspire program. Those students visit top Japanese universities and research institutes and meet students and researchers of Japan. The highlight of their visit is to meet some top Japanese scientists, including Japanese Nobel laureates and even the first official Japanese astronaut, Dr. Mori Mamoru. Such rare and invaluable opportunities will surely give a strong impact and motivate the young hearts and minds of India. I hope those promising youngsters of India will long remember Japan as well as the peers and friends they met in Japan. I also hope the friendship built during their visit to Japan will lead to future collaboration and cooperation between the two countries. As for the Sakura Science Program, I also should mention one important thing about youngsters of India. I usually accompany those students from India to various places, including Japanese high schools. I found that many Japanese high school students and teachers are amazed at the talent and get very good influence from the visitors from India. Generally speaking, people of India and Japan have something in common, but also something quite opposite. If we get together and work together, we can learn a lot from each other and influence each other a lot for the better. JST wants to expand and strengthen people-to-people relation, cooperation, and network through our Sakura Science Program. During my 3 years in India, I visited 45 cities in India and visited many schools and universities research institutes of India, such as 13 IITs, 5 ISAs, Triple ITDM, Jabalpur, University of Delhi, Bombay, Bangalore, Mysore, and many more. I also visited 13 World Heritage Sites all over India. And I found India has not only the attractive sites and places to visit, but also so many talented and attractive people to associate with and work with. So to end my speech, let me say, I'm strongly convinced that and hope that the people of our two peace-loving and hard-working countries will work together and contribute to the promotion of science, technology, and innovation, and also to steady and sustainable development of the whole world. Thank you very much, and let us keep in touch.
Thank you, sir, for the inspiring words. With great respect, we invite Mr. Kurihara Kiyoshi, Deputy Director, Director, Science and Technology Policy Bureau, Ministry of Education, Culture, Sports, Science and Technology, Japan, to address the gathering. We invite you, sir.
Thank you for introducing me. Can you hear me? Is it audible?
Yes, sir. Yes, sir.
Namaste. Good evening. This is Kiyoshi Kurihara in the Ministry of Education and Science in Japan. I'm very honored to be able to have a word in this valedictory session. Professor Saktikumar Sensei, Counselor Dr. Usha Dixit, thank you for inviting me for this session. And Dr. Ujiwara-san, we could not meet under this COVID-19 situation in Delhi this year, and I was sent back to Japan in July after my COVID-19 infection, and I am glad to see you here. And Dr. Mande, Professor Nair, Professor Krishna, Dr. John, on behalf of the government of Japan, I'd like to express my sincere gratitude for your active contribution to Japan-India Science and Technology Cooperation. I was at the embassy in Delhi, Embassy of Japan in Delhi until July this year. And I worked as the first secretary for science and technology under Ambassador Mr. Suzuki, His Excellency Ambassador Mr. Suzuki. Now I'm back in Japan and I'm starting to work at the Ministry of Education and Science in Japan. JSPS and JST are the organization under the Ministry of Education and Science of Japan. As Mr. Nishikawa of JST introduced, we are implementing various bilateral cooperation programs between Japan and India by JST and by JSPS. We strongly support the international activities of the JST, JSPS, and also JSPS Alumni Association. As you can see from yesterday's and today's workshop presentations, Japan has the world's most state-of-the-art researches, advanced researches, and we have been making many leading research results and innovation results. Japan has 21 Nobel laureates, and in this 21st century, Japan have the second largest number of Nobel laureates in the world in the field of science. I am now in the Science and Technology Policy Bureau in Ministry of Education and Science in Japan. On a week ago, 26th November this year, the government of Japan approved supplementary budget for new research project. Our new $700 million, $700 million US dollars budget for Moonshot program has been approved. This is one of the world's largest scientific investment. This Japan Moonshot program, this is the governmental strategic research, have 7 ambitious goals in science and technology areas. We are aiming for great result in quantum computers, artificial intelligence, AI, and biological networks in human bodies, and disease prevention and recycling of natural resources. Also, there are various research universities in Japan which introduced yesterday and today's workshop. There are many alumni in India, and Ministry of Education and Science of Japan supports them. Please search in the website some news articles last month. The government of Japan has decided to invest 100 billion US dollar in top universities in Japan. The Japanese government has decided to invest 100 billion dollars. Our new research fund of equivalent to 100 billion dollars, equivalent to the 10 trillion yen, 100 billion dollars will be distributed, and those huge funds will be managed in the stock market, and Japan's universities will build more cutting-edge research facilities and to invite faculties from all over the world. It will be permanently improved by the profit of this new huge $100 billion fund. Japan is making investment to have several world-class universities alongside the UK universities and the United States beyond China, Singapore, and Australia. Please continue to study in Japan and come to Japan and continue to contribute with Japan and collaborate with Japan. The active presence of this IJAA Alumni Association is outstanding in order to move forward our cooperation, Japan and India, Science and Technology Cooperation. I would like to once again to thank you for your effort and thank you for providing invaluable opportunity for such interaction and wish you all a most successful activity. Thank you very much.
Thank you, sir, for the encouraging words. With immense pleasure, we invite Dr. Ujjwala Turkey, Scientist, International Collaboration Division, Department of Science and Technology, New Delhi, to address the gathering. We invite you, ma'am.
Respected Dr. Kailash Manda, Professor G.M. Nair, Professor Prasad Krishna, Mr. Kurihara Kiyoshi, Mr. Eugene Nishikawa, and Dr. Annie John, my colleagues from India and Japan. I am very happy to get this opportunity to address you all in this special occasion of Nobel Laureate Science and Technology Seminar Series. These 2 days have given us all an enriching experience with lectures and keynote speeches from dignitaries covering a wide range of scientific topics. India-Japan Cooperative Science Program was initiated in 1993 to promote bilateral scientific collaboration between India and Japan. Program has various instruments for strengthening research collaboration between the two countries, such as funding joint research projects, joint seminars, visits of scientists or researchers, scholars and doctoral and postdoctoral students, and joint lecture series. Since school and college students are also participating, I would like to share some of the important programs we are having under India-Japan collaboration. We are encouraging youngsters, especially the research scholars, to carry out their research work with more exposure. They are actively participating in the Asian Science Camps, HOPE meetings, where they are interacting with the Nobel laureates and others Asian science luminaries. The Asian Camp, organized by Japan Science and Technology Agency, is aimed at enlightening the science talented youths through discussion and dialogue with top scholars, including Nobel laureates in the world, and to promote the international friendship and cooperation among the best young students of the next generation in Asia. Around 20 Indian students who are awardees of Kishore Vaigyanik Protsahan Yojana and Inspire Fellows are selected for participation in the Asian Science Camp every year, which is hosted by different Asian countries. And one of the camp was hosted by India at IISc Bangalore 2016. So every year, around 9 inspired PhD scholars from India are participating in HOPE meeting hosted by JSPS, and they are interacting with Nobel laureates. Mr. Nishikawa has already briefed about Sakura Science Program. So under the Sakura Science Program, till date, around 655 students and supervisors visited Japan. Because every year around 40 students selected by our department who are Inspire Scholarship Awardees, they are being deputed to Japan for participation in this exchange program. Very briefly, I want to tell about Inspire. Many of you must be knowing about it, but just a single sentence, that this innovation of science pursuit for Inspire research is an innovative program sponsored and managed by the Department of Science and Technology for attraction of talent of science. The basic objective of Inspire is to communicate to the youth of the country the excitements of creative pursuit of science, attract talent to the study of science at an early age, and thus build the required critical human resource pool for strengthening and expanding the science and technology system and R&D base. In addition, I would like to mention here about the National Children's Science Congress. This is specifically for our Japanese participants. This is a flagship program of Department of Science and Technology, Government of India. It is providing a platform to the young innovative minds of the children in the age group of 10 to 17 years to nurture their innovativeness, to find solution to societal problems of the country. And it has covered children from all corners of our country with diverse language and culture, school-going children or non-school goers, and also differently abled children. Over the last 28 years, it has covered diverse topics like weather and climate, energy, nutrition, biodiversity, land resources, etc. And thousands of children have been participating every year in this program, and many others as guides, resource persons, evaluators, organizers, etc. And they are getting involved with this unique program and its year-long activities with an effort to inculcate scientific temperament among the children. Professor Prasad Krishna has also been said about the scientific temper among the students. In the national event, students from ASEAN and Gulf countries also participate with their projects. We may invite Japanese students in this program. Around 5 to 10 students with 2 teachers may participate. If Japan has such activities for school students, they may invite Indian students also on reciprocity matter. With the concerted efforts from JSPS, JST, Sri Chitra Tirunal Institute, Embassy of India and Japan, and DST, the event was a success in these tough times of COVID pandemic. I congratulate each one of them. Thank you. Jai Hind.
Thank you, ma'am. Now it's time for the much awaited results of the poster presentation. To declare the results, may I invite Dr. Annie John, ICMR Emeritus Scientist, University of Kerala, and Joint Secretary, Indian JSPS Alumni Association.
A very good evening to all of you. I have been given the privilege of announcing the results of the students' presentation conducted at the Nobel Laureate Science and Technology Seminar Series, together with the 11th India-Japan Science and Technology Seminar. So let me get on to my job. Student presentations were conducted on day 1 and day 2. So the awards go accordingly. So on day 1, I'm taking the usual round of announcing the results. I'm talking about the 3rd positions. On day 1, 3 students have been awarded. One is Girishma B.N. Second is Anjida Ramakrishnan. Third is Padma Satya Muthukrishnan. So 3 people have taken up the 3rd position. So the 2nd position goes to 2 students, Gayatri Sundar and Amarda Nadarajan. And the 1st position goes to Devashmita Dubey. The certificates of the awards will be posted to you shortly. Now going to day 2. The 3rd position goes to Niveda. Only one student has been awarded the 3rd position. 2nd position again goes to only 1 student. Mano Ranjana Ponraj. And the first position again goes to Joseph Xavier. So day two, we have only 3 students who have been awarded for position 1, 2, and 3. Day two. Only 3 students have been awarded for positions 1, 2, and 3. I think I've made a mistake. In day 2, only 3 students have been awarded for position 1, 2, and 3. And day 1, position 3, 3 students have been awarded. For position 2, 2 students have been awarded. And for position 1, only 1 student. The names have been announced and the certificates will be posted to you shortly.
Thank you, ma'am. Congratulations to all the winners. We have come to the close of the valedictory function. I also request Dr. Annie John to propose the vote of thanks.
Once again, a very good evening to one and all. I thank the organizers for giving me this opportunity to say the vote of thanks. Respected Chief Guest, Professor G.M. Nair, President, Kerala Academy of Sciences, Dr. Prasad Krishna, Director, Indian Institute of Technology, Calicut, Mr. Kurihara Kiyoshi, Deputy Director, Science and Technology Bureau, Ministry of Education, Culture, Sports, Science and Technology, Japan, MEXT, Mr. Yuji Nishikawa, Advisor, International Relations and Cooperation, Japan, Science and Technology, Japan. Dr. Ujwala Turkey, International Collaboration Division, Department of Science and Technology, India, and other distinguished guests, ladies and gentlemen. The Nobel Laureate Science and Technology Seminar Series was all about celebrating the 70th anniversary of diplomatic relations between India and Japan. This was organized under the Azadi Ka Amrit Mahotsav, an initiative of Government of India to commemorate 75 years of progress in socio-cultural, political, and economic fronts. We are here this evening at the valedictory function of Nobel Laureate Science and Technology Seminar Series, organized under the auspices of the 11th India-Japan Science and Technology Seminar. The series started off by Professor Tasuko Onjo's address, followed by 2 days of plenary lectures and student presentations. It has been an invigorating experience in light of the current pandemic situation. On a personal note, and on behalf of Indian JSPS Alumni Association, I would like to thank Dr. Professor G.M. Nair for delivering the presidential address. The Kerala Academy of Sciences has played a key role in enabling science for society at the state level. I would also like to place on record our gratitude towards Dr. Prasad Krishna for his inspiring valedictory address. He has, by word and action, exemplified what all scholars should aspire to. It is an honor to accord our thanks to Mr. Kurihara Kiyoshi, Deputy Director, Science and Technology Bureau, MEXT, and Mr. Yuji Nishikawa, Advisor, to International Relations and Cooperation, Japan Science and Technology, Japan, for the ongoing and continued support and cooperation in the international sphere of science and technology. The last 70 years of official diplomatic relations lay a strong foundation to future endeavors. Dr. Ujwala Turkey is especially acknowledged for the strong support and hand-holding during the planning stages and implementation of this illustrious program. Department of Science and Technology, as our parent, has been a proud supporter of our activities. I, on behalf of the Indian JSPS Alumni Association, organizers and participants, gratefully acknowledge the Embassy of India in Japan, Embassy of Japan in India, JSPS, MEXT, JST, DST, and Sree Chitra Tirunal Institute for Medical Sciences and Technology for all support and sponsorship that has made today possible. We look forward to working with all stakeholders to enable future cooperation of science and technology filed at all levels. Indian JSPS Alumni Association wishes to place on record its grateful thanks for the cooperation of all partner organizations in the science and technology field for their enthusiastic support. The organizers wish to accord thanks to technical help from computer section, medical illustration section, and security services for smooth conduct of this two-day session. Science is the future, and our future lies in the schools of today. We wish all the students in participating educational organizations a successful science-based future. Thank you, and season's greetings to one and all. Arigatou gozaimasu. Konbanwa. Jai Hind.
Thank you all. Thank you.
The end of a story is the new beginning for many others. On this note, let's conclude the 11th India-Japan Science and Technology Seminar Series with great satisfaction. We are expecting a wholehearted participation from each one of you in all the future events of the Indian JSPS Alumni Association. May all that is beautiful and meaningful bring you joy this season and throughout the coming year. Stay safe and stay healthy. Thank you. Arigatou gozaimasu.